Yeah, it's side by side.
Right, to see what you got for the day.
Yeah. Um, starts at 9. Yeah, why not? For a little while. Go by yesterday's rules. Well, she said, let this up for a little while and then at some point put up the other one.
I mean, yeah, it's doesn't matter. It's fine. I just buy something.
She's got my text too. All right, she can use all caps like yesterday. Unmuted. It's like she goes, oh, okay. So I can mute both of them then, right? No.
Audio test 1 2.
And then you- can I hit that?
Audio test 1 2.
I hope everybody's ready for another exciting day of really remarkable science here. And I have the distinct pleasure of introducing Dr. Michael Schuler, who is, and this is a real mouthful, Eckert Professor of Engineering Emeritus in the Main Department of Biomedical Engineering and in the Smith School of Chemical and Biomolecular Engineering. You have to write that all in one piece of paper. That's hard. So he's also, more importantly, the CEO and president of Hespera, a company founded to implement the body on a chip system. I think in this meeting so far, we've seen some really great presentations from Hespera from people that work for him. And his research included body on a chip testing for pharmaceuticals for toxicity and efficacy, creation of production systems for relating genome to physiology. He's, more importantly, elected to the National Academy of Engineering and the American Academy of Arts and Science, and a fellow of numerous professional societies. So I think he's really a pioneer in this area. His company has done some really exciting research, and I think it's a real privilege to have him giving an opening talk here, and a privilege for me to introduce him. So thank you for coming. And I'll give you the stage. Yeah.
Well, thank you very much. I certainly appreciate the introduction. We're going to talk, I think, about body on a chip and the potential transform drug development. And I am both, associated with Hesperos, and I'm emeritus at Cornell, but I still have an active research program at Cornell. And outlines a little bit of personal history. I'm going to emphasize physiologically relevant multi-organ systems, the interaction between organs that occurs. And a particularly important theme, I think, for all of us in the MPS is the potential to measure human efficacy and toxicity simultaneously in preclinical trials. And I may speak a little bit on the maturation of the field and possible paths forward. So a little bit about motivation. I started thinking about this back in 1989. I was on sabbatical at the University of Wisconsin. Most of my work at that time had been up into bioprocess engineering. My oldest daughter has Down syndrome. My wife wanted to cure Down syndrome, and I didn't think I could do that. But one thing we did recognize, and we've been told by physicians, sometimes they were reluctant to prescribe particular medications for people with Down syndrome because of the 21st, they had 3 copies of the 21st chromosome. And since the drugs had not been tested on them, it was unclear sometimes whether it was safe and efficacious, even though it had been approved by FDA. So one of the thoughts here would be, could we develop a system which allow a human surrogate to be used to test for drugs? And also, toxicology is very important too, and I'm not going to talk much about that here, but chemical toxicology, environmental toxicology, foods, cosmetics are all important areas potentially for the development of this technology. But I'm going to focus on preclinical drug development. And the question we're going to ask is, can we combine physiologically based pharmacokinetic models with in vitro mimics of organs to predict human response? And do this particularly, as I said, in preclinical trials. So the beginning was 1989. We had this concept. The first graduate student I had to work on the project, Lisa Sweeney, began in that spring. And we began working with John Babish over at Cornell's Veterinary College. And the first model we published on was this article in 1995. It was a macroscale model. We used little tubings and little flasks connected together, but basically to emulate the body based on a PBK. And I'm going to show a little bit more on that in a second. We did this for rodents, and we were interested in naphthalene toxicity. We did about 9 more papers using this macroscale system. But in 1998, Greg Baxter joined Cornell's nanofabrication facility. And he met with me the second day he was on campus, and we talked about taking this kind of system and going from the macroscale system to make it a microscale system. The advantages of the microscale system would be in terms of reduced cost and improved throughput, and that was the main motivation. The first description of such a device was in this little article that was in the Proceedings of SPE in 2001, But really, the 2004, these series of 3 papers were the ones I think that established this system and potentially its usefulness. It was a paper where we looked at, again, toxicity of naphthalene, liver, lung, and fat. And I might add that fat's an organ that's very important that is usually left out. Most of our studies leave it out too. But in terms of the dynamics of many of these drugs, which are lipophilic, is an extremely important component. With naphthalene, when we first started on the study, it was unknown what the reactive metabolite was that naphthalene formed, which was toxic. And why did mice, well, why were mice 10 times more sensitive to naphthalene than rats? So we used this system, and by the time we get done, and other people have found things in the time we were doing this, we were able to show why rats and mice differ, because we had the model and we had the PBPK. We showed that the difference really came from not differences in enzyme levels, but just in the presentation of drugs and the ability to regenerate the compounds, glutathione compounds, which would be protective for the lung, and the timing between rats and mice were different. And we were able to show again how fat modulated the presentation and affected the overall presentation. This led to recognition in Newsweek. They had a October issue which focused on what they thought were big new ideas. It's one of the new ideas that they had. They're the ones who came up with the body on a chip term. We started building systems with both barrier tissues integrated with internal organs, 2009. 2009, it was a paper published from a Chinese group that used the term human on a chip. Now, up to this point, it may not be easy to recognize. There's a lot of resistance in toxicology and much more in pharmacology to any of those terms. So we usually use the term cell culture analog to start with. And that was less offensive to many of those people. But it was a human on chip was a term I was not brave enough to use back then. But once it was used, it was okay. I'm going to talk about our pumpless system. Up until 2010, all our work was with an external pump. The pumpless system has a lot of advantages I'm going to describe later on, and everything else we've done has been on the pumpless system. Also, 2010 was obviously a watershed year for the field as a whole. That's when Don Engberg's group came out with a paper, basically organ on a chip, and that had a significant impact on recognition for the field and funding for the field. And then Louise Marks' group had a paper that was the basis of the forming tissue use. He told me yesterday it was 2010 rather than 2012 it was formed, but it was the paper was 2010 on a chip-based multi-organoid system. In 2015, I'd done a little work with Jay Hickman before this, but Jay and I agreed to activate Hespros to become a body on a chip company. And 2016 was really the first paper based on the work done with Hespros, taking kind of the multi-organ system we had, but combining it with Jay's expertise in terms of functional measurements, particularly electrical and mechanical forces that are being generated by the systems. Also in 2016, we did a paper on the side actually, demonstrating a 13 organ system. And the importance of that was not so much the system itself, but we could build a system for 7 days, 13 organs, physiologically, realistically scaled, and operate successfully. And we built the first blood-brain barrier model with authentic barrier properties. That's probably the more important paper then. The one problem with the pumpless system we had originally is that it was reciprocating flow. Flow goes down one direction, comes back the other direction. Not so good for endothelial cells. 2018, we published a paper showing how you use a pumpless system, but you get unidirectional flow through the device. The next two papers, well, three papers really largely associated with my work with Hespros. A 2019 paper, Chris McAleer is sitting in the audience, and we'll be talking in the next session. demonstrating that in human preclinical trials, it was possible to determine both drug efficacy and toxicity. And I think that's a theme I'll come back to again. The inclusion of functional innate immune system with circulating monocytes, we published in 2020. And what's important there, the pump system I'm going to talk about, is the fact that you can circulate cells without damaging cells. And I think that's an important point. In 2021, there was a paper published based on the Hespro system. I wasn't involved in this work directly, but the Hespro system was used by a small company called True North, which got out by another company which got bought out by Sanofi. But Sanofi took a drug that they were repurposing, using the data from the Hespro system for an IND, and began phase 2 clinical trials over a year ago on that. And I think that's the first case of using a microphysiological system as a basis for an IND. So the concept here that we began with is using mathematical models. Now, physiological-based pharmacokinetic models were developed in the late 60s, early 70s by Bob Dedrick, Ken Bischoff, using kind of a chemical engineering approach. Divide the body into different organ compartments. You treat the organ compartments as either chemical reactors, absorbers, or holding tanks. And from that, you can predict the distribution of the drug throughout the system, time-dependent distribution of the drug throughout the system. And it was done largely originally for chemotherapeutic drugs to try to maximize the dose at the tissue of interest and minimize dose elsewhere, prevent side effects. You can also have pharmacodynamic models which says if the drug concentration is this at this organ, this is the response. And again, you do this not only for the parental compound, but for the metabolites. So we wanted to build a system guided by the PBK. And it's a little bit different than just being a multi-organ system. So multi-organ system, when it's guided by the PBK, where you get the compartments to communicate with each other in a realistic time-dependent manner. And sometimes to build a totally realistic model is difficult, especially if you want to get 256 organs in it, which is not really possible. But what the PBK does of the device allows you to get the fundamental parameters that you can put into a model for a human or animal and predict what's going to happen in those systems. Okay, one other thing that's a little bit subtle here that I think sometimes is neglected is if you're going to model the whole body, you need to include all the tissues, at least all the fluid in the system, because otherwise you can't get the dynamics correct. In our case, the other tissue compartments are often a fluid reservoir, which you need to run the system anyway. But the other compartments is very important. And particularly for compounds where there's a fairly rapid exchange between the blood and interstitial fluid, the ratio of fluid is fairly large compared to the cell ratio. You can do this. It's much more difficult with the blood to cell ratio doing something which is realistic. But it's very important to have a low volume system so you can see the effects of metabolites on the other organs. So this is basically what we have. We have a system where we have the PBK, we have a physical system, we make predictions about what's going to happen. And some of the assumptions here is one, the organ volume ratios in the body are emulated in the device. More importantly, the fluid residence time per organ translates into fluid residence time per organ chamber. And why is that important? So you go back to chemical reaction engineering theory. It doesn't make any difference what size the reactor is. It could be 10,000 liters, it could be 100 microliters. As long as the residence time, which is the volume of the reactor divided by the flow rate into the reactor, you get the same reactants form, the same degree of conversion, same reactants form. Now, there's some subtleties in terms of absorption stuff on surfaces that you need to worry about as you change size. But in principle, that's our basis for scaling from a human to this device. Now, there's a lot more issues in scaling if you really want to go into it. And John Wixel over here, he could explain all the different possibilities. But for us, this made the most sense in terms of doing the scaling. We built a system where the fluid dynamics are well behaved. So we a passive fluid flow, we didn't need active valves. We could predict what's going to happen with the distribution of flow fairly easily. One challenge here is always the chip has to have cells which will grow in a common recirculating medium. And that is potentially a challenge, but we've always been able to find a way to do that. Sometimes it takes, you may mix 3 media, and I'll talk a little bit more about that later on. And again, this is where Jay Hickman's group also brings a lot of expertise, because a lot of it's not only the media, but how the media interacts with the surfaces, which is important. So we like serum-free media, and the company always uses serum-free media. In my lab, we sometimes use serum-containing medium. There's lots of advantages to serum-free medium, and particularly in terms of control over what's happening in the systems. Not that it's cheaper, it's actually more expensive to build a good serum-free media than using serum. But you again need to consider the whole system, and surface chemistry is like I said, important. There's a couple of papers here which describe that. So what are the criteria to emulate a PBPK? The first 4 I've listed are relatively easy to achieve. I mentioned residence time as important in terms of scaling. Also, these systems have flow in them, and it's very important the flow rate past the cells be within a physiologic range. If it's if you're a static system, many times the cells don't respond with the same physiology as they have in the body. They need some flow, but too much flow, of course, is detrimental. So it needs to be within a physiologic range, and that's one of the design parameters. The other thing, which is much more difficult to achieve, can be achieved, but much more difficult to achieve, is the ratio of liquid to cells and systems physiologic. You want a low volume system if you're going to look at the formation of metabolites and the action of metabolites in other organs. Now, there's a paper by from Mandy Esch's group at NIST in 2020 where she had a device that actually achieved a blood to cell ratio, which is physiologic. Most of the systems I'm showing you won't be fully physiologic that way, but there will be low volume systems so we can measure the effect of metabolites. The last point is the most difficult. It's the reason that these systems will never be perfect. And that's because once you take something out of the body, put it in a system like this, it's very hard to sustain the same physiology forever. But we're getting much, much better at getting tissue engineering constructs, which are much more mature. IPS cells, I think, are going to be ideal. I'll mention that at the very end. So there's been improvements that way. But again, the device need not be perfect to be useful. If I look at drug development, for many compounds, you're talking about 10, 11% of the drugs that are tested going into preclinical trial come out as approved useful products that are marketed. And if we can change that ratio from 11% to 33%, that has a tremendous impact economically. It now costs you presumably 1/3 less to develop the drugs. You're not spending clinical trials on something that's not going to work. You know that ahead of time. So society gains in potentially getting many more drugs and many more drugs at a cheaper cost. And that's one of the real potential benefits of this. So even though it may not be fully perfect, it can provide guidance, which is, I think, very important. Now, I'm not going to spend much time on this. Don Einberg had a nice paper on this, but basically, you can have engineered systems where you put a membrane down, you get the cells, you put them in the right positions, et cetera. You have organized, which are basically self-developing. Both can be useful. We generally use mostly engineered systems because we have much better control over what's happening. We do the measurements much more easily. We do use organoids in some cases. Don had this paper in 2000, I think it was 18, where he talked about some of the advantages of engineered systems versus organoid systems. They both have advantages and disadvantages. You need to be aware of what you want to do in terms of what you might choose. One of the things that's important, and again, that you have to always do, and the company always does, is run the system for 24 hours without the cells to understand your drug losses through absorption and adsorption. And from that, then you make some prediction about what's going to happen. What are the doses you really need to have the dosage in the recirculating fluid? And it doesn't mean solubility because you can have proteins in there and they'll absorb to the proteins, et cetera. But what's going to be the concentration in recirculating fluid is going to be presented to the cells. And again, for some compounds, you don't really have much losses. For some lipophilic compounds, you can have significant losses. So again, you need to do this kind of experiment to anticipate what's going to happen. Again, this is a case where on the left-hand side, the model predicted from that data what we thought should be the concentration. And then when you compare it to the measure values, they come out basically the same. So in 2010, we published a paper on the pumpless system. And it's not only pumpless, it's self-contained. And sometimes people talk about having a little chip and then they have a whole bench full of equipment to support it. This is a device that can act by itself. It doesn't, you don't have to take it apart, etc. It can operate for 28 days by itself. So the basic idea of the pumpless system is you have the system and you have two reservoirs up here and here. And the reservoirs act against the other tissues compartment. You tilt this flow by gravity down here, driven by this degree at which you tilt. And then after you do that, you flip it, it comes back the other direction. So you do get reciprocating flow, and we'll talk about consequences there. But for most cell systems, particularly internal organ, reciprocating flow is acceptable. You can predict the flow rate very easily. The solid line is the Hagen-Purcell equation. The dots are our measurements of the flow rate. So over a significant factor of heights here, we can predict the flow rate through the system fairly precisely. The gravity-induced flow is very important. One of the biggest problems we had originally, operating systems for a long period of time, is the formation of gas bubbles, and the gas bubbles plugging up the microchannels. The gravity-induced flow gets rid of most of this bubble problem. Also, it's much more efficient to assemble and operate. Now, one thing I want to point out, and just as in general for all these devices, is if I ask you to go 28 days is our goal here because that matches what happens in a lot of drug development. If I asked you to go 28 days without eating, drinking, or going to the bathroom, you would have problems. Okay, this device would have problems too. So we try to emulate that in a crude way by removing a portion of the fluid from the other tissues chamber each day and replacing it with fresh fluid. The fresh fluid will replace the nutrients that have been consumed, remove some of the waste products. And we do that for all these systems, and we can operate them for 28 days or longer in this kind of system. So there's a lot of advantages here. Again, we got rid of the external pumps, but allows continuous recirculation, eliminates the tubing. Before, if we had external pump, we often had tubing. If we have tubing, that's a source for non-specific absorption. You want to reduce the non-specific absorption as much as possible. It's a low volume system, and it's particularly you get rid of extra volume by getting rid of the pump a lot of times. That means you have a system that allows you to evaluate both the parent and the metabolites. And that's very important because most of the compounds we're interested in looking at is not the parental compound, but it's the metabolites formed by the parental compound, which are important. The main reason we did this, it was robust, you can operate for a long time, 28 days, you get no gas bubbles, no moving parts to fail, so you have a high success rate. We tried doing this with a pump system to operate for 28 days, you lose almost all the systems before you got to 28 days. It's self-contained. Again, once you put the system together, you remove some sample from the reservoir, but that's about it. You remove sample, the sample you remove to emulate the waste removal and feeding, that sample becomes something you put in HPLC and mass spec. Okay. You don't have to take it apart because we're also going to talk about these functional measurements and their electrical and optical measurement. You don't have to take the system apart to measure it. You run many units in parallel. A rocker platform is pretty cheap. You can run, run 9 units on a platform. You run 4 platforms on a rocker if you want. You can run a lot of units on such a system. The other thing which is important is that Many times you want to look at different cells which circulate in the system. If you have an external pump in the system, almost always that pump will damage the cells or cause them to be activated when you don't want them activated. This facilitates recirculation primarily of immune cells and circulating tumor cells, the ones we've been interested in. I'm not going to show any data today on circulating tumor cells. It's something we're doing right now, but those experiments are ongoing. And the last thing, Jay Sung was the student working on this, and he had a vested interest in doing this because this decreased his time in the lab considerably. He put together 24 systems much more quickly than he could before. In about 1/8 the time before, he can get all these systems together than they could with the pump system. So the other things we've been doing to make the pumpless system work better, self-contained system. One is constructs which are more and more authentic. We do the integration of barrier tissues, and I mentioned the unidirectional flow, which we'll talk about a little bit. Again, the serum-free medium control surface chemistry is very important in this. Now I might add again that this is a modular system. Some tissues may take 2 days growing and to maturity, you can put them in the system. Others may require 21 days to grow to the point of maturity, you can put them in the system. You grow them to maturity and put them together in the system when they're mature. It also makes it a little easier to find a medium because you're not trying to grow the cells any longer, you're trying to maintain the mature metabolically active phenotype. So how do we monitor these systems? Well, we look at transepithelial electrical resistance for the barrier tissues, and we can do that in a sustained way. All the devices we make have optical access. So for example, if you have a fluorescence compound, you can look at the distribution of fluorescence compound in the system, et cetera. But we do the sample from the reservoirs, like I said, and that provides enough material to go do HPLC mass spec on, get a chemical measurement, not only of the parental compound, but metabolites or biomarkers that we need. But again, the functional readouts, I think, are even more important. And I'll talk about that, but this is really what Jay's group has contributed. The electrical activity and force generation of neurons and cardiac tissue is particularly important. Neuromuscular junction is one thing that we do a lot of work with. The functional readouts are non-invasive. It facilitates chronic monitoring. Again, if I'm going to operate for 28 days, I'm going to be looking at responses that are, the acute response requires a certain dose. The chronic responses, you usually have less dose, will give you a certain effect over a long period of time. If I'm going to emulate what's happens, you're going to take a drug every second day, I can do that with this kind of system. And it's a rapid, direct measurement of response. Sometimes, if you rely on cell viability, many times cell viability doesn't change, but the action of the cells change. In terms of response, you can see responses in terms of minutes, in terms of electrical activity and force generation many times. And it can be diagnostic, it can lead to a better understanding of underlying mechanisms also. So this is just an example. What I want to emphasize here is often people talk about 2D versus 3D when talking about microphysiological systems. I think it's much better to talk about organized and disorganized cultures. This is an example of a 2D organized culture. Now, if I take the cardiac cells and just plate them, I can't measure conduction velocity or anything about the electrical activity because it just gets dispersed. But if you control the surface chemistry, and lay it down like this U-tube shaped thing that's here, you can force the cells down here. And if you make this narrow enough, then the electrical activity is forced to follow this line. And that leads to the ability to measure things like conduction velocity, QT interval in the heart, et cetera. So although this is a 2D system, it's an organized 2D system and gives you real functional information. Again, this shows this. Again, I can stimulate here with electrical response, and I can just follow what's going to happen in the system. And it's you can, the change in conduction velocity in response to the drug can tell you a lot about the action of the drug. Or a chemical if you're looking at toxicity. Measuring force, you can use these cantilevers. And the way we do it now is largely a cantilever. The cells are going in the cantilever. They contract, cantilever bends. We measure the bending using basically a laser. And we look at the deflection of the cantilever. And from that, we do the back calculation to the forces generated by the cells. This is a little awkward sometimes to do on a large scale. So we've been moving towards a piezo-resistive method. I don't have any papers yet to describe this in any detail, but this is what we're working on right now. The piezo-resistive method would be even easier to implement than the one with the laser, because you have to move the laser around, ratchet it around to see all the cantilevers, etc. So this was first system, again, I think basically Hesperos put out. It was a 4-organ system. It had a liver compartment. Liver compartment can be 3D. You're looking at making your metabolites. And again, you have a reservoir here and a reservoir here, and they're going back and forth. Now, the neuronal part, the skeletal muscle part, the cardiac right here. The cardiac right here is electrical and mechanical. And you measure electrical activity and mechanical activity here. We're not measuring, we don't have the 3D cultures there. We have 2D cultures there. We're using these to measure the response of the body to particular metabolites. We're not looking at the generation of metabolites because we would need the full 3D structure to do that. But this is diagnostic in terms of the response. The liver is the one thing that we do have to worry about, the formation of metabolites, recirculation. Again, this is where if you take samples out of the thing, you put an HPLC, mass spec, you can measure the overall response. Okay. Now, one thing again is 28 days is a fairly standard operation for extended studies in preclinical trials, mostly with animals. We wanted to show that the system could operate 28 days and maintain a fairly stable baseline. And that's what this described in this paper. And because we have a stable baseline, we can look at the difference between acute and chronic responses to different drugs or chemicals or whatever you wish. So both efficacy and toxicity are important. This is a paper that Chris McAleer was the first author on. It was between Hesperos and Roche. And basically it was to evaluate the human cell-based in vitro system, both for on-target efficacy and off-target toxicity of parental compounds and their metabolite. And when done with 3 drugs, as an example, we'll look at tamoxifen and verapamil. And this is basic system. It was cancer, and we were literally looking at 2 different kinds of cancers, multi-drug resistant cancer and sensitive cancer cell line. And the liver and system, liver system, because we're looking at the formation of metabolites, that's important. And the cardiac was potentially in terms of side effects. In terms of drug side effects, and many that are important, cardiac and liver are 2 systems where drug side effects are really often very critical. This just shows tamoxifen, an anti-cancer chemotherapeutic agent. If we look at the sensitive cell line up here, you see there's a gradual, tamoxifen by itself is not toxic, is tamoxifen metabolites are toxic to the cells, kill the cells. 4-hydroxytamoxifen right here is the important one. You can see right here, for the sensitive cell line, as you increase, we see increased activity. With the drug-resistant cell line, this is a drug-resistant cell line because it overproduces P-glycoprotein. It's a pump protein. It prevents the input of the chemotherapeutic. If you have verapamil, verapamil suppresses the pump protein, allows the drug to enter, and then you see a big response to the drug. So you see the difference that way. You start to get a sense of the efficacy of the drug here. Also, you can look at side effects. These are cardiac side effects here. You're looking at beat frequency, conduction velocity, force generation right here. And again, the idea here is that there's very little change in viability. There's significant changes in terms of the functional response. And again, basically, if I look at the liver in the system, in terms of urea and Albumin production is fairly constant. There's not any sign of toxicity there. So the toxicity is specific towards the cancer. Okay. So can we ask the question, can we use the MPS to predict PKPD relationships? This is done jointly with AstraZeneca. I'm not going to go into much detail on this one. But the point here was, this was a drug which was confounding for them because the maximum drug effect was not driven by the peak drug concentration. There was a time factor. Drug was added, it was absorbed, then they had an effect. So there was a time can be a critical factor in that. That's often not uncovered until you do advanced preclinical trials. You'd like to know that ahead of time. This is the kind of system that can allow you to do that ahead of time. And we did those studies with terfidine. We did it with multiple species and showed that we can extrapolate between species. Fexofenadine, terfidine was withdrawn from the market because of cardiac side effects. Fexofenadine, you know, is Allegra, and that's the product of interest. And you can show, for example, there's two orders, you can't really see it. There's 2 orders of magnitude difference in the response here. Just the cardiac system, there's significant response. Once you put the liver in here, it metabolizes terfidine to fexofenadine and relieves the inhibition. So but if you're a person that has liver activity which is less active, then you have heart side effects. And again, one other thing that was in this paper, which I think is important, is that AstraZeneca had a compound, and they assessed it by their normal studies, not with flow or anything, quiescent studies. And they thought it was safe in terms of not having in vivo QT effects, up to 100 micromolar. But then again, the run in animal testing, particularly higher animals, they found that the QT interval increased by 22%, only 3 micromolar, which is unacceptable. They didn't discover that until fairly advanced preclinical studies. And the reason for that was it was forming a metabolite. But again, we showed that if they used the Hespro system, they would have found that 3 micromolar was in fact toxic ahead of time. So again, shows potentially the value using the system ahead of time. The other thing about this one paper that I didn't really emphasize is doing a lot of extrapolations. They were able to convert the data from the device into PK/PD relationships and make mathematical predictions. This is another paper, again, done largely by the group at Hespros, but done in conjunction with Hoffman and LaRoche, looking at recirculatory monocytes. And again, we wanted to look at tissue-specific damage and holistic pro-inflammatory response. But what was really key to making this system work was that you can maintain the system for 7 days, recirculate the monocytes, and you could do that without activation due to flow. Most of the time, first of all, a lot of the systems which would have pumps would kill the cells that were circulating. But even if you had a system which was general enough not to do that, often that movement would be enough that would kill the cells. Using a pumpless system allows us to do the recirculation in a way which is sufficiently general that we did not activate the monocytes due to flow. We did when we added chemicals, et cetera, to the system. And basically what we could do is show that we get the M2 phenotype and the M1 phenotype in the system. Again, we worked on some other recirculating cells. But again, I think the pumpless system, this design, is very helpful in working with cells when you're worried about inadvertent effects due to flow. Okay. Now the berry tissue organ work, and this is berry tissue. So we also have a lung model I'm going to mention at the end. This is mostly done at Cornell. The last 3 things I thought were done largely at Hespros. But Hespros has adopted a number of these things. Now we've done actually a lot of work on the GI tract. I'm not going to show you any data from the GI tract today. I'm going to concentrate on the blood-brain barrier. The, we worked with Sean Polachek and Eric Schuster at University of Wisconsin, and they had developed a way to take IPS cells and derive brain microvascular endothelial cells. And they had built a preliminary model with of the blood-brain barrier using the brain microvascular endothelial cells co-cultured with astrocytes on a porous membrane. We took that system and we built a blood-brain barrier. Now we had authentic residence time, same residence time as in your brain as in this model. What we have here is, and you probably can't see it because the slides aren't going to be large enough, but the astrocytes on one side, membrane in the middle, and the brain microendothelial cells on the bottom. Now you also notice this is indented from the flow. It's indented from the flow because the fluid effects can be significant. We put it back so the shear forces on the brain microendothelial cells were within the physiologic range. And if we did that and operated the system, we have, I think for the first time when we did this, authentic values, the sustained authentic values of the TER in the system. The TER for the human blood-brain barrier is somewhere between 1,500 and 8,000 ohm centimeters squared. And you see that we're up to 4,000. We're sustained around 3,000 for 10 days, a little bit longer. And you can see that the tight junctions are also measured here. So that we have a system which we think very effective in terms of emulating what's in the blood-brain barrier. You can also look at drug uptake to the blood-brain barrier model. And we were looking at 5 orders of magnitude here, difference with these compounds. And you see that there's a pretty good correlation between the log P, the permeability basically in the chip and in vivo. That this is, ideally be a 45-degree line. So we think we can build a blood-brain barrier, which is actually fairly authentic. Now, one thing I'm not going to go into because I don't want to take the time to do it. If you want to, you can look at this one paper. But basically, the rocket platform, before the flow goes down, flow goes back, and reciprocating flow. So we still have to have that flow like that. But we can rearrange the system. And again, Yi Wang was a very clever postdoc with me. Oh, sorry. Basically, this middle channel right there, you can have flow always the same direction if you do this right. And part of that's based on here, and I can't shine the pointer through the marker there, but basically there's those 2 circles there. There's 2 outlets and inlets in each of the chambers. When you flip it this way, the fluid goes on down, but there's enough fluid retained by capillary action. And then it acts as a passive valve, closes the valve. And by doing that, you can change direction of flow here. So you always get the flow going one way through the system. And again, that has a significant effect physiologically. This is unidirectional flow. You see the cells align, direction of flow, and then endothelial cells. Now, this is a model where we just had UVEC cells in the channel. I'm going to show you one result where we have multiple organs in there. The bidirectional flow, you don't see the orientation. You can see this measuring angular orientation, quite a bit different. It also has an effect in terms of the physiology of the cells. Now, when we put these systems together, we don't necessarily want them to grow any longer. In this case, you see we put them in here, and with the unidirectional flow, there's no more, the growth is minimized here. You get sustained growth in the system with bidirectional flow, and you have a little bit more growth when it's in a static system. So again, you can obtain a more physiological system by giving it the appropriate forces. We did this with a 3-organ system. That was just with the vascular. We did a 3-organ system looking at response to Tegophore and Uracil, which is a cancer treatment, colon cancer, bone marrow, and liver. And basically everything we saw in this system was consistent with the known response of to that drug. We've also built a breathable lung. Now, Don Engberg presented a breathable young lung yesterday, and did it many, many years before this. The difference here is this is in the context of a multi-organ system operated at the same time. But again, operating so you maintain an air-liquid interface moving back and forth takes some engineering design to get that to work right. We did this with lung, liver, and breast cancer. We looked at the treatment with one chemotherapeutic. The question we asked here is, would inhalation therapy be as effective or more effective than intravenous therapy? Potentially, inhalation therapy could be done as a home-based therapy. The intravenous therapy pretty much has to be done in a hospital setting. So it'd be a big advantage potentially to doing it with inhalation therapy. And what you can show is that the system is, in fact, we can do recirculation. Again, if you static treatment, you won't see any of this. You need also the movement that Don talked about in terms of seeing the appropriate response. But we did find that intravenous therapy resulted in a much greater killing of the cancer cells, 49% death versus 21% death for intravenous, and liver and lung death was insignificant. Okay, just mentioning again, increased complexity is possible. Again, this was just to show that you can put this system together, 13 organs, 3 external barriers, 2 internal barriers, 8 internal organs, everything's organized proportionally, except fat and muscle. And again, they were 1/12 their normal size to fit onto the chip. But otherwise, we could make this sustain for at least 7 days, etc. This again reminds me, we left fat out most of the chips I talked about. But fat can be really important in terms of overall dynamics here. So adding fat back in would be a good thing many times to do, particularly for any compound which is lipophilic. We can apply this to rare diseases to get work done with Virginia Smith at Hespros, applying it to a rare disease. And again, we think rare diseases is a opportunity for MPS systems to establish a toehold in terms of their ability to provide useful data in a regulatory setting. Myasthenia gravis is a chronic neuromuscular disease. Autoantibodies block this receptor. We look at a neuromuscular junction. Jay showed this slide yesterday, but basically, or something similar to this slide. But right here, you have the axon bodies, or the neuronal cells here. They form axons, and they have tiny channels. The cell can't go through, but the axons can penetrate. The axons penetrate to the muscle cells over here, innervate the muscle cells over here. So you get a neuromuscular junction system where this can respond to things on this side or this side, etc. You look at drug response. If we do this with this system as a model for this disease, we see the treatment with the antibody results in 3 different kinds of responses, all which are observed clinically. And so it does this in a way which can be used to test potential treatments, etc. Now, in terms of supporting clinical trials, this I was not involved with. This is a paper, and actually, Chris McAleer is going to talk, I think, later this morning about the system, this paper basically. But the data from this paper was used to provide efficacy data to gain regulatory approval again for this clinical trial for Sanofi for a repurposed drug. And the phase 2 clinical trials were for these 2 neuropathological diseases. But I think this is, again, the first example using MPS to gain regulatory approval. And I think that's an important step for the field. And again, Don Engberg talked yesterday about applying some of this to COVID-19, which would be another example of using MPS systems in terms of regulatory approval. And so I think drug efficacy can mean a lot in terms of regulatory evaluations. And again, you can start thinking about doing things for with MPS systems for groups which are not easy to do in terms of clinical trials of children, aged individuals. Ultimately, ultimate precision medicine is potential with these systems. You can design your clinical trials to target certain subpopulations, et cetera. In terms of further development, the things I show are things that people are doing right now. It's not that they're not doing it, but these are areas in which I think there's more and more emphasis on. There's going to be more and more sophisticated models being developed for the immune system, circulating tumor cells, reproductive tissue. The neurological disease is important. It's one that we're very interested in. But I think there's going to be, again, greater maturity in terms of its application in that area. More authentic tissue constructs is certainly something many people are working on. They can be incorporated. I'm going to talk a little bit more about IPS technology, but I think that's going to be an important technology for this. And again, personalized medicine, precision medicine is a way that we can ultimately go. I don't think we're really there yet, except for very specialized cases possibly. So IPS cells, I think IPS cells are particularly useful for MPS models. And that's because now you have a controlled cell source that replicates many of the advantages of primary cells, but no, without the variability. So now I can run, hundreds of experiments with basically the same cell base. And that's probably important in terms of really understanding what's happening in the system. I can do this to represent different genotypes. I get IPS cells from different populations. I could go back to the idea of Down syndrome in that. So we can mimic disease tissue as well as normal tissues in these models. And again, personalized systems. I won't spend much time on that. I think that's fairly obvious on that. And so in conclusions, basically, I think we can build physiologically relevant multi-organ systems with continuous recirculation, serum-free medium. And that's what we've shown, and I think that's desirable. The functional readouts, I think, are important because it gives you a way of looking at what's happening very rapidly to application of drugs or chemicals. If I'm looking at chemical toxicity, it's very important that way also. And it can be diagnostic. It can lead to even better understanding. In the heart, does the drug affect the electrical activity, the force generation, or both? I can start getting some insight that way. The pulmonary systems, I think, are very important. The barrier tissues, I think, can easily be incorporated with internal organs. And then I think for the field as a whole, looking for a toehold in the regulatory environment, I think looking at diseases, probably particularly rare diseases where there are not good animal models, and using drugs which already have been approved for human use, but not necessarily active in human use, looking at using the system for efficacy and those kinds of systems is actually a good model in terms of gaining a toehold in the regulatory environment, gaining confidence from the pharmaceutical industry that this can work. Also, in terms of things like foods, cosmetics, this also would apply as well. So again, this is support, and I just want to thank all that. Again, I might mention one thing about support, though. The first 20 years of my work in this, all the support had to come from NSF and the Army, because it would never get past NIH review at that point. It was really only after about 2010 that there was a reception at NIH for this kind of studies. We did submit to NIH, and you can get 3 good reviews and 1 bad review saying that it would never work, basically. So all our early work was supported by NSF and the Army. So with that, I'd like to, I can take questions at this time. If not, I appreciate your attention. So thank you.
Thank you very much, Mike. Wonderful keynote to get us started, an exciting second day. As it is a keynote lecture, we have not foreseen any questions for this. We have to digest this, I hope, with some coffee outside, and we have to move further with the program.
Very good.
I think it was a fantastic start. Thanks a lot again.
Well, thank you. I appreciate you.
All right, we're gonna get started. Larry, you want to come up? Want to pull the first presentation up?
And this is your pointer and your advance here. Okay.
All right. Well, first I want to say how nice it is to look out and see faces in real world, not on Zoom. This has been a great conference already so far. So it's been great to have this. We're based in New Orleans, so it's fun to have this in our backyard. And certainly want to say thank you to NCATS for putting this on and Hopkins and CAT for all the work that they've done setting it up. What I'm going to be talking about today is a case study in chemotherapy-induced peripheral neuropathy using one of our platforms, which we call a nerve on a chip. First, about the company and our team. We were founded in 2014. We've licensed our platforms from Tulane University and Johns Hopkins. I'll talk a little bit more about that. And then we're a growing team of 27. I'll say in the front, if anybody's looking for a job, we're currently hiring right now. So check out our website or catch one of our team after this. The two platforms that we utilize, I'll primarily be talking about the nerve sim model today, which is our nerve on a chip. That's used for neurotoxicity. Obviously, the case study I'll be sharing, but increasingly we're moving into the drug discovery side. I think the entire field sees that the impact there is potentially larger, and really, we want to capture that. And I think that's where the impact is. Obviously, Hesperos has now put in data into an IND filing. The reality of this is starting to show up. We also, towards that end, can work in rare diseases, ALS being one of them. The other platform is our brain sim platform. That is a CNS model. And you saw, or, if you were in the session, Johns Hopkins is where we licensed that. And they presented some data on oligodendrocyte precursor populations and ultimately myelination in that system. So it's a really elegant system. Background on peripheral nervous system, not something everybody here is maybe familiar with. It's basically all of the nerves outside of the brain and spinal cord. And these are really important, but frankly, understudied. When you look at, basically how this functions, you have the dorsal root ganglia and you have motor neurons that synapse either onto the skin, skeletal muscle, or the enteric nervous system. And we're going to be focusing today on the dorsal root ganglia as that interface between the sensory nervous system and the spinal cord. This is implicated in a big way in chemotherapy-induced peripheral neuropathy. Unfortunately, over 2/3 of patients who undergo chemotherapeutic treatments end up with a peripheral neuropathy. And this is not just a short-term problem. A little under 1/3 of them still have these issues after 6 months. And these are big issues. They affect mobility, chronic pain. And you can see, because these are such a big deal, they actually change the way patients are treated. As you see the numbers here, it can lead to kind of decreasing the potential efficacy by reducing the dose, and even some patients just stop altogether. And this has become a bigger focus within the pharmaceutical industry recently. And what we went to do here in our qualification study, basically these chemotherapy neurotoxicities kind of manifest in a couple of different ways. And we chose 4 of the primary classes that acted at different mechanisms. One of those is platinum compounds, oxaliplatin in particular. You can see where they are affected here. I'll kind of go through how we demonstrated that, so I won't spend too much time here. Bortezomib, a little bit of overlap there. Of course, there's overlap with all of them. And then we picked the taxanes to start looking kind of at the other 2 as well, and vincristine to cover the spectrum of these toxic mechanisms. Just background on the nerve sim itself. We took a tissue engineering approach to build a 3-dimensional scaffolding system. I'll show you a really nice video to help you understand what that looks like. But if you look at the image in the top right, in green there, we have beta-3 tubulin, and you can see we have this really nice polarized neural fiber tract. and having this 3D interaction between Schwann cells and sensory neurons, motor neurons, whichever the case may be for a given application, we get this really nice 3D compact myelination that you can see in the images below that. The other thing that we're able to see here, another clinically relevant metric, is population level electrophysiology. So we're not looking at the single cell level. We're really looking at a summation of all of the tens of thousands of neurons, axons that we're recording from, and look at that compound action potential. And then most importantly, the conduction velocity. And this is really one of the primary metrics that a neurologist would look at. And as I've already described, right, we're able to kind of pick and choose the cell types that we want in corresponding with the application that we're looking at. We take those in a very defined ratio and density, and we form them into spheroids to basically get the basis of those cell-to-cell interactions. Then we engineer that into the three-dimensional scaffolding system that is the nerve sim or nerve on a chip. We're able to isolate the neurons in this circular region of a keyhole and induce, constrain, define the both neurite outgrowth and glial migration, where we're getting tens to 50,000s of axons. We can come in after introducing a drug, look at what changes functionally, what happens to that conduction velocity, what happens to that compound action potential. And after we know functionally we see this decrease or this rescue, we can come in and we can look at the histology of this. This is analogous to a nerve biopsy. Again, clinically relevant, but then also in vivo relevant, which is really important for a lot of our pharma partners. What you're looking at here is toluidine blue staining, look at myelination, and then again, an image of electron microscopy looking at that really compact, thick myelination. The case study, we set out to multiplex our system so that we could start parsing out those toxic mechanisms that I talked about at the beginning. Already told you about the EPHIS, the histology. We're also able to, because this is set up in a relatively traditional in vitro layout, we are able to run more traditional studies like cell viability, looking at mitochondrial function. We can sample supernatant. So all of the things you might look at otherwise, we can stack on top of these unique metrics. Here, we're using the compounds that I showed you before from A to D. Those are the 4 chemotherapeutics. And then in E and F, those are the negative controls. And you can see, across the chemotherapeutics themselves, we see this dose-dependent response in the nerve conduction velocity, which is the black line that you see there, as well as the compound action potential that you see in gray. And what you can start to parse out here is some of them cause a much faster functional decrease. And I'll talk about some of the IC50 so that we can kind of understand, how to separate those out. We really wanted to understand not just seeing functional differences, but make sure that that didn't correlate directly to the cells dying, right? Those need to be separated for us really to look at the mechanism. And that's what we did here. In red, you're looking at a cell viability assay. And black is the same conduction velocity I showed you before. And with paclitaxel, you can see along the doses that we worked with there, you see that functional decrease. We do not see a decrease in the viability. That matches with the mechanism here. A microtubule stabilizer would be expected to act this way. When you look at oxaliplatin, highly cytotoxic, you see that those really correlate well with each other. That acts at the DNA level. Bortezomib, you see that they track each other, but arguably that function is a more sensitive metric. And we're really reinforcing that the mechanisms that have historically been seen are showing up here. And then finally, with the vincristine, another microtubule affecting drug, we see that viability does not change. And what's interesting, this obviously plays out in the IC50, but we saw a unique result basically with the kind of differentiating the conduction velocity and amplitude. You can see with the two, obviously there was no change in viability. We already kind of could see that pretty obviously. Bortezomib, you see preferentially this decrease in both functional metrics before you see the change in viability. Oxaliplatin, you see nerve conduction velocity itself really shows up as a more sensitive metric, in the amplitude and IC50 for the cell viability actually are exactly the same. We went next to look at mitochondrial function. This is a mitochondrial membrane potential dye. And what you can see, I think that's pretty obvious, FCCP, which is in F, is a positive control, basically kills off the mitochondrial membrane potential. But then with oxaliplatin and bortezomib in particular, you see a dose-dependent decrease there. And when we look at this quantitatively here, we're basically looking at pixel intensity. And you can see, FCCP obviously significant, oxaliplatin and bortezomib at the high concentrations are, but then oxaliplatin, you see a difference between the two doses as well. Went in now to look at myelin basic protein, what was happening at that level. In red is an MBP stain. In green, you're looking at beta-3 tubulin stained neurites. And what's pretty obvious from a qualitative perspective, at least, is with paclitaxel and vincristine, you see degradation of that MBP, where you don't in oxaliplatin or bortezomib necessarily. Again, tracks with those tubules being a primary mechanistic toxicity manifestation. We look and go in and are able to evaluate the EM images here. And this lets us really dive in, to the ultrastructure where we're able to look at fiber densities, whether it's myelinated or not, the thickness of the myelin, where we might see some myelin degradations. Can even, as you see in E, look at the nuclei themselves, see what's going on there, and then looking at fiber diameter. And that's certainly an important one. And when we put all of this structural data together, all this histology data together, you see some interesting trends that show up. Paclitaxel, for instance, we saw that MBP staining, but then we see fiber diameter is affected in a big way. Microtubule stabilizer, so you would expect that those diameters would see that tubulin build up and sort of expand when they're not in a healthy state. Oxaliplatin here, the eccentric nuclei that tracks immediately with the DNA damage. And in bortezomib, we see some of that, but we also see that the non-myelinated fibers in particular are affected. And then with vincristine, this is really happening with the density. So those fibers are degrading to microtubule inhibitor. So you would expect that breakdown to be happening. And you can see, at the last box at the bottom right, that those fibers are degenerating. We're able to now, the electron microscopy is a very slow way of getting back these responses. So we've been able to refine some of our metrics and we're really working a lot more with cryosectioning. You're able to, with this system, manipulate it in a very big way, specific way. And what you're looking at here is basically a cross section where we're able to slice this up. In the top left, you're looking at a cross section right where the spheroid is. So we're able to look at those cell bodies. And you can see that up close on the image in the right. We're able to then section farther and farther down the channel to see what's happening, more proximally to the cell bodies, what's happening more distally, really continue to differentiate between axonopathy and neuronopathy and understand, not just obviously that a drug is toxic, but why. And that's really important when a pharmaceutical company is trying to improve their compound, maybe tweak it, whatever it may be. They really need to understand in sort of a simplified MPS model what's going on. And antibody drug conjugates are a big one that we look at. They're I think something that's been proven very effective, and now they want to make them safer. So it's been really fun learning from our pharmaceutical partners on this, really how to tease out whatever data that is they might be needing. What you saw along the way here, we recapitulated the mechanistic toxicity for these 4 major classes. The high content data, once you put it all together, really lets you parse out the nuances of this. And where we're moving now, as I alluded to before, is really neuroprotection. We're working on a big case study here demonstrating that we can recapitulate data that's been published where you're looking at, whether it's co-administration or administration of a neuroprotective drug after the damage, seeing that reversal or seeing that protection. And I'm going to kind of close by showing you a little bit of a teaser for a poster that we have later today. We have launched a new platform upgrade to improve our throughput, which is a really big step. Because this system is unique in its geometry and the three-dimensional nature of this, we had to basically take a step back and completely customize the electrode layout, the geometry, the materials we were using to make sure it interfaces with the 3D environment. And as you can imagine, that's not trivial. 3D provides a lot of advantages, but it also makes a lot of things difficult, right? Imaging, being able to interface with the system. And what's nice about this, not only does it increase the throughput as well as the reliability, it allows us to gather more metrics from the system, and it lets us test the system with time as well as across multiple electrodes. This is an example of the layout where we have 10 electrodes through, essentially that same keyhole shape. You see we have the circles on both ends just for a little bit more flexibility. And we're able to grow the axons directly over these electrodes. And we have that in our 24 well system. So we're increasing the throughput, certainly, not yet at 96 well, but we really position this as high content, more so necessarily than 96 well, MEA, something like that. When we start to kind of look into what this actually looks like, I think I mentioned, or if I didn't, I'll say now we're able to stimulate and record from each of the electrodes that you saw before. This is an example where we stimulated at electrode 5. So obviously distally, down the neurite outgrowth microfiber tract. And then we're able to look at that signal that propagates both proximally and distally. And you start to see some signals pop up as we move closer to the cell body. And as more fibers presumably are recruited, we see that these responses are larger in amplitude. And then ultimately, when we see it at the cell body, we see these really big somal responses. And we're able to gain something out of each different location, looking at distance between between the stimulus and multiple of the electrodes all the way back to the cell body. And within each of those are different metrics. And those endpoints are basically drawn out of the data that we analyze with velocity distribution. You see here those 3 distinct peaks, which I'll show you correspond to a really important biological metric. And we're also able to, and this is a really unique one that's opening up some doors that we're very excited about. What you're seeing on the right is basically from top to bottom, increasing the stimulation. So increasing the current of that stimulus and looking at the response. And you can see, at the lowest current at the top, we don't see any response. But then what you see is, that response begins to show up with the increasing current. And then you see more of those responses at the highest level there. And that's really important if we're talking about using this as a pain model. We can see sensitization. We've done a preliminary study here where we knocked out the signals. And then when the signals came back, not only did we demonstrate that they were biological, but they were actually more sensitive to this stimulation. And so we're about to embark on a project with a big pharma company to really dive into this pain application. Neuropathic pain is a big problem. I'm not going to run through all this. I encourage you to go to poster number 31. Corey Roundtree, our lead scientist, will dive into this much more than I will. You can see here with all of those different metrics, and I didn't even talk about half of them on the left, it corresponds to those different histological endpoints that I referenced already. So we're able to glean out of this, basically what's happening with the structure without even necessarily having to go in and look at that structure. Now, of course, we need to demonstrate that correlation many, many times, but the imaging is typically one of the biggest bottlenecks. So we're really excited to package this all into one set of endpoints. Again, go check out that poster 31. This is a really quick demonstration of a little bit of the data. So the pink is not showing up, but basically the entirety of that circle that you see there is the vehicle control. And you can see around the outside, the different metrics. And I'm not sure what's going on here, but the colors are mixed up here. So the No, I'm sorry. The green is, yeah, the lowest dose of paclitaxel that we tested here. That's about a medium strength of toxicity. And then blue is increasing that. And as the basically, circles shrink or those points shrink, that means that those values are going down. And what you can see with amplitude of peaks on the left, velocity of peaks, there's a dose dependent response there. And what's interesting on the right, when you look at medium, slow, and long distances, we see that basically immediately with that medium dose, the medium and long distances are knocked out. But we see specifically that these slow fibers are decrease in a dose dependent way. And that's really important to be able to differentiate between those different fiber types, between those different populations. And so, with that, I would like to take any questions, and please go see poster 31.
Okay. We have time for a couple questions. Please step up to the mics. So I have a question. When you're looking at the different fiber types, do you think that you're actually recruiting as if you would during increased activity, that you have your dominant fibers and then you had your less dominant fibers and you recruit them as you increase your stimulation activity?
That's what we think, definitely. And we're really going out to demonstrate that more and more as we look at that excitability, the dive more into the different stimulation levels. And then we really want to prove that with some different compounds that are known to maybe knock out specific populations and see, when that begins to manifest itself. So we do think that, but we want to validate that further just to make sure that that is what we're seeing, because obviously this system is an artificial microfiber tract, right? And so before we state that too strongly, we really want to be able to parse that out from a number of different angles. But if that bears out, that's a really big deal that we're excited about.
No, I agree with you. Any questions? All right. No more questions? Larry, thank you very much.
Thank you very much.
Lillian, do you want to come up?
Hi, good morning to you all. My name is Liliana Teixeira. Right here, thanks. I'm from the University of Twente, and I'm also a co-scientific lead of the Organ-on-Chip Center of Twente. So today I'm going to give you an overview of how we can be inspired by physiology, actually pathophysiology as well, to create more organized or more structured, more mimicking organs on chips. So this slide is a bit redundant here, but what I wanted to call your attention for is that we should understand and really mimic human pathophysiology and take the cues that are given by nature to instruct cells to do their best, to behave in the way that we want them to behave, to have our models more and more effective. And with that, we can also make them more translatable and more personalized. And at the end, what we want is to have pathologic specific models. And also, basically, we want to target drug development. That's one of our main goals. And for that, I'm going to talk about 3 case studies today. And the idea is to show you how we can play with architecture of instructive elements, as well as geometries, and as well as how we can put tissues together to add complexity to organ systems. So the first case I wanted to show you today is about a lung-on-chip model, actually an alveoli on a chip model. And here we added complexity by adding 3D instructive elements to the chip model. So what we wanted to do here was not to have a flat membrane, but to recreate really the alveolar structure, the 3D environment that the cells are exposed to. And the reason for that was that we noticed that curvature plays a really important role in how the cells that are present in the native tissue, how they mature, how they behave. So it's really important to take into account which cell sources we use, of course, but it's also equally important to steer them to mature properly and to have the diversity of phenotypes that we want to have at the end to again have a more effective model. And what we did was to look at the alveolar structure and really flatten down these structures and recreate these, let's say, 2.5D or or hemispheres that we have in the native tissue. And for that, we used a membrane, okay? We used a thermoform membrane that is also porous, that really recreates these structures we wanted to mimic, also regarding to the size. And that membrane was also very easily incorporated into an on-chip system, as you can see here. So we addressed also the perfusion to guarantee that the flow would not affect the cell seeding. And all the cells were uniformly distributed throughout these kind of membranal structures. So with this, we recreated a, let's say, 2.5D or 3D, as we can call it. And we seeded primary cells, human primary cells. And again, what we wanted to see was that whether we could mature these cells to the different phenotypes we wanted. So we wanted alveolar type 1 cells as well as type 2. And we also assessed for the epithelial markers. So we saw that these cells behave differently depending on the curvature that they were exposed to. So at the bottom, the cells were having different phenotypes than more at the edges where the curvature is a bit more sharp. And not only we seeded primary cells, alveolar cells, we also seeded endothelial cells, so from the microvasculature, and they could be equally nicely seeded on the other side of the membrane. And with this, we recreated a more 3D instructive organ-on-chip alveolar model. So moving on to the next case study. In this example, I'm going to emphasize on how the geometry can also play an instructive model. And again, here it's really looking at the question you want to address. So the model I'm going to talk about, okay, is a cancer colon model. Cancer model on the colon, actually. And what we wanted to address here is how the drugs penetrate into a solid tumor. So colon cancer was an example of a solid tumor. It can be extrapolated to many other solid tumors, of course. And the idea was to see how a nanoparticle would diffuse inwards. And the idea was to recreate a window into a tumor, like we would cut halfway and see the penetration of these nanoparticles. So what you can see here is the colon cancer cells in the middle chamber, where we seeded them in a 3D matrix. Here, the idea was also to start with a simple 3D matrix, but the point was to have more instructive elements here as well. And this matrix can be stiffened to recreate more and more the natural environment. So the pathophysiology again, and how this cancer microenvironment progresses throughout time. So the matrix can be stiffened on demand. So here, as I mentioned, the cancer cells are in the middle, and in the surrounding channels, we have microvasculature. And what we wanted to see was how we could achieve controlled gradients of drug-loaded nanoparticles. So we chose a standard drug to treat colon cancer, just to assess how this drug would diffuse inwards. And here we can also play with the size of the chip. So this was relatively small, but you can also control these kind of parameters quite easily and assess the diffusion of these particles inwards, and as well as the specificity into the tumor cells, to attach to the tumor cells, and how the viability of the cells are affected by these drugs. So we wanted to also check if this drug, so these nanoparticle loaded drugs, would affect only cancer cells or also vascular cells. So I'm not showing this work here today, but it has been published already. Okay, moving on. What I want to do now is to move on to the next case study. So far what you saw was that we can quite nicely control the environment of the cell. So you can see these approaches as creating a house for the cells and instructing them the best way as we can. But when it comes to complex diseases, one house or that hosts a couple of cell types the best we can is not sufficient. And for complex diseases such as, let's say, osteoarthritis, we need to go a step further and create a neighborhood of houses that permits the steering of cell differentiation and communication between tissues. So I'm going to talk about how the idea of the development of joint on chip came to be, and the idea of this coupling of different tissues to form a joint environment was to focus on creating a model towards osteoarthritis. And osteoarthritis affects a huge amount of people worldwide. So basically 1 in 10 people are affected. So we will not most likely die of it, but we will be highly disabled in terms of mobility. So the incidence is quite severe, as well as the damage that it causes in the society. And this disease, in spite of its incidence, has an unknown trigger, so we don't really know how to treat it. And the causes might be in one of the different tissues or in the different, affecting a couple of tissues altogether. So the idea here was to couple these tissues in joint on chip model. So taking, for example, the osteochondral unit, the synovial unit, the fat pad, and putting all these tissues together to recreate the joint, and also add mechanical stress to simulate, for example, movement or harsh movements that would aggravate the disease progression. So what you can see here was the first step, was the model created to reproduce the osteochondral unit. And what you can see here is a model that already has the actuation system that simulates the movement that the articular cartilage is exposed to, as well as the bone. So this movement is not one impact, let's say, it's more of a wave movement, and the cells feel it differently. So that's what we showed in this paper here. I'm just going to give you a glimpse of it. But basically, what we see is that this kind of wave forming compression or mechanical actuation really steers cell behavior, and the cells are quite affected by this kind of steering or input. And you can see this in pro-inflammatory factors. So cytokine release profiles were analyzed by SPRI, and you can see already quite a striking difference in the wave movements on the actuation in these different compartments that enables this wave-like compression. We also looked at how the matrix was being affected, how the matrix deposition was being deposited as well as remodeled. And we also saw quite striking differences in the collagen type 2, which is specific for cartilage, in this kind of wave-like actuation movement. But as I mentioned before, joint is not composed only by one tissue. So what we did after was to look at how we could recreate another tissue that plays an important role on disease progression on osteoarthritis. And that organ was the synovium, so a synovial membrane. And here what we did was to recreate this type of membrane and the shear stress that it's exposed to. So it's more recreating the hydrostatic pressure of the fluid that is in the intra-articular space. But what we could see here is that this is fundamental to recreate the pro-inflammatory profile that is typically in the native tissues and in the progression of the disease. So now I wanted to also do some shameless advertisement to the Organ on Chip Center. And taking into account what I mentioned before, yeah, it's important to recreate the housing system for the cells, as well as the neighborhood for the cells, but also what matters is the community that we create in the development of organs on chips. So in this center, we want to connect researchers, we want to connect different skills, different expertise in all steps of developing the chips, so from design to validation. And we have that expertise in-house to give input to this kind of providing this kind of complexity to the systems and also recreating the best we can the pathological cues. And also one of the main keys of the center is not only connect researchers, but also connect chips. So I'm just giving you an overview of the work that is going to be presented later on by Andries. Basically, what we want to do is create complexity, but in an easy way that we can also enable standardization and automated control. So the idea here is to create a plug and play system where we can connect these chips in an easy way. So just enable the connection of different designs and also enable different readouts in an easier and more facile way that we can all understand and work with. So just to bring the whole story together, yes, we need to balance complexity, but we also need to keep in mind that we shouldn't make our lives difficult. So what I showed you here was that we can easily implement these kind of cues that steer cell behavior in an easy way. So these instructive elements are easy to add to the chips. We just need to ask a specific question to justify this addition of complexity. So again, the models need to be designed to fulfill specific applications, and we do need to understand the limitations of the models to make them efficient. And the technologies developed that can be directed to enable easy translation and validation of the models. So as we heard already quite a lot until now, we need to guarantee the acceptance of these models by the regulatory entities. And what's the main goal here is walking towards animal reduction. So yes, we talked about animal replacement, but I think at this stage we can contribute already quite a lot for the reduction of the animal use. And well, this was what I wanted to show you here today. Just a word of thank you to the people that contribute for being here, as well as to the people who conducted the research, so the PhD students involved. And if you have questions, I'll be happy to answer them. Thank you so much.
We have time for questions. Please come up to the mic.
Helene from University of Pittsburgh. Thank you for the great talk. So for the synovial or chip model, so what kind of cell did you include in that chip?
So we used to sit on top of, let's say, the membrane were synovial fibroblasts, primary human synovial fibroblasts.
Okay, thank you.
Hi, that was a very interesting talk. I just had one question. I noticed several of your slides mentioned actuation. I wanted to know if that's basically driven by the shape of the membrane, or is it more actively actuated?
Yeah, so it's a good question because it might not be really clear there. So I can explain a little bit more on that. It's not the typical membrane that you'd see on the chips. So in that way, it's a vertical membrane that gets actuated in a sequential way. So basically you push the gel that is in the center chamber downwards in a sequential manner, simulating the bodily the movements of the joint. Yeah. So it's a PDMS-based membrane.
Hi, Frank Barilli, Humane Society. Thank you for your talk. I'm wondering with the AT1, AT2 lung cells, did you see any evidence for formation of tight junctions in your curved cultures or any other cell-cell interactions?
Yes, absolutely. So what you saw here was just one example of one cell type we used, but this has been shown or we published before using different cell types. So the different cell lines that are typically used, such as KLO3. And yes, we saw very different ways of the cells being connected and forming this kind of uniform covering of the membrane, depending on the cell type. And the best one that we saw in terms of behavior of making this uniform spreading and connection was with the primary alveolar cells. Yes. Yes, it's very nice already. Within a couple of days, the cells do spread independently or where they are in the curvature. Yeah.
Hi, thank you so much for the talk. I was wondering if you had done any characterization of surfactant production in the lung epithelial model that you had generated.
No, that's a good point and absolutely needed, but for future work. No, it was not done as of yet. So this is quite recent work, so it was just published and it's just opening doors for next questions and we can definitely address that. Yeah, thank you.
Any other questions? I have a question. In the deformation of the system, do you see maintenance of the tight junctions after you deform the cells and then it comes back? Or do you see a gradual destruction of the tight junctions with time?
So that's also a very important question. Thank you for asking. So what you saw here was a membrane that is not elastic. So it's based on polycarbonates. And here you don't see the cells being affected at all because they are just in a stiff-like environment. So the idea was to start with giving the instructive element as geometry. Later on, it was done also with an elastic polymer. And there you already see that the cells, it disrupts because the environment is quite dynamic. So that was not an ideal system. So there again, you have to look at the limitations of the system and how much you can expand and give this kind of elastic movement on these membranes. So that's one limitation that we faced with using elastomers. Yeah.
Okay.
I guess I have a question about the lung model. You mentioned the curvature matters in terms of for the model. I'm wondering, have you looked into what scale of the curvature would matter, like in size of 100 micron, 10 micron? Just wondering.
So we looked at how the alveolar structure was and replicated that kind of blindly, assuming that if nature did it so, that would be a good starting point. But a lot of work has been done to see the influence of the different cell types in curvature in different levels or ranges of curvature. And the cells are affected by it. Different cells affected more, some cells affected more than others, but it has been published by others, not specifically in this work. It has been done with the right range of different geometries, even not only in this kind of hemispheres, but also in tubular shape and membranes. Yeah.
Any other questions? I have a couple of minutes. We want to try and stay on time because this is being live video cast, and so we don't want to get off of time. Have you looked at trying to interconnect the different systems?
Yeah, so now we have two systems that we know that work. And basically we connect them via the intra-articular-like system, and we do that via tubing. So that is working quite okay. I think the complexity will be added when we start adding more and more tissues. As I mentioned, the next one in line will be the fat. So simulating the Hoffa's fat bed. And that will create yeah, some questions that we need to address in terms of compatibility with the media, and also at which stage we need to connect the chips. So in which stage of maturation will also play a critical critical role there. So they can be connected, but we need to address all these challenges in terms of timing and connectivity in terms of medium performance and enabling the cells the best environment they need to. Yeah.
Okay. Vijay?
Yeah, really quickly, Jorge Mojica Santiago, University of Florida. I wanted to ask you about the joint-on-chip platform that you're developing in terms of the dynamic studies that you're doing at mechanical simulation. Have you looked at fatigue or multiple sequences and the dosing of the actual stimulation that you're applying in your cells?
Yes. So what we looked at was basically looking at physiological loading as well as pathological loading. And we assumed that when cells started to die, basically when it was affecting cell viability and increasing dramatically the pro-inflammatory factors. So yes, you can have a wide range, and our limit was when it was affecting viability. So you can have these ranges quite well controlled in simulating just normal loading, exercise, pathophysiological loading regimens that would really aggravate the disease further. Yeah.
All right, one last question, Wesley.
So in the clinic, there's a lot of work trying to regrow cartilage, especially. Does your chip, can it model this at all?
Growing cartilage?
Yeah, regrowing or, just kind of.
So what we do is use already cells. So we use chondrocytes to start with. So we do not differentiate same mesenchymal stromal cells into chondrogenic lineage. But the cells do behave quite well depending on the matrix that you put them in as well. So you need to let the system mature, and this takes basically 2 weeks, so 10 days to 14 days with differentiation medium, to be sure that the cells already produce a sufficient pericellular matrix that will enable them to feel these forces that we are exposing them to. And in that way, we can also see the remodeling of the matrix later on as an effect of these pro-inflammatory responses.
Thank you.
All right, let's thank our speaker again. So next we have Alan Ken from MIT.
Hi, everyone, and thank you for the introduction. My name is Ellen, and I'm a third-year PhD student at MIT, working in the labs of Linda Griffith and Roger Kam. And so today I'll be talking to you about my project on creating an in vitro microfluidic system for observing angiogenesis into synthetic PEG-based hydrogels from perfusible microvasculature. And I actually got switched from a poster to this talk just a couple weeks ago. And since this is the case studies session of the conference, I wanted to focus on really situating this project within our lab's larger goal of creating vascularized models of endometriosis lesions, and then talk about some of the interesting phenomena and challenges that we've encountered along the way. And so to start with some background, in my lab, we're interested in endometriosis, which is a chronic inflammatory condition defined by the ectopic growth of the endometrium outside of the uterus, where it doesn't belong, forming these structures known as lesions. And the endometrium itself is a complex multicellular tissue that's composed of several cell types, including epithelial, stromal, endothelial, and immune cells. And the growth, repair, and breakdown of the endometrium is controlled by the cyclic fluctuations of the hormones estrogen and progesterone. And so there's a regeneration of about 1 centimeter of tissue without any scar formation there. And what happens in endometriosis is that this endometrial growth, breakdown, and repair also happens with the ectopic lesions, resulting in debilitating pain and infertility and affecting about 1 in 10 women in the world. Unfortunately, there's not a lot that we know about the disease at the moment, and that's partly due to things like delays in diagnosis, heterogeneity in clinical populations, as well as limited preclinical animal models, and a lack of reliable therapies that go beyond just targeting symptoms of the disease. And so as a result, we think that there is a critical need for developing new techniques and model systems that allow us to really elucidate the underlying mechanisms of disease to develop better therapies for patients. And so one thing that we've been doing in the Griffith Lab over the last several years towards this goal of developing a in vitro model of endometriosis starts with creating these synthetic polyethylene glycol or PEG-based hydrogel systems. And so this is something that we've applied for a variety of different cell types in the lab, but briefly we use an 8-arm PEG macromer that is functionalized by vinyl sulfone groups. And then it's modified by a number of peptides, including peptides that or integrin binding peptides, as well as ones that sequester cell secreted ECM, as well as a peptide that cross-linking peptide that contains an MMP degradable site. And polymerization occurs under slightly basic conditions here, allowing us to create a PEG hydrogel network into which we can encapsulate cells. And so the benefit of this system that we found to be really useful with all of the different applications that we've been trying to use it for is that it is tunable and modular in that we can really control the different cellular, physical, and molecular parameters to really tune it to what kind of cell type or tissue of interest that we're trying to culture or mimic. And specifically related to the endometrium, this is some of work that is on bioRxiv right now by Juan Necco and Alex Brown from our lab, where they've been able to create a functional co-culture of two of the main endometrial cell types, endometrial epithelial organoids and endometrial stromal cells inside of these droplet PEG-based hydrogels. And specifically, they identified that there are two major integrin binding peptides that are essential for being able to culture these cell types together. One is a collagen-derived peptide that's important for the epithelial organoids, and the other is an RGD that has a synergistic binding site that's important for the endometrial stromal cell side of the co-culture. And when we found that when you combine these two cell types together in this PEG-based matrix, that you can observe the function of the cells looking at things like hormone response, stromal cell desigilization, and maintenance of epithelial cell polarity and morphology. And so this is a great step that our lab has taken toward looking at models of the physiological endometrium, as well as thinking of perturbing it for pathological purposes. But one thing that is currently missing is the concept or the role of vascularization here. And we know that vascularization is a major hallmark of lesion development due to the fact that lesions can grow up to several millimeters in diameter. And to really hit home why this is important in this disease, this is an example of a tissue sample of the endometrium is shown in this bright red stripe at the bottom, and then the myometrium or the muscle layer of the uterus is at the top. And what's depicted with this zoomed in portion is it's a cousin of endometriosis, adenomyosis, where the endometrial tissue ends up growing inside this muscle layer. And in our lab, we've used tissue clearing techniques through the clarity method to really visualize what this looks like. And as you can see from the red staining here is showing the vasculature, and the lesion is this it was this empty cystic structure in the middle, and it's really, we can see that vasculature is really prominent throughout the entire lesion structure. And so what we've been doing to try to really integrate this vasculature into our existing functional co-culture of the endometrial epithelial and stromal cells starts with this microfluidic device design. And so the beginning is very similar to a lot of the vasculogenesis in vitro techniques that others in Roger Kam's lab have presented on already at this conference, as well as others here. So briefly, we have a PDMS device where there are three channels, a central gel channel, and then a central gel channel, as well as two media channels that flank it. And in the central gel channel, we seed endothelial cells and supporting fibroblasts. Here, I'll be talking about using human umbilical vein endothelial cells and lung fibroblasts. And over the course of about 7 days, we can see there is network formation through spontaneous self-assembly of the endothelial cells into these perfusible vascular structures. And what's different about this system from many of the others in our lab is that we ended up keeping these 3 empty compartments, if you can see here, which have no fibrin gel and no vasculature there. And that allows us to then, at a later point in time, add our endometrial cells and surround them with this polyethylene glycol-based gel that I talked about earlier, where we've optimized its formulation for co-culture of these 2 cell types together. And the goal here is to then be able to observe angiogenic sprouting towards this lesion model that we have in this device, as well as be able to measure other kinds of vasculature-related functional metrics, such as permeability, for example. And so this is kind of the big picture of what we're working towards in our lab. But for the remainder of this talk, I'll focus primarily on some of the protocol development and platform characterization that we've done with this system, looking at angiogenic sprouting, and primarily focusing on using lung fibroblasts rather than the endometrial cells for the time being. And so the first thing we wanted to demonstrate is, can this unique geometry of our microfluidic device work, where we are able to retain this kind of empty structure in the center and have perfusible vasculature elsewhere. And so this is showing a confocal microscopy image, a bright field image on top, and then we have GFP HUVEX at the bottom, where the PEG hydrogel is in the middle, surrounded by the fibrin networks. And after 7 days after adding the spheroid, which is 11 days after initially seeding the networks into the devices, we can do a perfusibility test using, here is 10 kilodalton blue dextran. And that allows us to see that the dextran can really permeate through all of the vessel structures without penetrating into that central PEG gel compartment. And so the next thing we wanted to look at is whether or not, once we add some kind of cell types into that PEG gel compartment, whether we can observe angiogenic sprouting there or any kinds of changes in the morphology of the surrounding vasculature. So here we started with using spheroid aggregates composed of lung fibroblasts and macrophages that have been polarized using IL-4 and IL-10 toward an anti-inflammatory phenotype. The rationale there was that we know that those are pro-angiogenic cell types and can be used for our initial platform development when we're exploring for the first time kind of combining these 2 types of hydrogels together in the system. And so to orient you again, this is a bright field image showing the spheroid is in PEG and the spheroid itself is the dark mass right here. And then on the outside is the fibrin and the networks are there. And then, so here I'm outlining where the spheroid was. And again, using GFP HUVEX, we can see that over a few days of culture, there is sprouting angiogenesis occurring right in the local vicinity of the spheroid. And then if we go further out, so this is now 11 days of culture after adding the spheroid initially, zooming into this portion, we can see there's even some anastomosis occurring between some of the sprouts. And this is a time-lapse video, just really putting all this together to visualize the sprouting that's occurring. And so this right here is, these are all maximum intensity projection images. So we wanted to really confirm, is the sprouting happening going into the spheroids and into the gels, or is it just, perhaps migrating on the interface of the gel at the bottom or along the bottom of the device? So to look at that, we used a version of our PEG hydrogel that has a fluorescent peptide incorporated. So now the PEG is all of this green around the edges. The spheroid is the dark mass in the middle, zoomed in, and then the HUVEX are now RFP. And using an orthogonal view here, we can look at a slice of the XY plane and then see that there's overlap of the, the red and the green, showing that there's sprouting indeed going into the PEG hydrogel. And then similarly, when we're looking at an overlap of the dark region where the spheroid is in the green, that there is some kind of sprouting that appears to be happening into the PEG hydrogel itself as well. And then we were also curious about, since I talked about our ability to really tune the different parameters of our PEG hydrogel, whether we can alter some of its properties. So as an example here, we looked at the biophysical properties, primarily thinking about the PEG weight percentage or the polymer concentration, as well as the cross-linking ratio. So here, this is the same image that I showed 2 slides ago. Now I'm annotating that we used a 3 weight percent PEG gel with 40% crosslinker here, and overlaying the GFP in the bright field so you can see where the spheroid is. We found that if you decreased the cross-linker concentration, we got more extensive sprouting occurring in the vicinity of the spheroid. Whereas if we increased the PEG weight percentage, creating a stiffer gel that is now with an elastic modulus on the order of 2 kilopascals rather than 300 pascals, there is significantly less sprouting occurring just right there in the vicinity of the spheroid. And so some other things we wanted to try to explore with the system, and this is all some more recent data, is that we were interested in seeing what exactly is the role that maybe the macrophages that we initially incorporated into these spheroids is playing versus if we just used fibroblast spheroids itself, since there's definitely been a lot of foundational work demonstrating the pro-angiogenic nature of lung fibroblasts themselves. And so here along the top, I'm going to be showing the original fibroblast and macrophage spheroid combination that I talked about earlier, while along the bottom, it will be fibroblast only spheroids. And now here the spheroids are labeled in red. And we basically saw over the course of 7 days that there's quite different morphology and progression of the sprouting that's occurring. And if we're quantifying kind of the overlap of the sprouting over the area of the spheroid and how that changes over time, there's significantly, it's not significant here, but we saw that there seems to be more from some of the replicates that we did in this initial pilot experiment. And I think most exciting that we saw is with the kind of larger sprouts that occurred in the fibroblast only spheroid condition, we could do this perfusability test with dextran and see that some of the dextran does appear to end up in the sprouting, the sprout structures that are where the spheroid is, indicating that this might be something that indicates that it's a useful platform for really thinking more about just spheroid-based angiogenesis here. And then a final thing that we explored with this platform is now wondering, okay, if the fibroblasts are important for inducing angiogenesis and maintaining perfusible network structures, what happens if we put them in a different kind of spatial or spatial organization? Instead of having them in this compacted spheroid aggregate form, what if they were suspended just inside of the PEG hydrogel throughout that entire compartment? So here, this is showing what happens with a perfusability test when we just had the original spheroid aggregate form of the fibroblasts. And we see that there is decent perfusability of the networks around the vicinity of the spheroid. Whereas then if we looked at instead suspending the fibroblasts throughout the gel, the PEG gel in the middle, we actually saw this interesting observation that they seem to be pulling on the networks right around the vicinity of that compartment. And then when we did a perfusability test, the dextran leaked all over into that internal compartment, as well as appeared to be the vessels around the edges seem to be much more permeable as well. And so these are just some of the observations we've made with characterizing the system, but some of the ongoing work that we're currently doing to really adapt it toward endometrial cell culture and ultimately toward our goal of studying endometriosis. One example that I want to show here is we used endometrial epithelial organoids and stromal cells inside the spheroids, as I initially outlined in our in the schematic from before. And this is just re-showing what that adenomyotic lesion looks like with its sort of cystic glandular structure here. And we saw that in some of our experiments that the epithelial cells, which are denoted in the EpCAM, the red stain here, they seem to migrate to the interface of the fibrin and PEG in a way that could potentially be reminiscent of what that structure of the lesions looks like. And looking at the endometrial stromal cell side of the picture, we also had done a preliminary experiment where we created spheroids with the endometrial stromal cells and saw that they very dissimilarly to the lung fibroblasts, they did not induce sprouting angiogenesis and in fact invaded, breaking down the gel, it seems, and invading into the networks, as well as significantly reducing the perfusability of vessels in their local vicinity. And so I think these are a lot of observations that we found here and a lot of phenomena that I think we still want to follow up on. But to sum up, we did see that angiogenic sprouts can be observed to extend from perfusible vascular bed into a synthetic PEG-based hydrogel that's tailored for specific cell types of interest. we also saw that we can tune some of our pegs by physical properties in order to modulate this extent of angiogenesis. And then we saw that the identity and distribution of the cells that we added into the spheroids or into the spheroid compartment can impact the extent of angiogenesis and local vessel integrity that we see. And so, as I mentioned, there are many things that we want to further explore about the platform itself and thinking about what kind of mechanisms are causing what the differences we see between, for example, the fibroblasts in spheroid form versus in suspension and the role of the macrophages. But as a bigger picture, as we're working toward applying this for endometriosis, some things that we are thinking about adding to the system include how do we support the addition of cycling hormones and other potentially such as pro-inflammatory stimuli? How do we enable the real-time study of immune cells since they're extremely implicated in endometriosis? And then thinking broadly about how we explore how endometrial cells, how they have crosstalk with the vasculature, especially if they're manipulated to mimic pro-inflammatory lesion phenotypes. And with that, there are a lot of people in both of my labs and other supporting labs that I'd like to thank, as well as my funding sources, the NIH NCATS program and the NSF GRFP program. And then finally, there are a lot of people from both of my labs at the conference this week. So these are all of their posters and talks that are coming up for the next couple of days. And with that, thank you for listening, and I would be happy to take any questions.
Hi, that's really very nice work. I have a couple of questions. Firstly, as you know, I'm sure one of the roles of extracellular matrix is binding growth factors and sort of presenting those and establishing gradients. Do you know if your PEG gels will bind and hold onto growth factors?
Do I know if our PEG gels will bind and hold on to growth factors?
Great.
I am not certain about that part, but I know that one of my next steps for some of these devices was to stain for the extracellular matrix and see what was being produced, at least by the spheroid types of cells that I'm including.
Right. I think that'd be something useful to know. Second question, more observation maybe. When you put the fibroblasts into the PEG gel, it looked like it collapsed the gel. Is that what you were seeing?
Yeah, that's what it seemed to look like for the experiment where I was suspending them within the PEG gel. It didn't seem like that was happening for the spheroid format. And they definitely seemed to, they looked like they were spreading out more when I looked at higher magnification images for the suspended fibroblast condition. So I think that's something I actually only collected those images a few weeks ago. So still trying to think about how to interpret that.
Yeah, it's an issue a lot of us have had using fibroblasts in these devices. They can, under some circumstances, just really become contractile and the whole thing just collapses. So, yeah, thank you. Very nice.
Thank you.
Okay, sorry, given the time, we probably have to move on to the next presentation. So next, let's thank our speaker again. So next we have Christopher from Hasbro.
Thank you. I didn't know I was going to get a cheering squad. I'm Chris Mackler, principal scientist here at Hesperos. Can you hear me okay? I apologize in advance. I'm part businessman, part scientist, so there will be shameless plugs here, so forgive me for that. I'll be talking today on the classical complement pathway inhibition in the human on a chip model of autoimmune demyelinating neuropathies. And Mike alluded to this earlier today, and I think it's really apropos for this case studies of MPS, because to our knowledge, it is the first microphysiological system data to be included in an IND that's been allowed to go to phase 2 clinical trials. So we started with a company called True North Therapeutics, a small biotech company, like happens in all of the biotech world. They got gobbled up by Bioverativ halfway through this process. And then shortly after the initial phase of this finished up, Bioverativ got gobbled up by Sanofi. And then it sat on a shelf for a little bit and they finally decided to put this into an IND. Before I get started, though, I do want to make sure I give credit where credit is due. I want to thank John Wayne Rumsey, Dr. John Wayne Rumsey. He was a scientist leading a lot of the oversight of the day-to-day operations. He's now the director at the Duke Vaccine Institute, and Case Lawrence, a research associate who did most of the grunt work for this. So one of the really nice things I like about these talks is that I don't have to convince you guys about the microenvironment, surface chemistry. I don't have to convince anybody here about the design of MPS, right, or the cell types that are there. But I do want to talk shortly about, like the MPS design is important. The microenvironment, the surface chemistry is important. We use a lot of non-biological self-assembled monolayers. It allows us to do photolithographic patterning of the cell surfaces for spatial dimensions of cells for cell-cell interactions. Also, like the spatial organization of that for cell maturation that Mike alluded to in some of the cardiomyocyte data that we have earlier. And for us, it's really important. We use, as Mike alluded to, also, we use a serum-free medium, right? It's chemically defined. It is a little bit expensive, to be truthful, right? But, we look at that and say, if we can define the nature of all the ingredients that are there, and we know the interaction of the cells to the surface, to the medium, right, in the microfluidic device that we have, right, both computational fluid dynamics, right, how that affects, shear stresses and flow rates, and how that affects nutrient utilization, right, we have a better understanding of how the system works. And shameless plug for Hesperus, we are a vertically integrated company, so we do in-house have everywhere from a CNC machine and like people who manufacture them to computational fluid dynamic modeling to Python coders to surface chemists, analytical chemists, and a bunch of biologies and engineering team led by Dr. Long. If you see him in the hallways, you can stop and say hi. So I'm sure that many of you who have seen any talks at Hesperus have heard functional readouts over and over again, right? And I'm going to beat it in again, right? We really pride ourselves on the ability to measure these functional parameters, right? And so I think you alluded to earlier today, there's functional parameters many times that, for our devices and depending on how you design them, precede cell death by quite a bit of time. We've even seen the functional parameters, say conduction velocity of the heart, actually precede troponin or creatine kinase release into the biomarker release into the medium, right? And so they're really sensitive measures of cellular function. And for us, it's really important because, we also pride ourselves on this ability to measure not only acute dosing, but repeat dosing, right? Or progression of disease over time, say in Alzheimer's or ALS, right? As you press in, some of this data will be in some posters that you'll see later, right? Where you can measure the differences in the genetic makeup of the cells, right? And how it progresses over time and the onset of disease. And we think that it will augment the biomarkers that are needed, right? We don't have to predict what the biomarkers are going to tell us about function. We can measure that function directly. And so we were approached by True North Therapeutics to study multifocal motor neuropathy and chronic inflammatory demyelinating polyneuropathy. And they said, do you think you could build us a model of this? I said, sure, let me go Google what MMN and CIDP are, because I don't know. So for any of you out there who don't know what MMN and CIDP are, they are very similar disease phenotypes that for our purposes, we're looking at only the motor neuron portion of it. They do exhibit different clinical manifestations, whether the CIDP is symmetric and affects both motor and sensory portions, where multifocal motor neuropathy is asymmetric and only affects the motor unit. And so what the common features of these is that you get persistent conduction block of the neurons, and that a lot of the patients are shown to have GM1, IgM, and IgG antibodies circulating. And the theory behind this is that the autoimmune antibodies, the GM1 mediated IgG, IgM antibodies will bind to neurons. They believe at the node of Ranvier, and that binding of the IgG, IgM will induce the complement cascade and then affect the motor neuron conduction. And so True North Therapeutics had a compound that was a C1s inhibitor, and they wanted to look, it's called TNT-005. I'm assuming it was the 5th generation of that. And they wanted to see whether or not that that if it would block the complement deposition, and B, if we could measure conduction block and its ability, the patient sera sample's ability to modulate conduction velocity of the neurons and their molecules' ability to ameliorate that conduction block. And so we thank you, Jeff Guptill from Duke University, who gave us multimodal neural neuropathy and CIDP patient sera samples. So the first thing we wanted to do was to ensure that the patient sera samples that we got, a lot of the patients go IVIG therapy that's supposed to remove a lot of these autoantibodies. We wanted to ensure that there were GM1, IgM, and IgG antibodies in each of the patient sera samples that we collected. We got an ELISA to do that, and this is just quantification of the differences in the IgG, IgM antibodies in each of the patient sera samples that we received. And so before we move further, we wanted to test the utility of the system. Can we ever even measure complement deposition? So in a very controlled environment, before we wasted a bunch of very rare patient sera samples, we wanted to test a secondary only antibody for complement deposition. We did a normal human serum control with 0.05% lyophilized complement, and a positive control was an anti-GM1 antibody where we added complement. And we looked at both Schwann cells and motor neurons, and we can see anti-GM1 mediated complement deposition both in Schwann cells and motor neurons. And if you can tell, there's a lot more in the Schwann cell portion. And so with that confidence that the system will be able to measure GM1 mediated complement deposition, we took the patient sera samples and we wanted to ensure that the patient sera samples would deposit IgG and IgM antibodies onto both Schwann cells and motor neurons. And they do, more so on the Schwann cells. And then you get a lot more of the anti-IgG, which is clinically what you see. And so the next question was, do these IgG and IgM GM1 mediated antibodies activate the complement cascade from the patient sera samples? And so again, we took monocultures of Schwann cells and motor neurons, and indeed, both in Schwann cells and motor neurons, you get C3b and C5b through 9, and in the Schwann cells, you get quite a significant deposit of the C5b through 9 complement deposition. And so before we used a lot of the TNT-005 antibody and more patient sera samples, we wanted to ensure that through our control mediated GM1 system that we can get TNT-005 blockage of the complement deposition. And when Dr. Rumsey first showed me this data, I said, clearly you have mislabeled your images, because there's no way that you got this much blockage of the complement. So I do want to state that this is blockage of complement deposition and blockage of conduction block. They're co-administered, both the patient sera sample and the TNT-005 antibody were co-administered simultaneously. At some point, we'd like to look at the ability to reverse this phenotype, but we thought the most staggering results at the very beginning of this project would be co-administration and blockage. And so I said, repeat it. They repeated it, got the same answer. I said, okay, well, I feel comfortable then to move forward with the patient sera sample. And these are two representative images of the complement deposition. And these are co-cultures of both Schwann cells and motor neurons together, that you get quite a bit of complement deposition in all the serum samples. And TNT-005 was able to reverse or ameliorate that complement deposition anywhere from 88% all the way up to 97 or 98% reduction in complement deposition. And so Bioverativ, well, at that point, it was True North Therapeutics, was extremely happy about this data. And they said, okay, great. So you can block complement deposition, but what does that have to do with conduction block? Do we actually see any conduction block of these motor neurons with this complement deposition? I said, okay, so we built a microphysiological system to study this. And so an SU8 mold and PDMS design, it was very simplistic. To guide axons, we'll put motor neurons on one side, Schwann cells on the other, allow the Schwann cells to migrate into the tunnel, allow the axons to go through the tunnel, and you'll get Schwann cell motor neuron communication inside that tunnel. I'm sure that some of you guys are going to ask us later. We tried to measure the extent of myelination in the tunnels, but trying to take the tunnels off or trying to slice them or trying to vacuum antibodies through caused a lot of damage to the cells. And so at this particular point, we're still trying to figure out how you look at the extent of myelination within the housing design. But right now, all we know is that there is either trophic or physical interaction of Schwann cells and the axons within these tunnels. If you want to look at any of the myelination work that we have done, Akash is giving a talk later today, or a poster later today, poster number 27. You can look at our myelination model, both using primary human Schwann cells and iPS-derived Schwann cells. And so, as you can see, is there a pointer? This thing? As you can see here, we align the PDMS tunnel over the electrodes, and you can see multiple axons that run through. Because of those multiple axons, you do get a, so what we wanted to really look at was at least 3 microelectrodes in a row where you get a signal that propagates across, right? And since we know the spatial distance between each of those electrodes, we can measure the conduction velocity of the neurons. And because we have a range of axons that are there, ooh boy, how do I go back? We have a range of the conduction velocity of those axons. And what we found is that the average conduction velocity is about 0.2 meters per second, which is similar to in vivo unmyelinated small diameter axons. So we were pretty comfortable that they're physiologically relevant. And so we wanted to look at both the negative control and the positive control, right? Just normal human serum samples with a TNT-005 isotype or the TNT-005 inhibitor. And what we saw is that it really doesn't matter. You get some fluctuation up and down of the conduction velocity with a negative control. But in the positive control, you get a massive reduction in the neuronal conduction velocity and also in the action potential firing frequency, and that the TNT-005 was able to ameliorate that phenotype. And so then we quantified that both in the negative control group where there was no difference between the isotype treated or the TNT-005 treated, and both in the firing frequency and the conduction velocity. However, in the isotype treated, you get approximately 80% decline in both frequency and conduction velocity with the isotype treated, and that is statistically significantly ameliorated by the presence of the TNT-005 in both the positive control. And you sort of see a graded response with it. We took all 15 of the patient's serum samples and lumped those together to quantify, and you get about a 50% reduction in both frequency and conduction velocity across those patient serum samples, and that also is ameliorated by the TNT-005. And so that data was put together in a package with Sanofi to go into an IND that was submitted in December of 2020 and allowed to go to clinical trials in April of 2021 and is currently ongoing. And so what I hope that I showed here is that we build a system to model a subset of clinical features of MMN and CIDP, and that their proprietary antibody TNT-005 was able to block both the complement activation and deposition as well as conduction block. And I think we're working on this model at this particular point has therapeutic value in quite a few rare diseases like Friedreich's ataxia or Charcot-Marie Tooth syndrome, right? Or even Pompe disease. And in some parts, we're looking at its application towards ALS. And so I will open up the floor to any questions that you might have. And shameless plug. If you like anything that you saw today from Mike Schuler or Jay's talk yesterday or anything that I said today, we are currently hiring quite a few PhD level scientists, an innate immunologist, a liver biologist, and list goes on. We do have a booth that you can go. And if you have any wants or needs, we pride ourselves in our ability to build whatever you want. I didn't know anything about MMM or CIDP when we started this. And here we have an IND, so we can build anything that you ask us to.
I have a question for the speaker.
Come on, everybody, wake up.
I mean, feel free to not ask any questions. I'm okay with that. Sure.
Please go to the microphone.
We either wowed them or bored them.
So the technical question is the images you show for the max system, right, of the complement. So you show just one color. So you're using one specific antibody that identifies all the components of the system.
So we actually stain for both. There's a anti-C3b antibody and a C5b through 9. It's a polyclonal for that.
Okay.
What we found was we initially were upset because we couldn't see any C3, but if you, and this data is not in there, if you stop the systems earlier and fix them and stain them, you'll actually see the C3. I think the way that the system is designed, when the C5b through 9 complex gets laid down, it blocks the ability to really see the C3. So a lot of it you'll see was in red because that's the C5b through 9, right? Downstream of the complement cascade, right? But if we stop that early, you see more of the C3. And so we have that data, but it's initially we were concerned, but temporally speaking, it makes sense.
Any more questions?
Dr. McAleer, thank you very much.
Thank you.
All right, we're gonna have our last speaker, Jorge from University of Florida.
Good morning, everyone. My name is Jorge Mojica Santiago. I'm a postdoctoral associate with the University of Florida in Gainesville. And I'm going to be presenting today on a microphysiological system that we have developed to test anti-atrophic potential using electrical pulse stimulation. But first, before I jump into the science, I wanted to acknowledge the pretty large collaboration that we have among interdisciplinary scientists. Dr. Shaban Malani, who is the PI of this grant, put together this massive group of bioengineers, clinician scientists, aerospace engineers. And we are looking into doing a skeletal muscle microphysiological system. Essentially, muscle itself is a complex tissue that is very dynamic and active. We know that skeletal muscle cell is a form of satellite cells that fuse into myotubes. Here we can see in red, these myotubes are fused in multiple nuclei in blue. Age-related muscle wasting, or sarcopenia in particular, is characterized by the gradual atrophy of the muscle. And so the shape that we see here gets changed over time, but it also causes a dramatic decline in mobility on an increasingly aging population. Unfortunately, because the accumulation of these changes do take time to occur, we do not have effective therapeutics to really address the problem of sarcopenia at this time. Here we can see an example, let's see if that plays up, of a young, healthy muscle thigh against an older sarcopenic patient. You can see an accumulation of the fatty tissue, and so and a gradual atrophy of the myobundles or the fused myotubes. Now, what's interesting is that astronauts have for decades shown similar changes in skeletal muscle atrophy, and we thought that microgravity could help us as a model or an accelerator model for degeneration of muscle. So in fact, we are interested in understanding how human muscle biology in microgravity relates as for aging. As we all know here in this conference, microphysiological systems are great models for helping understanding human diseases in vitro. Microphysiological systems for skeletal muscle have been traditionally used in a couple decades using these two, in general, is using tensile, applying a tension on the skeletal muscle cells and in a 3D hydrogel matrix. Some folks have used photopattern hydrogels. Others have used general bundled frames that are deformable or fixed. And many have used microfluidic channels to also allow for perfusion, continuous perfusion of the skeletal muscle tissue. Now, as I mentioned before, we are interested in studying how microphysiological systems in microgravity, we are looking at how in microgravity we can model the aging of the skeletal muscle itself. Therefore, we are we're developing a platform that we can indeed look at in microgravity, how the contraction parameters change, and also eventually look at the transcriptomics changes as it returns to ground. So how we did this is we first started looking at multiple cell matrix environments. We started by combining a multiple collagen fibrin blends and also comparing this against a matricial collagen formulation has been previously published. We found that laminin and HA were important components for the collagen fibrin blends to produce a general myotube distribution across the whole the skeletal muscle. And we can also see that matricial collagen, as it's shown in the literature, does appear to form this myotube fusion index. So we decided to move forward with the matricial collagen formulation, but before we added any cell types of this, we use simulations of computer fluid dynamics, as others in this conference have shown. Simulations allow us to really look at the concentration distribution across the whole chip. And so because we were looking at a rather complex geometry, we were interested in both how the volume fraction of air is moved out. So in top here, you can see hydrogel being injected through the ports and then just kind of gradually filling up this chamber. Now, over time, we see that the skeletal muscle cells pull on the matricial collagen matrix and form this rather elongated bundle of myotubes. Also, we studied how much of the flow rates we needed to apply such that we not only flooded the entire system, but also allowed diffusion of the component of the media to reach the cells that were in the middle. So we found that simulations were incredibly helpful in really finding what the right parameters were for input. Now, having developed this geometry and understood really what parameters we needed, we then took satellite cells from patients. And I just want to clarify that we looked at multiple human subjects, both on older sedentary and at young active populations. But we did pull the cells together. And so all the work that we'll be showing here has been multiple combinations of multiple samples of the microphysiological system of the pooled cells together. So what we did is we first plated the cells in 2D, differentiated them, expanded them, and then we pulled together and plated them into the collagen matricial formulation that we developed earlier. And we applied a differentiation media and then eventually observed how the myotube fusion occurred over a span of a week. And then after 13 days, we did start electrical stimulation to apply and to the cells and look at the contractility of these cells. And we saw that older sedentary and young athletic both did appear to form the myobundles over time. But we definitely noticed that there were some differences in the morphology of these bundles, which goes to show how these cells respond and increase variability among different patients. Then again, we looked at the simulations of applied voltage to understand what electrical field were being distributed across the whole geometry, and specifically to look at what the cells were experiencing. We identified a range of multiple voltages, and we then narrowed down to a 3 volts applied to this platinum electrode located along the media channels to achieve a desired geometry, desired electric field intensity at the center line where the cells were encountered. We also found that this voltage was the one that allowed us to have the minimal heterogeneities in the electric field in the middle. After applying electrical stimulation, we took videos of these bundles contractions, and we were able to use digital image correlation analysis to map the horizontal and vertical displacements of the pixels within the bundle in the different areas of the bundle. Now we define a region of interest that at least accounted for the PDMS pillar, and we map these together, all these vertical and horizontal displacements, and obtain a vectorial summation of the displacement occurring at each location of the region of interest. With this, we then applied an average of the different displacements that we have found, and we eventually plotted this signal with multiple peaks over time. Now, what we found is eventually these cells that respond to stimulation, but rather differently. And this work has already been accepted for publication. What we noticed is that during the resting phase, before we applied electrical stimulation, there seems to be some rather spontaneous contractions occurring, or spontaneous peaks that we can observe, especially for both young athletic and all sedentary. And then as we applied the electrical stimulation, the young athletic cells were the only ones that really responded in a more asynchronous manner with the stimulation that we were applying, whereas the all sedentary cells they still continue to have these random peaks, random contractions over time. After recovery or after the stimulation was removed, the cells that gradually responded stopped contracting in the same way. We found that also this behavior rather correlates with the way that young athletic and all sedentary fusion looks. So if you look into the fusion index, which is a metric of how many nuclei per myotube are present in the cells, you can see that the younger athletic cells were highly fused, and then also had the better response to the contraction. We then applied signal processing techniques to reveal contractile differences between these signals that we observed. I'm going to just briefly describe here that we took the mean displacement signal of the stimulation curve that we found. We also look at the average of displacement peaks and average of displacement valleys, and then normalize these against the mean displacement of the baseline phase. We also applied a fast Fourier transform analysis to obtain the average contraction rate of our bundle. In summary, we really looked at how young athletic stimulated ships had higher frequency of contraction than the all sedentary cells. And in addition, we also had differences between phases. So we were able to show that the stimulation phase were consistently higher than the baseline phase and then the recovery. We also looked at multiple, using an RNA-seq, a PCR array. We looked at expressed myogenesis and contractility genes. In specific, we see group 1 genes that were downregulated for myogenesis. This is the group 1 over here. Group 2 were actually upregulated myogenesis genes. So it's this rather range of genes over here. And then group 3, 4A and 4B were all focused on the contractility aspects. We saw that group 3 were mostly genes that were downregulated for contractility, and 4A and 4B were upregulated. By a 1.5 to 2-fold specific tight intrapointed family of genes with electrical stimulation were found to be significantly regulated. And then since aging and increased physiological capacity are counteracting and have been shown to counteract in opposite directions in previous studies, we sought to evaluate how changing the increased the electrical stimulation regime that we applied would hopefully counteract aging in effect. So we then continue to look at these all sedentary young athletic cell types populations, but now we have applied a different electrical stimulation regime over time. So instead of just doing the one pulse stimulation per day, we apply twice stimulations per day. And what we found is that, what we found is that in general, by just sheer increase in the number of stimulations per day, we obtain a higher mean displacement, signal than the one stimulated per day. But we also had a more consistent frequency. So remember that the older sedentary cells were a little bit asynchronous and they had more spikes all over. We were able to sort of rescue the older sedentary to have a more synchronous response to the young athletic by just sheer exercising these cells. Now, not only are we interested in looking at the contractility aspect, but also what is it a way that we can use the system to test for drug screenings. And so we're looking at contractility recovery verified with timatidine, which is an antitrophic compound that is obtained from green tomatoes. So timatidine has been shown to, is thought to increase the protein synthesis, mitochondrial DNA, myotube size, the expression of anabolic gene, and also cause cellular hypertrophy. We hypothesized that by adding timatidine into a skeletal muscle system, we could look into increasing muscle mass and strength, and also improve the exercise capacity. So to really validate this, we combined both the aspect of all sedentary cells and young athletic cells being exercised twice per day with different doses of timatidine. So specifically, we tested a 10 micromole in the dosing of the timatidine against a 5 micromole. And we all compared this against a control that was ethanol dose only, so no timatidine added. What we found is in general, the dominant frequency of the myobundle from these older sedentary cells were again synchronized with the young athletic derived myobundles like we had seen in the past with just twice stimulation per day. Not only that, but the mean displacements of the young athletic continues to be higher than the older sedentary. But one really interesting fact that we weren't accounting for is that this time around, the older sedentary cells during the eastern phase appear to just continuously increase with time. So it appears as though the cells are not, the muscle bundles are not really recovering and just continue to remain in tension during the stimulation phase. That was definitely not observed when we applied the timatidine drug treatment, both at the 5 micromole and the 10 micromole treatment. So as you can see, the blue line here, the older sedentary do not have that continuous tension increase with respect to time. As of now, this is preliminary data, so I just want to highlight that this is continuously being worked at, and we're looking at replicating these results further and hopefully obtaining more parameters like the ones that I had explained before. Another thing that we're looking at this time on ongoing work is looking at the localized displacements across the entire bundle. So as we mentioned earlier, we have the ability to map out how displacements occur on the bundle in the different regions. And we hypothesize that there will be differences across the whole bundle because our bundle is rather large. And then we really observed that there were no differences across the displacement on the overall mean. You can see here multiple regions of interest, branded here by different colors. And as the muscle bundle moves, even though we appear to see differences across the regions, in reality, whether we average by rows or average by columns, there are really no significant differences in the myobundle signal. Now, in summary, I hope that I have conveyed today that our young athletic stimulation myobundles appear to have higher dominant frequency and also higher mean displacements of the older sedentary. As you can see here, in terms of the mean displacement alone, it's basically 10 times, 4 times higher just by applying twice stimulation to the once daily stimulation. However, when we combine the tomatidine treatment again with the twice stimulation efforts, we appear to improve contractility of the older sedentary cells actually to degrees that are fairly closer to the young athletic, as we can see here. So we're roughly around the same values that we were observing for young athletic before any tomatidine treatment was added. So this is really encouraging data for us, and we are looking at hopefully replicating these results. And we will be updating folks as we know more. Lastly, I would like to wrap up with indicating what how this actual model of muscle atrophy can be used in as an aging simulation model. We did develop this model for a space flight compatible. We actually had we flew it back in December 2020. Unfortunately, for technical difficulties, we weren't really able to have multiple replicates. And so the only thing that I can show you here is as we were able to observe in space how stimulation causes young athletic cells to contract the same way that we observe in the ground. So all of the data that's shown before were ground data, whereas this data right here is from actual in the International Space Station videos. Now we're looking forward to fly next week in another rocket launch. We're looking at having this system. I apologize. So we're looking at having this flight happening next week to hopefully getting a prolonged time frame and multiple replicas of this data. And in September or later in the fall this year, we're going to look at not only applying electrical stimulation studies, but also doing the drug delivery system that we just explained. And with this, I will just like to thank you everyone for listening, and I'll happy to take questions.
Thank you. It's wonderful. I'm a muscle biologist by training, so it's very intriguing to me. So have you looked at innervation for motor neurons to look at not just the sarcopenic or reduced cross-sectional area or atrophy in that, but also motor recruitment?
Truly not. Our system hasn't been developed to look at the interface of neurons and basically the neuromuscular junction interface. It is an interest of mine personally to look at that in the future, but with this collaboration that we have, we're focusing entirely on just the skeletal muscle parameters alone. But it is a very interesting area to pursue.
And then just one other. Have you looked at different exercise paradigms to look at, because I think the model would be really nice for looking at fiber type toxicity, right? And looking at like, say, a HIIT training, sort of like your young adult versus like a chronic low frequency stimulation, like endurance walking, and modulating that fiber phenotype and how drug toxicity differs between them.
Our group has not looked at multiple different doses of electrical or of the exercising regimes, but it is certainly of interest to us it is kind of future work or future aims to really look at multiple different, not only exercise regimes, but also different drug treatments. So it is in the works. We're currently looking at ways to characterize that, but we do have a limited, with the milestone-driven grant that we are under, we do have a very specific set of aims to accomplish.
Yeah. Anyway, I'll catch you after this. I've got quite a few more, but I'll let him go.
Hi, super informative talk. I'm definitely going to be eating a lot more green tomatoes. But as my question is, how long do you take these cultures out for with this repetitive testing?
We take them out for 18 to 20 days the longest. We don't go past that time frame usually.
So how does that in vitro timeline correlate to in vivo?
That is a great question. We have actually devised we have focused our timeline to be more in line with what we want to do in microgravity. So in reality, the in-ground studies that I just showed you were designed to be comparable to what we're doing in space. It is of interest to see how both co-cultures of different cell types and also extension of the culture time has an effect in the actual contractility parameters. We haven't looked at that yet, though.
Gotcha. Thanks. Good luck with your launch.
Thank you.
So I got a question. So it looks like you weren't actually monitoring the movement of the posts like most people do. You're actually looking at the fibers themselves, which is interesting. And it looked like in the young samples that you had a more uniform motion capture, whereas in the older individuals, you had intense areas and then areas with nothing. Did you see the areas that had intense with the older samples getting larger with time, or their intensity going up? And did you see increased fusion, or did you see more of an upregulation of the contractile apparatus of the myoblasts or the myotubes that were actually contracting?
I'm going to take that question by parts. So the first part of the question, I would say, we do not look at, so we did look at multiple regions of the pixel intensity changes, right? And we did see what appeared to be localized differences across multiple samples. But when we compare statistically, there were no differences between older sedentary and young athletic patients in general, as far as the region differences go. The other part was about.
Do you think that you think that you're getting increased fusion areas for recruitment in terms of it, or do you think that you are actually getting upregulation of the contractile apparatus and making the myotubes that were actually contracting stronger, or do you think that you were actually creating additional fusion around those areas that were contracting in the sedentary?
That is a great question. We have not looked at temporal differences because we take these videos, essentially the way that we take this is at the end of the culture period. So we do not track this over time, unfortunately. At the end of the culture period, then we look at how the muscles respond to the stimulation. And it is actually, I did not cover this, we do apply stimulation daily over, I think it's an hour that we apply it to. But now, these simulation videos that I showed you were just 40-second snapshots. So there might be some differences between how this muscle is responding over time that we are not capturing, unfortunately. There is in the works in the Malini lab looking at having a camera system that we can track the muscle bundles with time. So it is currently in the works. We do think, based on the gene expression data that we have observed, that there seems to be differences between the older sedentary and the young athletic patients, and that the stimulation does appear to have some effect. So we suspect that it's more on the gene expression side.
One quick question.
Yeah, actually, this question is related to what Jay just mentioned. It's kind of, I'm not sure whether it is a recruitment issue or it is the maximum contraction issue. So yeah, I think you partly answered that question. Another question is, I don't know whether you have tested like a frequency dependent summation of the muscle. Because when you increase the stimulation frequency, the muscle has some summation and it goes up, climb up the tension. Have you ever tried that? Have you ever found anything?
We have not tried doing the summation to reach up to the titanic contraction that you're describing. We have not tried that. At the highest we did try was a 15 hertz frequency, and we did see that it appears to add up, but we didn't have enough samples to replicate that at the time. What I would say though is our camera system that we use to track the videos of these motions, they're not as high frame rates. So even if we apply a higher than 4 hertz frequency, we're not going to be able to capture movements within that range.
Okay, thank you.
You're welcome.
All right, well, let's thank all our speakers today. It's a very interesting session. This concludes the session. Thank you very much for your attendance.
And don't fall off the step, right?
I do everything. I have a green point, so if you feel like hugging, you don't seem to be the type, but I don't know. Yeah, come in.
Can I have the slides up, please? So I want to make sure we don't lose time for this interesting debate today. So I want to thank everybody for coming today. I know it's lunchtime, so it's really nice to see so many people made the effort to come. Maybe because there's free food at 1:15 later, I'm not sure. I'm Phil Hewitt. I'm from Merck in Germany. So I'm not going to say too much. I think the best thing is to get straight on with this. And we did have a lot of discussions in the organizing committee about what a debate should be at such a meeting, because I think everyone is very like-minded here. So it's very difficult to find a topic which is black and white. So we agreed on this topic, so many choices, what models are best, but that's more or less saying, where does MPS fit between standard in vitro and animal studies? And I think we have 4 speakers today instead of the usual 2. So I think it might be more of a discussion rather than a strong debate. So we have 4 wonderful speakers, so Ivan, sorry, I always say it wrong, Lorna, Annie, and Jason. So I won't say anything more apart from hand straight over to Ivan, who will start us off. And maybe introduce yourself as well.
Great, thank you. Thanks, Phil, for having me, and thank you all for coming in person. I'm Ivan Rusna. I'm at Texas A&M University. I'm a professor, and I'm chairing a regulatory science and toxicology training program there. And as Phil said, we do have quite a bit of agreement on a lot of topics. And so what I would wanted to do to start us off is to perhaps stir things up a little, and to bring up some of the discussions that I'm hearing a lot in these types of meetings, being a toxicologist, coming to these meetings, and people say, well, animals have failed us. So I wanted to start with maybe some considerations as to where that comes from. And as I was looking for facts rather than fiction, I actually wanted to find some supporting information. So there are 4 statements here. They're direct quotes. They are kind of a transition point from research translation, or from the laboratory to the clinic, is hard, to perhaps animals and are not useful. So the facts are, this paper in 2003, these folks have done a literature search of the best journals and papers who made explicit claims that their findings are translatable to humans. And then they look 20 years later as to what happened to those. So it's an interesting thought experiment. And what they found is that about 18% of things that were claimed in Science, Nature, JCI, and likes that would be translatable actually only translated. And it didn't really matter what model they were from. Were they human model, animal model, molecular, and cellular model? And so that paper was cited in 2014, and kind of lamenting the fact that it's hard to do translation. And then people for ethical treatment of animals picked that up and said that animals failed us. And they used the same statistics, and they cited the same papers. And unfortunately, to me, the top cite comes from NCAT's press release, which wasn't referenced to anything, but basically uses the same arguments that animal research has failed us in the clinic. So is it fact or is it fiction? And I'll come back to this at the end, but basically I wanted to point out that misleading statements like the 2 on top are counterproductive to research translation. So the utility of preclinical animal data and drug development. So if you ask folks who are here on the podium, and many in the room, related to pharmaceutical companies one way or the other, there are 3 papers cited there, and each one of those papers says that there's great value in animal experimentation for developing of safe drugs. And then if you look on the other side, and this is a paper from British Union on the abolition of vivisection, so obviously you understand where these people stand. And in their paper, they do say that they don't believe that animal experimentation was actually useful, but they do acknowledge, and these are again direct quotes, that a positive signal in an animal study actually is quite valuable to human risk. So let's talk about purpose, right? So again, on the here, joined by me, all people to whom the chemicals are drugs. And I'm a toxicologist, and to me, chemicals are much more than that. They're agrochemicals, they're industrial chemicals, they're pesticides. And while we're taking drugs by milligram or smaller amounts, we're actually using all those other chemicals by metric tons, and we buy them actually in ounces. Now, for most people in these meetings, FDA is the only regulatory agency, but because there are many more chemicals than drugs, there are a lot of other agencies, and purpose for them will be very different. And I invite you to, again, all learn about that, and there's a great paper that summarizes all of this. Now, let's talk about two purposes. So one purpose is, again, these environmental compounds like pesticides, and this is a list of tests that are required for registering a pesticide. And if you look at some of them, they're like acute toxicity tests for particular toxicity. They're called six-pack. There are already a lot of in vitro and in silico models that are developed that are replacing them, and none of them, to my knowledge, are MPS. Now you go down the list and there are studies that take days or months, and you are really not sure what you're looking for. So when we open an animal, we examine 45 tissues or so. So try to think about 45 tissue chips, all connected and over 90 days. Now you go further into cancer studies, and those are usually years. And again, we're not sure what we're looking for. And I also wanted to acknowledge that a lot of models on this list are actually in silico or in vitro models already. So it's not like the only tests are there for animals. Now for FDA, if you, again, are trying to have a purpose as a drug, then you need to think whether it's good for your investigational new drug application, which is before clinical trials, and you need to prove to FDA that the drug is safe. And then in clinical trials, you need to prove that it's both safe and effective and intended for purpose. So there are a lot of, again, on this table, assays and phenotypes that need to be assayed. And some of these can be done by microphysiological systems, safety pharmacology, for example, but many others are not. And again, this is where another 10% argument can be obtained. Once you're in clinical trial, 90% of clinical trials fail, but largely for reasons that are unrelated to the safety package that went in. It's for actual clinical trial purpose and your population of individuals. So I wanted to conclude, and again, I hope, looking forward to the conversation, that we all should celebrate, those of you who are in this room developing microphysiological systems, because they are definitely very useful, potentially useful models. But we also need to make sure that we have models of different level of biological organization that includes humans, but it also includes other organisms as well. All models are wrong, including in MPS and in vitro, and many of them fail. So we really need to do a better job in research translation. But I want to make sure that you understand where I stand in this conversation, is that animals will remain in use to assure safety of drugs and other chemicals because they're required and because they are actually irrefutable evidence that they actually save human lives. And finally, I think we need to meet in the middle, and I'm looking for this conversation. And as a toxicologist, I need to do a better job educating my colleagues in microphysiological systems as to what is needed. But also we need to stop, I think, making some ridiculous claims about animals are wrong and MPS are best. So thank you.
Great. Thanks, Ivan. I don't need to move. I think next I'd like to introduce Lorna from Emulate. I think has a very, not a very different, maybe a different perspective.
Thank you. Am I driving the slides? Yeah. Okay. So as we say, the topic of today's discussion is about how many choices we have to use to pick the best model for the purpose we want to use. And my disclaimer here is that, of course, my background is drug discovery and development. So again, this conversation from my perspective will be very much taken from a drug discovery perspective. Okay, so what we in drug discovery are trying to do, of course, is predict human response to drugs. And I'm glad Yvonne actually showed the figure of 90% failure rate, something him and I have talked about a number of times. And I think it is very fair to say that clinical attrition is particularly high. However, this is often an aggregate score. And you know, what we really need to think about is the progression of good candidates into phase 1. Because if you don't get a good candidate into phase 1, the data consistently tells you that the probability of approval is only around 10%. So later stages in clinical trial tend to be, for sure, more predictive and greater chance of success. But what we're focused on at Emulate is actually understanding what are the challenges towards getting those better drug candidates into phase 1. And for us, we believe this is about having good human relevant models. Now, another point that Yvonne did touch on, and certainly an area that we've had experience about as well, is that species-specific toxicity really does play a big role in how drugs are developed. Now, you know, I am a trained in vivo pharmacologist. I, for one, would definitely say that animals do provide value in the drug discovery process, but there are very many documented cases where animals actually haven't provided us that safety assurance that we were looking for. And thiouridine is often cited as a good example of this, and for the chemists in the audience, they'll recognize that this particular molecule is really a mimetic of adenosine. And what happened was it was being developed for hepatitis. It went through the clinical trial, through the animal models, and no effect was seen, but unfortunately 5 out of 15 clinical trial participants were unfortunately died as a consequence of taking this drug. Subsequently, it was found that because it's a mimetic of adenosine, it interacts with the ENT2 transporter, and there are species differences there. And that was largely responsible for why this drug does cause steatosis in human, but not in animals. And we actually showed in a publication in 2019 that actually rat chips, so rat model, would also have failed us at this point. So if we are using rat chips early in the discovery process, one does still have to be mindful of species differences. Now, we've heard a lot in this meeting about interesting science, and we've heard several people, Don Ingbert and Mike Schuler again this morning, Jay Hickman yesterday, talking about the fact that actually our technology, microphysiological systems, is actually coming of age, and it is moving from just interesting science to IND submission. And I think, again, I want to bring us back to this concept that we're focusing on building models that actually get better candidates into phase 1. So with that in mind, what we're looking at predominantly, and my guidance perhaps and conversation I'd like to have with this team here, is that picking the right model and building the confidence that human relevant models will drive better candidate progression relies on, first of all, developing a good model system. There are guidelines out there. I think we could benefit from more guidelines. Several of you have actually mentioned that to me during the course of this particular meeting. Experimental design is absolutely critical. Understanding how many drugs to test the model system on, how many donors, perhaps to look for reproducibility. Important, I'm a pharmacologist, so adjustment for protein binding is also essential. And good hygiene in terms of blinding scientists when they're preparing these data sets are, again, going to be key to developing good models. And finally, once we have got those two things, we need to, of course, evaluate the performance. And many of you know that we've been doing this in Emulate. It is on bioarchives. We're focusing on the liver. We've looked at 27 small molecule drugs now across 3 donors. And repeatedly, we see a very high sensitivity in the ability of our model to correctly identify hepatotoxic drugs that have been in the clinics. This is a retrospective analysis. And because we know they've been in the clinic, we can say that if the animal model did detect a hepatotoxicity, it was certainly not used in the decision-making to prevent that drug from going into the clinic. So here we show the performance of these types of models, and of course more would need to be done. So lastly, I've talked about progression of molecules into phase 1, and where I would advocate then a model such as ours would actually reside. And we firmly believe that this model resides in the lead optimization process, and importantly should coexist with the animal model. So I think that might surprise some people in this room. I'm going to say that for the time being, these models can coexist. But we believe that if you place this before your early dose range finding studies, there is the potential for us to start addressing whether animals are actually even needed. If we see a toxicity in our liver chip, then our recommendation is that is a real stop and think moment. In summary then, if you've developed the right model, you understand its context of use and you're using it to progress molecules into phase 1, we do believe that this will cause a reduction in the failure rate we see in the clinic. Thank you very much.
Thanks so much. Working.
Yeah, so Lorna, thank you so much, and I appreciate the perspective and the commercial for Emulate as well, right? So who's probably a sponsor, right? So briefly, I think what we need to really think about is the cost and the need and the volume of testing that needs to be done. And I think it's totally fine to have fantastic systems that can be inserted surgically in a particular point in either drug safety development or in any other safety paradigm. But to kid ourselves that this beautiful science that all of you and us are doing is going to replace animals, I think is preposterous. And I hope that we have a conversation about that.
I'll just adjust the microphone down. So, thank you, Yvonne, for those comments. I won't directly rebut those at the moment, but I will rebut some items I saw in your presentation. So you talked about misleading statements for translation, and I do actually sadly agree with you on that point, and I think we should have a bigger discussion about how one uses the data that's available to us. So for example, folks sitting in the audience that are from the pharma industry know that actually some of this data isn't published on a regular basis. It's quite out of date. It doesn't reflect therapeutic indications, nor does it reflect therapeutic classes. So I think if we could get greater granularity on that data, I think it would certainly help us understand whether our statements are really true or genuinely misleading. And my other comment really to you as well is that we're all learning here, and I know that I've seen John Wicks go somewhere in the audience. I saw him earlier, he's got a beautiful pink jacket on, but I can't see him right now. And so John, I am going to steal one of your quotes. So you talked about models being wrong, and actually I do again agree with you on that point. A model is a model, and John has a beautiful way of describing this, which goes along the lines of, if you want to model a cat, you need a cat, preferably that cat. So I shall leave you with that and pass over to Annie.
Okay, good. I got it now. So I'm Annie Moisin, Program Director for HOPE, so Human Organs, Physiology and Engineering at Wellcome Leap. Prior to that, I was in mechanistic safety at Roche for many years and developing MPS and also running standard toxicology assays. So from both angles in my work. So thank you, Ivan and Lorna, for the evidence, the numbers, and so on. I think I'll be the simple-minded one here, perhaps, and bring this question down to fundamental biology. Species are different. And it's a fact that species-specific expression level of even a single molecule on a given cell type can lead to dramatically different body responses. So illustrated here, the blue cell type you see was activated by an investigational new drug in human, and it wasn't activated in the monkey. Consequence, catastrophic organ failure in 6 men at a dose 500 times lower in men than in the non-human primate before. So I'm referring, of course, to the infamous TGenero trial. So in that case, the CD28 superagonist antibody activated Tregs and promoted tolerance in animals, switched on T cell helper T cells in humans, so triggering a cytokine storm. And this is not just one anecdote, so we've seen more of that. Do I switch slide? Okay, yes, yeah, and so let's be a bit more forward-looking now. So there are more biologics to come, not only in the pharma industry, but also in agrochemicals. And generally speaking, and more biologics then will mean more species-specific or human-specific reactions. Generally speaking, you all know that compared to small molecules, chemicals, biologics are most of the time more target specific or target specific to their target sequences, and often they don't even cross-react with animals. And that poses a great challenge, of course, for preclinical development if we don't have good human-based models around. And second, unlike small molecules, biologics are bigger, highly engineered, and often they are recognized as foreign bodies by the immune system. What happens, there's a T cell dependent antibody response. The antibodies can alter the PK, the PD of the drug, compromise efficacy, compromise safety, and this is very poorly predicted with animal models. So we need human models for that. I'm not an expert, certainly not in agrochemicals, but seeing the growth rate of biologicals, and most of them, I think 50% being microorganisms, microbiomes, I really wonder, depending on the exposure, of course, what's the risk for our microbiomes? And microbiome, that's also an area where, forget about animal models there, we need human systems. So we've learned for many years that in terms of immune biology, mice and humans are very different. And we are learning now with more studies, and there's a recent one from 2022 that we are also quite far away from monkeys. We do have similarities, of course, but if you look at level of divergence, and now we are learning about cell identity, cell type frequencies, expression level of key receptor for drug development. Just some examples here: MHC, FC gamma receptor, the STAT pathway, interleukin, and so on. I think there are sufficient evidence that either we start also humanizing non-human primates like we do with mouse, because if we want mouse to be predictive, we humanize them, right? So shall we do that? I don't think so. I think we should just go ahead with human-based systems. And so we can use blood, human blood. That has some limitation, of course. We can look at acute responses with fresh blood, some cytokine release, for example. But you've probably, you heard that also this morning in the session, immunity occurs in tissue. That's where antigens are sampled. That's where immune cells crosstalk and they meet, and they mount the adaptive and innate immunity over time. So what we need, if we want to recapitulate the human immune response and then be more predictive, we need to create this tissue microenvironment, seeing immune cells being infiltrated, recruited, and responding over time. So we need longevity. And the good news is MPS can actually do that. I think we have sufficient demonstrations from yesterday, this morning as well. We'll see more pointing towards feasibility. We thought it was impossible. I think it's getting really possible to do. And I think it's not really scientifically sound nor ethical to try, fully humanize a mouse or a monkey. It's not an animal anymore. So let's just push forward and bring MPS to the next level. And the time is right to do that, because now we can read tissues thanks to spatial multiomics, single cell analysis like never before. So we can extract the information we need as guiding principle for engineering these tissues. It requires resources and tenacity, of course, but I really do believe that MPS is the future, and I hope we have the courage to address the human relevant question with complex models like MPS.
Thanks, Annie. Final speaker will be Jason.
Yeah, hi everyone. Yeah, Jason Eckert. I lead the complex in vitro model group at GSK. This will be my opinions and thoughts on this area. Okay, perfect. So where I stand, I think it depends. So when I say it depends, it comes back to the context of use. It has to be dependent on what you're looking to do with a system or a animal. And I put this up here. This links with some of what Ivan was talking about. So on the right here, looking at clinical experiences, and this was a recent study in the biopharm space, looking at how our failure rate, which in the drug discovery is quite poor. And then on the left here, where I'm talking about from thinking of the context of use, and this is from a recent paper where there was a workshop with the FDA and then the IQ MPS affiliate, we put here different buckets where we see the potential for MPS in the future. So where we're hoping to push the limits of having MPS earlier in discovery, and then also bridging that gap where Lorna was saying, can we use MPS to help better predict early toxicity? And then also that rather than potentially replacing straight away, it will reduce the number of animals. And then eventually, and this, if you read this paper, we talk a lot about depending on the context of use, for example, in the safety or ADME space, it's going to be a weight of evidence where there's going to be an increasing amount of MPS data and that will support the animal modeling as well. So where I wanted to show two different areas where from a context of use, and this also links with quite nicely with Annie's comments about biologics and also looking at cell therapies, is on the right here, there's a plethora of different models where GSK has been working on in the oncology space. So from tumor spheroids, which have been around for decades, to more recently using tumor organoids, which are a static system, where we've been using tumor organoids to look at drug-drug interactions. So that's moving closer to the patient where we can look at these drug-drug interactions rather than waiting to go into the clinic. And then that can help determine what potential animal models we could use. So where we could do a organoid model, and then you can actually do a PDXO model in an animal. So you still potentially may need an animal before then increasing your confidence of then going into the clinic. And then as you see going down the diagram, there's increasing complexity of these models where you'll have a multi-layered system where we've been using a triculture system, and this was looking at PARP inhibitors, and then vascular models where we've been using models where you have tumor cells embedded with a vascular system, and also where we've been looking at movement of T cells across endothelial barriers on the right here. So it depends, as I said, on the context of use. And then you get right to the bottom here where you can have microfluidic models that have an ex vivo tumor tissue, and this will be even closer to the clinic. And I have on the left here, and I've crossed it out, you can't see it, hiding behind that X is the animal. And I think eventually that's where we want to be, but we're not there yet. And then just one switching gears, and this links with what Lorna was talking about from a liver in vitro models. We've been developing a number of different liver models within GSK, and from where we've had in the past using the sandwich culture systems. We've used liver spheroids, and we've used the liver spheroids quite successfully looking at early hepatotoxicity. And so this has helped drive using a battery of different assays along with the liver spheroid models to better predict which models are going to have that hepatotoxicity. And so therefore we will stop the progression of those molecules depending on that battery of assays. And then on the far right, you've got different liver on a chip models. And we've been using these to look at different diseases such as NASH, and you could use it in the infectious space for HPV. And so for those models, you have to think about from a cellular context and cellular architecture of what cells you would need when developing that model. So from my point of view, we're moving in the right direction, but we're not quite there yet. Thank you.
I see Ivan has his hand up, so. Yeah, I mean, I just want to thank all of you first before we carry on, so I don't have too much to say on top of that, but I want to also encourage people to ask questions now. I know Ivan has a rebuttal or a comment, but please, what is your opinion? What do you think? Are there people in here that believe we don't need animals? So please be active, but over to you first, Ivan.
Well, thank you all again for speaking up your mind. And as Phil started at the beginning, we really do not disagree, right? And this is not a debate about animals, right? This is really, I think, a conversation about where we go, where we are, and are we there yet, or how soon will it take for us to get there? So I certainly don't think that in my lifetime we'll be anywhere with, emulate. The only thing that is on the drug development path is emulate chips, or any other chips for that matter. But I think I wanted to come back to a couple of points that Annie made, that MPS is the future. I think for certain things, absolutely, that they will be used as was shown in drug development in certain places, absolutely. And I think we all need to develop basic research and move this forward. However, we need not to forget about the reality, and the realities are challenging. Realities are challenging in terms of cost. Realities are challenging in terms of throughput. Realities are most challenging about the cell availability. Realities are challenging about reproducibility and how many experiments you need to run to really, have statistical power to make any conclusions. And to do that, I think the group think need to occur. And I think the developers and the users need to, again, do a better job educating each other about what the needs are and what the challenges are. And that's what we're hopefully going to have a conversation about today.
Anybody else? Are there any questions? Please go ahead.
Yeah, when I saw the title of this, I thought we were going to actually be talking about different MPS models rather than whether or not animals will be part of the pipeline. So I wanted to maybe see if the panel might think about the topic that when I see the vast number of different MPS models that are being developed, it seems so much larger than the number of animals models that we have: mice, non-human primates, rabbits. And so how do we think about the hundreds and hundreds of different MPS models that are being developed? How are they going to sort of fit into the pipeline? I'm not quite sure I know the answer to that. I would be interested to see what you guys think.
Yeah, I can take a stab at it first. It also will be dependent on if it's fit for purpose or context of use. If it's in the safety space, I think where there's discussions through the IQ MPS affiliate and with regulators, is that we need certain standards to start off with. And then with those standards, depending on what your specific use case is, that you then may have to have further standards or certain criteria that you have to set up.
Maybe just a quick comment. We tend to think that we have mice, rats, non-human primates. These are preclinical models, but think of all the strains, that you can choose. You really choose also your mouse specifically for the question you want to address, and often you keep changing the strains until you get the answer that you really want. That's true. So there's quite a range of I think animal models as well. Very wide range here. Yeah, just a quick comment.
And I actually think it was an excellent question. Thank you for asking it. And I think one thing I would bring us back to is folks who sat through Mike Schuler's presentation this morning had a really nice history lesson on where a lot of these concepts started. But I think most people in the room would agree that while there are good examples of these types of models being developed 20, maybe 25 years ago, the whole field's really come of age in the last 10 years or so. So I think at the moment, yes, you are seeing lots of different ways. The example on the slide there is great, it shows lots of different availability of liver chips, I think, with standardization. And I think as we really put those models through their paces, what we'll find is that there'll be some models that are better, and we can discuss what we mean by better, of course. and those will be the survivors, if you will, as we go forward. So I see it maybe a wee bit differently from yourself, and I don't think we're going to advocate for having 10 liver MPS models in the drug discovery process, for example, but I think through time we will eventually see one or two that are more superior and to Jason's point, fit the context of use.
I also had, one of my questions would have been on that similar line, that we have a lot of models, we've heard about a lot of them, but one of your first slides, Ivan, in talks, we need to look at, well, you mentioned 45, but I guess normally 30 something organs. We don't have enough models for most of those systems. So we're a long way from reality, from talks, to actually replace an animal.
But also, I think what we need to think about is what does success look like? And to many developers of new models, success only is true if you're replacing something or if you knock something off its existing position. And it's one way to look at it. But I think what we're seeing in biology in general, and biomedical engineering is not the only field that is undergoing tremendous expansion, is that it's okay for scientists to try different things, to try hundreds, thousands of different models. But scientists at the same time need to understand what the utility of those models will be to increase the chance of creating something that will be translatable. Because as I've shown you in one of my first slides, just declaring that something has great promise really is very opportunistic. So if we lower the temperature and if the success doesn't necessarily look like FDA now starts accepting these data or model X replaces model Y, then we can all coexist and all walk together to the future, which again, hopefully I retire by then.
Thomas?
So I really like the role of an agent provocateur, which Ivan did take here. And I'm so relieved that all the others which have background from pharmaceutical industry were so much more positive about MPS, because that's what is really driving their use. I think we should, the problem we are doing is any type of black and white. To say it is either animal or MPS is simply not realistic. If you're spending upwards of 3 billion for getting a successful drug to the market, every model which will change the odds of getting something faster or better out will be used and should be used because it is increasing safety. But I think we should also not say it is an abuse of data, then cite Olson paper of 2000, which shows that rodents only found 43% of the adverse effects of drugs. The Olson paper was praised for how well animal models helped us. For most substances which are not drugs, we only have rodent data, and 43% is simply not enough for me. I would really like to have systems which are finding more of these, and at the same time, I think we're also killing people because we're killing substances with wrong animal data. And this is not only an animal problem. I don't want to know what the genotox test battery has done to drug candidates because we are sorting these out in the development process with something which typically more than 90% does not hold as soon as you go into the animal. So we have a problem, and this problem we can only solve by more cleverly integrating the various approaches. One of my most recent papers I have entitled A Call for In Vivitrosi, in vivo, in vitro, and in silico combined. And that's actually where I see the future. And MPS somewhere is in the abbreviation as well, but I think is a driving force for making this actually a very successful mix. So one was no question, but a comment from my side.
Can I start with it? Thomas, thank you so much. And again, I'll have to reread the Olson paper to fact check the 43%. But I wonder, what is the alternative? You know, we all talk about, you know, a case example of one clinical trial, you know, killing 6 people. Thalidomide is a favorite for animal abolitionists in toxicology. Those case examples do exist, and no model is perfect. You know, engineers are great at engineering best things. Cars still break down, rockets blow up. things will fail for reasons that are uncontrollable by us. And what I am concerned is that we need to treat people type of argument. We need to have better data on people, that we're actually talking about either not testing anything and throwing it straight into people, or testing it on MPS or any other in vitro system and then going straight to people. I am concerned that people will become test species for anything, drugs or chemicals, without any filter before. And I think, unfortunately, we're seeing the practice of this with cosmetics. Cosmetics cannot be tested in animals anymore. And yeah, I don't care about cosmetics. There's enough perfumes and ingredients and other things. But if we're going with politicians pushing us the same way on agricultural chemicals and industrial chemicals on drugs, I have a real problem with that. I don't want to be the test for something that could have been tested in other models, including MPS.
So Ivan, I'm going to rebut you on that in relation to some of the comments you also made regarding the work that we're doing. And I think you did use a word that I use, so I take that as a compliment, which is coexistence. And I think what Thomas is saying, what I'm saying, and other people in the room will say, is that we're not saying today we're ready to replace the animal and we'll remove the animal. I think we actually have to go through a process of learning about these models. And that was why I was advocating the approach that I align myself to now is thinking about good experimental design and the pursuit of data that will really allow us to understand whether these models are part of what I would term that integrated risk assessment. Now, I take my inspiration from the work that's been done in the cardiovascular safety pharmacology field. So again, we can go back to the 1980s when drugs were withdrawn because of the torsades. And it was several years later that the molecular entity, the hERG channel, was identified. It was 10 years after the identification of the hERG channel that there was then some regulatory guidance for the inclusion of a GLP hERG study. If I bring that right up to date, that community have worked hand in hand with the regulators, some of whom may be in the room, and are now saying that it's an integrated risk assessment with a hERG, with the dog in vivo cardiovascular study, with the SEPA initiative, looking at the other other ion channels, I beg your pardon, that drive the action potential, and importantly still a clinical thorough QT study. Okay, and it's when you see negatives across all of those that's what drives the confidence that that is safe to progress into humans. And I would like us to use that as our inspiration of when we're building our new models. We focus a lot at the moment on safety, but remember these models will be good for efficacy as well. They will help us in areas beyond drugs, but I think we should use that learning and how we can maybe shortcut some of that time process that it took from the 1980s, where we discovered torsades, to today, where we're now maybe getting into a place where regulators and pharma can coexist. So for me, coexistence is the key.
Yeah, just one additional point to Thomas's. I think it's a great initiative or thought of where I know GSK is probably not the only pharma company where we're talking about in silico modeling group, both in the safety, so looking at potentially how you can incorporate into adverse outcome pathways, and then also from a PK or PD point of view, when we're modeling for dose prediction, we're speaking with them as well. And also looking at from a quantitative systems pharmacology approach to understand what cellular components do we want to have when modeling our MPS models.
I want to follow up a little bit on what Thomas mentioned at the end of his question. And I very much appreciate the discussion on these models, but we are talking experimental models here, and these models are not run in a vacuum. It seems to me we're missing a big component, which you just kind of mentioned about modeling, which is the computational analytics end of this, which is, as we know, going forward into omics type analysis is becoming more and more not only important, but critical, key components. So I'm thinking that these MPS models, or maybe even animal models, can't be run as a standalone answer anymore. That has to be integrated with a more and more sophisticated computational approaches. Maybe you can address that. Thank you.
Well, I wonder if I can, Larry, maybe expand on that and ask my colleagues who've been at pharma or are at pharma, did the recent example of omics, did that really, speed things up, lower the cost? improved safety profiles, realistically? It's been 15 years of genomics,
I'm
right?
sure you'll get a different answer from every farmer. I know at Merck, I would say no. But I would also argue that after those 15 years, now it's a standard tool where you can take it out of the toolbox and you will run it. And I do agree myself that all the data that's been generated in these systems will rely on some sort of, I hate the word AI because you use that for everything these days, but that has to be an integral component to understand all this stuff. It will never go anywhere.
Yeah, and I know that I've spoken with a couple of people here. For me, the modelling side of things actually will help drive the improvement in some of the wet biology models. So less about the omics, Yvonne, but one thing that I'm particularly interested in is how we can use mathematical modelling to help us understand the right model to build. And so I was having a conversation with someone earlier about the hepatocyte. If I produce a hepatocyte in my liver today, it will be in my liver for the next 9 months. And, what we have to ask ourselves is, do we therefore need experimental models, in vitro MPS type models that last for 9 months? And I don't know if we can actually genuinely answer that question. I don't know if Adrian's in the room, but Adrian for many years ago had a nice slide up that they were culturing hepatocytes over time. And what they found was that those hepatocytes became less hepatocyte-like during the culture period. And so I'd like us to use mathematical modeling to help us understand the kinetics of the biological response and use that in a good way to design the right model. And ask ourselves the question, do we really need 9 months of hepatocyte viability in a model system to allow us to really replicate what's happening in vivo? So I think there is multiple areas where mathematical modeling and AI type approaches can actually help enhance what we're all trying to do.
And also to the omics, omics was mentioned, I think, in the identification of better biomarkers. That's what we need to do, also to have a non-biased integrated approach to biomarkers, because we are constantly looking for biomarkers that are predictive of responses, and it cannot be a single biomarker giving us the answer. So just going like fresh reset and then look at the data with the new technologies that are available now.
Conceptually, this is great, but I'm not sure how many of us have looked at the bottom line and the budgets that we have. So conceptually, everything, throwing everything and integrating, right? It's fantastic, but the reality bites, unfortunately. And I think that's should be something that everyone considers as well. Is what is the most useful model for the purpose that I have? And regardless of how much beautiful stuff is out there, we probably will not use 99% of it.
So I would say over this.
So this debate about what model is best and what model is most purposeful, I think it can be settled at least partially if we can come with some ground rules as to how we are going to validate the model. Now, if you're making a microphysiological system and you want to go and publish this in JCI or any other traditional high impact journal, for example, or you're putting a grant, it's almost very common that there is going to be at least one reviewer who's going to ask you to validate your model with a mouse model. Now you can do an entire study with the mouse model and go publish in Nature, and there's going to be no reviewer who's going to be asking you to validate a mouse model. So this dogma that a mouse model is physiological has to be broken. Now that I think is more preposterous than anything, that even though we know that first slide that you showed, Ivan, that mouse model is considered physiological. I think that should be part of this conversation as to how we can inform the community and how we can break this dogma. Only then other experimental models will thrive. And it is particularly important for early career and mid-career scientists, because if they want to grow and they want to build this field, then they really need this space where they're not tortured by this idea that they need to validate their models with the mouse model.
You're preaching to the converted.
No, I like the comment very much, and I think we need to bridge worlds, like the academic worlds, the pharma, and also regulators, because what happens when you submit a manuscript, it's going to be reviewed more by academic labs, PIs, and some lack of awareness of how we actually back translate and validate the models using preclinical data, using clinical data. And why? Because this is also not public. So there's a problem maybe also of sharing approaches. So how in pharma do we think a validation should work? And then what is happening in really in academic labs? That's really two, unfortunately, often separate worlds. So bringing these communities together first and then raise the awarenesses doesn't make sense to validate or to compare to the mouse if I'm developing a human model. We need to improve that.
So I appreciate this comment about reviewer number 3, because exactly the same argument I've heard in a couple of different meetings in the last year, that the reviewer asked us to compare this to some, in vivo model, and this was the wrong question. I don't think it's a validation question, it's the relevance question. There's nothing wrong with asking, how does your model, whether it's on flat plastic or 3D or something else, how does it replicate something real? And perhaps reviewer 3 was inartful in explaining that, but to me, it's not a validation question, it's a relevance question. And relevance is a very important scientific concept.
So Ivan, maybe a question back to you about, I know the flip side could be comparison to a 2D. A lot of the work that you did in for the validation center was the 2D. Was there thoughts of having a different comparator?
So it depends on, so Jason mentioned, we have a Texas A&M tissue chip testing consortium, which originally was started by funding from NCATS, and now we have 8 members who contribute each year, and we do experiments together to try different things. So in our point, you have to have some sort of an anchor. And if you have an anchor to human physiology or to animal physiology, you'll be benchmarking to that. But frequently the anchor or the benchmark is the status quo or standard of care, right? Just like in drug development, you're always or almost always comparing your drug to the standard of care. The similar concept applies in MPS testing. If we would have a particular MPS replicate a particular human condition, and if we would have enough replicates or to run multiple drugs, perhaps you can do that. But in absence of throughput and in presence of very high cost and complexity, you can only compare to 96,384 well plate, which everybody does and makes decisions. And we are here today because of those models.
We have the time, but.
Hi, I'm Chris Boyer from Tessara Therapeutics. I'd like to pick up on the question of which specific MPS model would choose. And Lorna, you talked about a shakeout that is likely to occur. You also talked about what factors determine the best model. And we can think about reproducibility, we can think about cost, physiological relevance, other factors, but certainly one of them is the reference data set behind the particular model. And that seems to be one that's cumulative and sort of self-reinforcing. And in the end, it becomes almost like a network effect that you want to test your compound in that model just because it has such an impressive reference set, and you can make inferences based on the other molecules that have gone through. So I'm interested in your thoughts and the panel's thoughts on how that reference data set would probably become the dominant factor determining which MPS systems go forward.
Yeah, so I'll maybe explain a little bit in detail about what we did. So we used guidance from a third party, it's the IQ MPS Consortia, who actually published a manuscript in 2020 and said, if you are going to develop a liver microphysiological system and look for drug-induced liver injury, there are 20 drugs that we would recommend that you test. Now, what was in those 20 drugs, a couple of features. First of all, they represent the major mechanisms of drug-induced liver injury in the small molecule therapeutic class. secondarily, they had all been in the clinic. So there was, you know, that true outcome, or if you will, there was the ground truth was, what did it do in the human? But they also asked for, they gathered together 7 matched pairs where these were structural analogues, and one of them, so trovafloxacin and levofloxacin is a good example, where trovafloxacin has been shown to be far more toxic in the clinic than levofloxacin. So they were also asking for the ability of a model to start to distinguish those. And that's important because, as you know, in pharma, quite often you're rank ordering, you're trying to promote and progress the most promising candidate within a chemical series. So there was 20 drugs there, and we supplemented. When we did our study, we used 27 because we supplemented with some other ones that we were interested in, that other in vitro models had failed to potentially correctly label as toxic or not. So there isn't a consensus data set. There's certainly not a consensus number of drugs, but we believe that it's one of the largest studies, single studies that's been conducted to date. And I've spoken with a number of people who have asked if we can make that data set more available to do exactly as you've said, and use that to start benchmarking own internal compounds. But if we take inspiration from outside of the pharma industry and perhaps look at some of the work that's been done by folks in the audience who are more on the chemical side, they are further forward and developing, you know, official guidance, in some cases OECD guidance, that says these are the test drugs that one should test, and build, you know, and show the relevance of the model to that. So I think we've still got a long way to go. I'm exceptionally proud of where we've taken our models so far, but fully recognize there's still a lot more work to be done.
In the true spirit of debate, I'll point out that I think it's a false premise to try to just define your reference set, and then after that, everything will fall into place. Yes, OECD test guidelines do have that, and that's the basic benchmarking of your model, but that doesn't guarantee acceptance. The folks at the National Toxicology Program have done a yeoman's work and tried to define something as what should be very straightforward: estrogen receptor agonists and antagonists and inactives. And they really went and spent a couple of years on that. And still, it's useful to show that there is promise, but it's not going to guarantee. Pretty much every QSAR model has 75% or 80% accuracy, but the domain of applicability of any application, 20 drugs stay in the drug lane, and only particular chemistry, do not go into agrochemicals or PFAS or anything like that. I think we need models that really have confidence, and that confidence will come in part from reference sets, but in part from just robust use, and robust use means throughput and cost.
Yeah, but costs mean that you need users to reduce the cost. So I think we should also not just like wait oh, but that's true. It's the wider adoption and application, and that will make the assay robust. And so we have to, at some point, also be confident and have the courage to use them and distribute them and test them and reduce the cost.
Just quickly, as a testing center, we calculate the cost and we compare different models, and the cost varies 1 or 2 orders of magnitude already. So it's not the users and then the prices will come down. Some things will never come down, and that's the very unfortunate truth.
So to add from a cost point of view, instead of going towards a more complex model where we went at GSK to using the liver spheroid model, and there's a paper, I think it's 2 or 3 years ago, by Will Proctor and others looking at, I think they had 150 compounds for looking at liver spheroids, and more recently, so Chris Schofield and others at GSK have a paper looking at 170 compounds. So it's depending on how many compounds you want to look at and different classes that you'll then understand the sensitivity and specificity.
Thank you.
So, I mean, I would just add on to that you'll never have enough compounds. You ask a statistician and they'll always say they want more. But I still think it's a good place to start. I mean, the more complex the model, the fewer compounds you will be able to test because of time and cost and resources. But it's that confidence. Once you start getting confidence, then you'll get more people testing it, then you generate more data, and then at some point, maybe in the near future, we'll be in a better way. But I'm looking at the watch, it's exactly 1:15, so I think we should stop there. I think there are no more questions in the audience. I want to say a big thank you to all of you. It was a nice discussion. And thanks, of course, to the 4 speakers up here. Thank you very much. I just wish you all a nice afternoon and see you soon. And I'll send to them next door that way. At lunch, there is lunch.
Okay, good afternoon, everybody. Bon appetito. You're very welcome to our session on the importance of standards for advancing the MPS field. My name is Maurice Whelan. I work for the European Commission's Joint Research Centre in Europe. I'm based in Italy, and the JRC is providing independent scientific support and advice to policymakers in the EU. I'd like to thank the organisers for facilitating us to work with them to organise this session on standards. And, you know, as the title of the session would imply, we absolutely do think that standards can play a very important role in advancing the field. Recently, I was reading in a policy document in Europe, and it had a statement simply saying that setting the standards sets the trend. And it's a very powerful statement. And I guess when we reflect on standards, we often think of the commercial aspects around standards. You know, needing to comply to standards, set the standard for gaining market share, to comply with regulatory requirements for entering the market, and so on. And indeed, standards do facilitate that translation to commercial applications. But I think what's less appreciated, you know, is the important role that standards play in actually driving innovation in the first place. You know, how they're a knowledge integrator, community builder, and very importantly, the relationship between standards and research, how more use of standards in research itself can make results better, more impactful, and equally how we need to put science into standards. So we really hope this session will give you some you know, useful insight into standards and understanding them better. We really hope that the discussion will encourage the MPS community to embrace standards and make standards an important aspect of this community's work going forward. So I just wanted to thank our speakers in advance for participating in the session. See the details here. So with no further ado, I'll move on to our first speaker, Monica Piergiovanni from my own group in JRC. Monica.
Hi, everyone, and have a good lunch. I'm very jealous that you're eating and I'm not. So I will start by introducing you how standards can support innovation, generally speaking, and then we will go a bit into the specific case of organ-on-chip. So Maurice already told you what's the mission of the Joint Research Center, so thanks, because I can skip that. But what we actually work on is a unit of chemical safety and alternative methods that incorporates ECVAM, which is the European Union Reference Laboratory for Alternatives to Animal Testing. And we are mandated by a directive to work into research, validation, and promotion of non-animal models in science. And what do we mean by non-animal models? Well, you know that there are a bunch of those, starting from in vitro 2D cultures, 3D, organ-on-chip. We then also talked about omics and high throughput screening in the past session. And of course, let's not forget about computational and mathematical modeling. And I think that the integration of all those tools is really relevant. But of course, let's not forget that the vast majority of these methods are basically in a very low technology readiness level, meaning that they are just starting out of the lab, of the research or university lab. So if we want to increase their use in the real world and bring them as tools that can be trusted, of course, we need to increase the scientific credibility and the technological confidence in those, so that the end users and the regulators, of course, will believe in the data that these methods produce. And here is where standardization and harmonization come into play. So we spoke a lot about the biological relevance of the models and the fact that they are becoming more and more complex. But we have to acknowledge that they are integrating more and more technology into those devices. And so we cannot avoid talking about standards. And by standards here, let's start by a simple definition. Standards are technical specifications that define requirements for products, production processes, services, or test methods. And let's remember that standards are generally a voluntary documents that are used by developers and end users. They become mandatory, of course, if they are incorporated into regulation. So in Europe, for instance, we have the medical device regulation that has an annex with a lot of standards that are required to prove the safety and the quality of the devices. Standards are, generally speaking, used for a broad variety of things. So they ensure interoperability and safety. They, of course, reduce costs if we are talking about mass production. And they also facilitate the integration of the companies with other companies and the value chain in general. But who is developing the standards? So you probably know this standards developing organization. We have, at the international level, we have the ISO, IEC, and ITU. We have some regional organization. In Europe, we have Sensenelec and ETSI. And of course, we have a lot of national bodies. And here I just put in the slide some examples from Europe. But are those the only ones that are developing standards? Of course not. If we go around us, there are many other organizations that are developing standards. Here I put just some examples. So from the medicinal domain, we have ICH guidelines, for instance. If we go more on the chemical safety, you for sure know, they were already mentioned, the OECD guidelines, for instance. And I just put the OECD test guidelines for the testing of chemicals as, I would say, one of the best examples of harmonization, because those test guidelines are actually internationally agreed and used to assess the safety of chemicals and products. Let's go now a bit talking about the role of standards for an innovation in general. So this means any technological innovation in the world. So for sure, the first role of standard is to build focus and cohesion in order to have a critical mass that can go on with the development of the technology. But I would say that the most important role of standard is the help that they can do in helping companies to demonstrate to the end user that their innovative process or technology really possess the characteristics that they claim. So this means that standards for measurement and tests will help the technology developer to assess the performance of their product. And a second, let's say, advantage of the use of standards is the guarantee that the product will have an acceptable risk for the safety, the health, and the environment. And of course, let's not forget that standards are the third pillar, together with scientific publication and patenting, in order to diffuse science and technology. Even if I have to admit, the scientists really treat the standards as a last resource after all of the others. This slide I took from our colleagues in Sensenelec, and I would like to guide you through it, because it's important to understand that for every level of development of the technology, we have a specific standard type that can help us. So I hope you are all familiar with the TRL, so the technology readiness level. If not, let's just start with the first. So the TRL level 1, when we are actually talking about the basic science, discovering the principle up there, up to the TRL 3, that's when we have a proof of concept in our lab. Here we are actually still developing the knowledge on the basis of the technology. And in this point in time, it's important that scientists can talk to each other. So in this point in time, it's important that the vocabulary and the terminology are agreed. And that's when we have a role of semantic standards. If we go on the development, so in the TRL 4 to 6, we are building our prototype, we have different components, we are integrating them, testing them in the lab or outside the lab. And here is when actually the measurement and testing standards can really help the development. So this means that we are not constraining let's say the fantasy or the ideas of the developers, but standards are only there to assess the performance of the technology. And finally, later on, of course, we have the interface standards that agree interoperability with the world around it. And when we go later on in the business development phase, of course, we have to use quality and safety standards that will go, around the business case that we are creating. So if we go back to the organ on chip, so to the MPS field, I hope that we all agree that standards, generally speaking, can demonstrate the technological and biological relevance of the devices and also increase the implementation by the industry. And for sure, we have to also acknowledge that they provide basis for regulatory acceptance. So that said, last year, we decided to go deeper into the organ on chip field. And we published this paper on lab on chip, which is what it's traditionally called a prenormative analysis. So we took the organ on chip field, we decoupled it in all the aspects of the technology that can be standardized, and we made a gap analysis, so an analysis of the status and the needs of standardization in the field. So you can, of course, have a look at the paper, but I would just point out that the paper is accompanied by a database of 87 already published standards that are developed in other fields, but can be somehow related to the organ on chip technology. And those were published and developed by organizations like ISO, ISTM, IEEE, and so on. So I encourage you to go there and have a look at those documents so that you can maybe try to understand and get an idea of how a standard is built. And if yes, you can then contribute to the development of standards for organ on chip. Following up on this analysis, we organized last year the Putting Science into Standards workshop on organ on chip, which maybe some of you remember. And this was the first time, I think, where we brought together not only developers and end users, but also the standards developing organizations in Europe, which are, of course, as I mentioned, CEN-CENELEC. And I will just now go briefly into some of the main highlights of those workshops to let you understand that we are not starting from zero. So there are, of course, some things that still need to be addressed, but where the work is processing is going on. So first, we all agree that the biological models is the first component of an organ on chip. And standardization here can play a role in the definition and the protocols of manipulation of cells and tissue. So if we talk about IPS or cells from a patient, we of course need to have SOPs. So we have to have a clear way to manipulate those. And a similar concept is valid for the media formulation. So this means that we should try not to standardize a media, because that's of course not possible, but at least to clarify and declare all of the chemical components of those. And similarly, we can also talk about biomaterials, because usually we do incorporate collagen or matrices or scaffolds inside, but very rarely the performances and mechanical characteristics of those materials are clarified. Then, of course, we would like our biological model to reproduce an organ, but what's the characterization data set that we have to have for each organ. So we need a list of biomarkers and other indicators of the activity of the organ. And here I have to say that we are quite advanced in that, because we already mentioned several times the IQ MPS consortium papers that are a great example of this standardization need. And finally, I want to highlight again the need of reference compounds. So those lists of compounds that can guarantee that we are working in the context of use that we are declaring. And if you would like to know more, here, unfortunately, I forgot to put the link, but on our website, we have what we call the JRC list of lists, where that's a curated databases of compounds for some context of use. So if you want to go there, maybe you can find something that's useful for your application. Another part, another component of standardization that's maybe sometimes a bit underestimated by the scientists is the reporting of the methods and of the data. So it's agreed in the regulatory field, it's an agreed practice to use reporting templates, standard reporting templates, to communicate the data of the test method that's used and the data that are produced to the regulators. So this is also a form of standardization, because of course agreeing to a test method template means that we have to set down the specific characteristics of the MPS. And in this slide, I think that my personal opinion is where standards have the most advantage. So to assess the performance of the device. So it's very important to have a definition of the material, of the chemical properties, of the mechanical properties of the materials. And all of these can, of course, be fulfilled by also a standard document that puts down some requirements. If we take, for example, the molecule adsorption, so we all know that this happens. We all know that we have to face it, but there should be a way that we can calculate it, estimate it, if we cannot at least avoid it. And of course, if we are integrated sensor inside our organ on chip, we need to have a proper calibration and sensitivity. And this is also something that in the standardization field is very, very usefully tackled. And I will not go into the fluid flow and microfluidic things, because you are going to listen to somebody more experienced than me. And finally, the usability. So in this field, it's very important when we want to transfer a technology to another end user, to make this end user able to use it easily. So if we work with standards to ensure the compatibility with laboratory equipment, but also with organ on chip of different solutions, we can, of course, increase the reproducibility of the data that are produced. So if you want to know more about the outcome of those workshops, you can, of course, go here into these two publications that came out last year. I would just like to finish up and pass the words to the next speaker by saying that the main outcome of this workshop was probably the fact that Sensenelec decided to establish an organ-on-chip focus group, which is actually the first time when they will like to start formal activity in standardization in the field. So with this, I will leave you here my contacts, because I think it's a bit awkward to go there and scan the badge. And yeah, buon appetito again.
Thanks, Monica. And we have time for a quick question, if there's any question. No? Okay, you're off the hook. Okay, thanks, Monica.
Bye.
So our next speaker, speaking actually of the Sensenelec focus group, the newly elected chair, Andries van der Meer, is giving the next talk, but Andries is from the University of Twente and he's going to talk about an open platform technology for organs on chips. Andries, over to you.
Thanks a lot. Thanks for the introduction, Maurice. And indeed, I think it's very nice that Monica ended her presentation with the establishment of the focus group by Sen Senelec. And indeed, like Marie says, I am the chair of this focus group. Personally, I am, as you see, affiliated with university. I'm in academia. I am a developer of organ-on-chip technology. And for me, definitely standards is a key aspect, I think, also of our dissemination. So Monica mentioned that for a lot of scientists, it may be a little bit of an afterthought. For me, it's really an integral aspect, I think, of doing our work. So very briefly, I wanted to talk about two aspects basically in this talk, very briefly about standards, because it was already nicely introduced, I think, by Monica, but also afterwards about open technology. I showed this figure. This is a figure from a European project called ORCID, Organ-on-Chip in Development. If you want to know more about that project, you can check out the website there. The project has ended, but the key output actually of this project was the establishment of the European Organ-on-Chip Society. And I took this figure from one of the reports of this ORCID consortium, in which a roadmap is given, as you see from the left-hand side, initial development all the way to implementation application of organ-on-chip technology. And what are the key aspects and steps along the way? And standardization obviously is one of those key steps that was identified by this broad consortium. And well, again, Monica has introduced all the efforts currently underway in Europe when it comes to standardization in organs on chips. If you would like more information about this focus group, and I think also it's good to clarify that a focus group is something different from a technical committee. That's something I realize already quite often, that people think that we're already developing key standards for organs on chips. That's not what this focus group is about. It is about finding priority, timing, synergy with ongoing activities. As Monica very nicely mentioned, of course, there's so much already going on in this domain. So we try and involve all stakeholders in this focus group to identify in the coming year and a half, where to take action as soon as possible. And the nice thing, of course, is that we do this now in the context of Sen and Senelec. So we have also their listening ear, they're watching closely. How can they help us and support us? And where is the first steps that we need to be taking for standardization in organ on chip? If you feel that you, like I just listed, let's say a random group or word cloud here, but if you have opinions or you think you have a strong opinion, something to say about some of these aspects, definitely this is an open focus group. So feel free to just go to that website, reach out, and become part also of this initiative. Okay, so like I said, I wanted to only touch very briefly on that standards aspect, because I wanted to highlight something else from this same figure that I showed before. And that's also the aspect that I mentioned in the title, and that is the importance for open technology platforms that was highlighted and acknowledged by this European consortium. And I wanted to sort of educate you a bit. And of course, it's very, let's say, very much intertwined, I would say, with standards and standardization, but maybe it'd be good to learn something about open technology platforms. So if we're going to be talking about what a platform is, or what what open technology platforms are, then we start maybe with the definition of a platform. And I think this is important because we throw this term out a lot, saying that we are all working on organ on chip platforms. But like if we make it explicit, then a platform, it comprises a set of components with shared interfacing rules, from which you can, by combining them and interfacing them, build higher level applications. So this is, of course, a very broad definition of a platform, but this is what we're talking about. And I think another very important aspect of of platforms is that it always includes multiple parties coming together, right? So typically you could define the roles that are important in the definition and use of a platform into these groups. So users, developers, manufacturers of components of that platform. And then that final category, sponsors, is basically a term that defines groups of people, stakeholders, that define the interfacing rules, the components, like what is actually what are the components that make up this platform. So they don't necessarily need to just produce anything, but they have a very strong say in what that platform should look like. And the interaction between these groups can take multiple forms. And I just plotted, let's say, visually 2 ways of how these groups can come together to define a platform, right? And on the left-hand side, we see, I think, a very collaborative way. All these groups come together and actually together define something that then in the end becomes the platform. But what we see quite often, of course, is that there's one party, as we see on the right-hand side, that maybe has the role or integrates all the aspects of the platform developer, the platform sponsor, manufacturer. So they define actually what all the components will look like, and then just make this available to users and listen to users what they would like to get out of it. So this is just, let's say, a framework of thinking a bit about these platforms. And then it's going to be important if we then transition into thinking about open platform technology in a minute. So if we think about platform technology and apply that more to this domain of organs on chips, so I just try to outline it a little bit more general, but if we now apply this to organs on chips, then it's clear that maybe in a very implicit way, we are, of course, very much used to thinking about platforms in organs on chips. And what is interesting there is that stems also from a certain need, right? So on the one hand, you have people that are using organ on chips systems, and they are focused on certain functionality, getting physiologically relevant systems that are easy to use. But on the other hand, of course, we have developers and engineers, scientists developing new organs on chips. And for them, it's important that the systems that they're working with are flexible in the sense that you can integrate new functionality, that you can maybe just replace components and tweak the system to fit the new purposes that are demanded by users. So thinking about the field of organ on chip and thinking about the role of platforms there is that it stems from this very strong need for ease of use as well as flexibility. And this is, of course, where let's say a system that consists of components that come together via interfacing will actually give that power. So to make that maybe slightly more concrete, like, so we actually have quite a few examples, and I just listed 3 random companies here, and we see a lot more actually in the meeting this week that have made their own organ on chip platforms and designed it. And these are all proprietary platforms that I'm showing here on the slide. And typically, we can identify key components of such a platform. For example, here, like working our way down the columns from left to right, is that typically you have chips, and those chips go into sort of like holders, and the holders then can be clicked into a system, etc. Right? So this is a very, again, like a modular component-driven way of building your platform. And all of these aspects, of course, then require certain ways of interfacing. So I think this is typical, of course, what we see here with these proprietary organ-on-chip platforms is typical for what I showed you just before, is that these platforms, in a way, they are very much open for users. And like, these are companies, so yeah, users are their customers. And of course, like, they will work hard to make these products usable and accessible for their customers, for their users, make sure that it fits the needs of these users, etc. But they are very much closed, of course, for external developers, manufacturers, or sponsors. All of that, of course, is just kept in-house by that company, and they decide what the platform will actually look like. So that comes, of course, or brings certain challenges. So that means that whenever we pick a component from one of these proprietary platforms, and again, I just show an example here of an emulate chip, and then try and combine that with some holder or component from a tissue system, of course, there is not going to be any interchangeability. Like this was never designed to actually interface in that manner. And this is something, as a developer or more an academic developer, something that we run into, of course, a lot, is that if we are developing or designing our own organ-on-chip devices as a component in the laboratory, then it is not very simple, of course, to take that component and integrate it into the platform that is already out there into one of these proprietary closed platforms. And I show here an organ-on-chip device, but we interact, for example, a lot with either groups that work on sensor technology or companies that work on micropump technology, and they really would like to help out and make their products available for users of organ-on-chip systems. But it's not easy for them because they just need to somehow manage to find their way into these closed platforms. And that typically only works by working directly with that company and trying to convince them that you have a key component that will make their platform stronger. So none of that, of course, facilitates actually the participation of new developers, new manufacturers of these platform components. So this is why we actually have a need in this field, particularly if we focus also on innovation and making sure that actually we have quick exponential growth of working off of each other's innovations. We need these open platforms. And I believe very strongly that that will then speed up also development of next generation organ-on-chip systems. And this is also why we focus at the University of Twente, but also within bigger consortia like the National Organ-on-Chip Consortium, HDMT in the Netherlands, on open platform technology. And I'll show you a very, let's say, short movie like that illustrates the problem or challenge that I just gave you, right? Like, so what if we have developed a new organ-on-chip device and we cannot really go and immediately fit them into one of the closed platforms? And then this should be, unfortunately, all too familiar for a lot of developers here in the audience. We end up with systems that sort of look like this, right? Like big spaghetti of tubing, all the bubble trouble that comes along with it. So we really need to try and fix this. And this is why we focus on this open technology platform called TOP. And central to TOP is actually what we are seeing there on that microscope slide. And what we see there, that white rectangle, is the fluidic circuit board. So this is something with the exact footprint and dimensions of a microtite or a microwell plate, but it actually contains all sorts of microfluidic channels and thereby provides also a standardized interface for new organ-on-chip devices. And the interfacing rules for how you can actually click devices into these slots that are highlighted here, those interfacing rules are based on ISO documentation. So this is something that you can just find. You can find what is the inlet diameter, what is the inlet pitch, etc. Like all of that is out there, and you can just design your component so that it becomes automatically compatible with such a fluidic circuit board and with this translational organ-on-chip platform. So we have quite a few of these projects actually going on applying this platform, right? So for example, there are people in the audience here, Anke and Eric, they helped yesterday also with the educational workshop. They're working, for example, on integrating multiple chips from multiple technical universities in the Netherlands, and then also new university medical centers in the Netherlands to all fit into this open platform. But I just wanted to show you one more, let's say, internal example of how we are applying this platform. And that is via this example of this heart-on-chip device that we developed in our lab. So the exact details of this heart-on-chip device, of course, are not like really important in this particular talk, but it's a very, let's say, typical organ-on-chip in the sense that there's an open-top microcompartment, a row of microcompartments containing cardiac tissues. Then there's a porous membrane, and then there's a microchannel running underneath that is used then as, let's say, the vascular compartment. So we've all seen Don's talk yesterday. It's very reminiscent, I would say, of the lung-on-chip layout. This is then, if we look from the top, we see that we can culture endothelial cells in that vascular compartment, and we can culture three-dimensional cardiac tissues in that top compartment with a certain geometry. And then I think this is a more like clear view with bright field microscopy. So we see a beating cardiac tissue there, separated by the vascular or from the vascular channel by this porous membrane. So actually in this project, when working with these devices, we ended up at what you just saw schematically or via artist impression in the movie, right? We ended up with a system, and again, this should be all too familiar for a lot of people in the audience, huge system sitting in the incubator, lots of tubing connections, this whole spaghetti of tubing going on here just to interface with that single chip sitting here in the middle in a Petri dish. And we decided to use this, let's say, organ-on-chip device as a use case for can we integrate this in such a translational organ-on-chip platform? Can we click it in a fluidic circuit board and then use that to actually drive flow through the vascular compartment? So this is the end result of what, let's say, that project looks like. And it's also summarized in that lab on a chip paper that is referenced in the bottom. And I just wanted to just give you a glance of what we designed there. So here we see an example of a fluidic circuit board in pink. So it has that Welsh plate footprint. It has the interfacing rules that are laid out in the ISO documentation. And we have multiple components interfacing with each other via this fluidic circuit board, both at the top, and you can hardly see that, but actually the hard-on-chip device is sitting at the bottom. And if you would like to see this, then I think at least this particular fluidic circuit board is still somewhere in probably Eric's hotel room right now. So you'd have to go and try and find him, but he can show it to you. So the components at the top, they are not things that we developed in-house. They are commercially available components that we just interfaced with each other via these standardized interfacing rules. So they are solenoid valves for switching the flow from one route to another. They are a commercially available flow sensor. They are commercially available glass reservoirs, etc. None of these vendors, of course, knew that we were going to integrate this in the system, but that was also not necessary. We just knew that these components actually fit the right standards that we wanted to interface with each other via this platform. So that brings me basically to this wrap-up slide and saying like, okay, and the top is that open platform. And I want to clarify one thing there is that whenever I talk about open platform technology, people always tend to, the first thing to go to in their mind is to say, oh, so that's sort of like open source. So probably everything has to be free. So it's going to be more, let's say, only for the academic community or so. But I want to stress that an open platform means that it opens and welcomes participation, right? So this is what we do with this fluidic circuit board. We as University of Twente, we innovate this fluidic circuit board and all the design rules are based and out on freely available ISO documentation. But if you come in as a industrial partner and you have a beautiful building block, a flow sensor or a micro pump that you want to have interfaced with this platform, then it's not necessary for you to also subscribe to this whole open philosophy. You can keep your building block closed as long as you follow the right interfacing rules. It will just connect and click into this platform seamlessly. So this is something I think is important to keep in mind. Then I don't know if our timer is, but I wanted to like maybe in the last 2 minutes show a bit also that we are basically continuously developing this platform as well, right? So what I just showed you, that example with hard-on-chip, is actually a relatively straightforward fluidic circuit board, and like with just some micro channels connecting each other, but it serves as a nice example. But we are actually focused on making this platform extremely powerful. And one way to do that is by promoting automation and multiplexing. And we do that by making a use of what is known in the field of microfluidics as Quake-style pneumatic valves. And so it's developed by the lab of Stephen Quake. He was the innovator or the inventor and pioneer of this element. And it's a very simple element, right? It's just a way of automatically opening and closing microfluidic channels as depicted there. So an open state, and then you pressurize a green channel, and then a closed state of the flow path. And we are using this to make multiplex devices with, in this case, 64 individually addressable microfluidic chambers for cell culture. And then we put these types of chips on these fluidic circuit boards that I just showed you. And inside the fluidic circuit board, you make use of these same automated pneumatic valves. And then this is basically the end result of something like that. So we have here 3 of these multiplex chips. So in total, 192 individual microfluidic cell culture chambers. And as you see on the far right-hand side, there is the pneumatic tubing coming in that controls all the flow to each of these individual flow chambers, and also decides which of these chips are being addressed at that particular moment in time. And as we see now with the final chip, we actually have that down to the resolution of a single microfluidic flow chamber there. Like, so every culture chamber can be addressed individually here. And this way of automating the cell culture can actually be quite powerful. So we, for example, do stem cell culture in there, stem cell differentiation. On some of these protocols take multiple days, up to 15 days in this case, where we see that in the end, we got human pluripotent stem cells to express NKX 2.5, a cardiac marker, and actually contract in these systems. And none of this actually needed any intervention of the researcher. This is all handled by the automated system. So it really goes to show that this is going to be a very powerful platform also for you. So in the end, I want to thank a long list of people at the University of Twente who are collaborating to make this platform a success. Then also within HDMT in the Netherlands, we are working very hard and now actually trying to spin this out a bit also towards the European community. I highlighted the names of the people that are also in this meeting, so go and find them if you want more information also about this platform. And with that, I'd like to thank you for your attention.
Thanks so much, Andries. That was a great example of utilizing existing standards, being simply aware of them and then using them, and then also putting, new science into new standards. We got time for one question, Ivan.
So thank you, Andries. It's a very interesting proposal, albeit as a person who's seen probably 30 different configurations of chips, I can tell you that maybe 3 as the ones that you showed will actually fit into your open platform and 27 will not. But that's beside the point. What I wanted to ask you is, we have a lot to learn from, again, genomics. I hate to bring it up, but that has been the previous wave, at least, that some of us tried to catch, right, unsuccessfully. So, and standardization of that technology was on data. MAQC, Microarray Quality Consortium, was on data, not on platform. There were, everyone was making their chips, just like chips here. Then there were 2 companies, now there's only 1. And no one is talking about standardizing technology. Everybody's talking about standardizing the data. So how would you consider an answer in the MPS world to that?
Yeah, so it's a very good question or comment, I would say. So when it comes to the standardization of data, for me, this is a key component of the effort I talked about in the first place, like the focus group standardization organ on chip. This is definitely an aspect we need to address there. It's not, however, the only aspect that standardization can have an impact. And that's the point I try to make, of course, with the open technology platforms. The way I see this or believe this is that we are actually at a relatively early point in the field of microphysiological systems or organs on chips. So we see devices, but all these devices, I think 10 or 15 years from now, we'll look back and think that was actually very rudimentary basic technology. In the end, actually, all of these systems, if we can integrate components from all the great minds in this audience into single platforms, that's actually where we will get technology, organ on chip systems with levels of functionality that we currently can hardly imagine. So I think really this is, let's say, me being maybe a bit of an optimist there, or maybe just thinking that I need also a job 10 years down the line as a developer. But this is really what I believe is that, just a chip with 2 compartments is probably not the endpoint for the field of MPS.
That's great. Okay, thanks, Andries. Hopefully we'll have some more time at the, is it quick?
2 quick questions. Claudia Gärtner from Chip Shop. So we are a manufacturer of whatever plastic or glass components you need for microfluidics, or be it organ on chip. 2 questions to you. I like the approach. We discussed this 20 years ago also with the microfluidics. First of all, how do the manufacturer come into play? Because when I look at your devices, you still have a lot of components you add. What could be for free if you do it in a manufacturing way. So interfaces and things like this would come into play for free. And the other thing, what I see, it looks a little bit electronic dominated, and you nicely made the reference. But when we look in the microfluidics world, I would always look more on the SPS and microtiter plate and slide world. So this question, first of all, how manufacturer and why does it look so electronic-like?
Yeah, so I think the latter, is maybe just a matter of taste and well, not so much to maybe go into, but you're definitely right when it comes about manufacturing. That's an element that I didn't really stress here, but I did mention, of course, manufacturers as one of the parties that actually comes together when it comes to designing or using platforms. And also in this case, so some of these fluidic circuit boards for prototyping, for example, we make them in-house simply by doing PMMA micromilling and things like this. But at some point, we feel that, okay, this is actually at the point that we are going to use it and widely use it or need it. then we just go to local companies like a company like Micronit and just order, like a couple of boxes of these plates. This is, let's say, I think, again, the point that I'm trying to make here is that this should also be open for manufacturers, right? So if, the competitors of Micronit say, well, I actually can make this a lot better or cheaper, then it should be open for them to actually do so. This is at least, the vision or the philosophy that we will follow.
Thanks, Andres. And of course, your laptops in front of you, they only exist because of that. I mean, every technological device we use is here commercially available because of conformity to standards. So, but Ivan brought up the topic of data, and our next speaker is Martin Golubiecki. And Martin's from the Heidelberg Institute for Theoretical Studies in Germany. And Martin's going to talk about integrating data and metadata. Martin, over to you.
Yeah, thank you. And I think the question just before highlighted, as well as the debate before, highlighted the importance of data. And we also heard in the keynotes about that. So I want to give you a few insights in the data standardization that is needed and that is already to some parts available, at least for some parts of the data. And when we talk about data, it's a manifold of data, actually. I mean, you have all the functional measurement data from the experiments, you have the endpoint measurements also, and then you have the process data from your MPS system. And you then have to collect and finally also to integrate that. And also in the debate, it was highlighted as well as in the keynotes, the connection to the modeling. And here, interestingly, you can make a bridge also via the standardization, because when you then collect your data, then what in the end you might want to do is to simulate models. So you have to set up models. For that, you have to integrate all this data, and the data has to fit. And then when you simulate the data and go back to your experimental results and make refinements and eventually start a process again, that all needs a certain degree of standardization. And the data types that are relevant here for the MPS systems are data and metadata, so data describing the data actually on the biological samples and the sampling. So it all starts actually with the biological material. Then you have the sensor data and the endpoint measurements, so biophysical data, physiological data, toxicology data, chemical and biochemical data also, bioactivities, omics data. I mean, we heard about the connections to the omics world also before, and the imaging data, et cetera. I mean, you have all the different kinds of measurements here. And then you have the process data from the devices, the tissue or cell culture data, microenvironment data, and the like. And when you couple that to the modeling, computational modeling, you have also the modeling and simulation data. And when you use human samples, medical data also comes into play. So you have a whole range of data that needs to be standardized in order to fit to each other. And the challenges here for the data processing and integration also are that you want to integrate across the technological silos. So the data from the different parts and from the different readouts have to fit to each other to be interoperable, to interface with each other. And the devices, I mean, you heard about coupling of devices just before, and the devices also have to to be interoperable and exchange data. And then for the data analysis workflows, they also have to be standardized and harmonized in order to fit to each other. That's another challenge here. There's a need for the data provenance standards. So provenance means that you trace back data to the original source, the original material, but also to the original raw data and pre-processed data. You also have the need for metadata. standards. So metadata are these data that describe actually the data that comes out from your measurements and from your experiments. That includes also the terminology. So which terms you use to describe your system, your readouts, and all that, as well as data quality standards. The data access sometimes needs to be controlled, not only for proprietary issues, but also when you think of human data. I mean, it's highly controlled very often, the access to these data. And that are all these challenges that you have to face here. To put it in one word, what you have to achieve is to make the data fair. Fair means findable, accessible, interoperable, and reusable. That does not mean that it's completely open. It means that it's really findable for everybody who has the right to find it, and accessible to everybody who has the right to access that and the like. And to make the data actually fair, you have platforms. I mean, we developed with partners of the team of Carol Goebel from University of Manchester in the UK. At HITS, we developed the SEEK platform over the last 15 years, actually, in the context of the FAIRDom initiative. And there we have established a platform that is used by many consortia in Europe, but also worldwide and locally used for data management to actually structure data models, SOPs, so standard operating procedures, and other information on the assays. And the structuring is also done in a way that the data that belongs to other data can be found together. So you can structure data from assays that belong together or that were done based on the same material into studies, and several studies can be grouped into investigations. So there's a standard even for that, the ISA standard, investigation study assay standard, that can be used here and that we have implemented in the system. And there you can then find actually the data that you are looking for. So it's also implementing controlled access and all that I mentioned for the challenges in the beginning. For actually making the data fair, you have different tools and different things that you can use actually to make the data fair. So to make it findable, you need to use rich metadata, persistent identifiers for the parts of your data. Persistent means that the identifier does not change over the time. You need index data repositories, et cetera, to make that data findable. To make it accessible, you need standard communication protocols, you need authentication processes when needed, and also the metadata needs to be consistently available. To have the data then interoperable, you need to use standardized vocabularies, you need to use linked metadata, so metadata catalogs that provide also a set of minimal information that you need to describe a certain aspect of your data or of your experiment. And finally, to make the data reusable also, I mean, you want to connect that to other results and you want to integrate that, for example, for the computational modeling, as I mentioned in the beginning. The metadata have to have really multiple attributes, minimum information catalogs of the attributes describe a minimum set of metadata that needs to be there for a certain data type. And also the data provenance, I already mentioned that is really relevant. For actually tackling the fairness of the data, you have a whole forest of standards in the life sciences for describing data, but also for formatting data. And these standards can be grouped in three major categories. So one category is the data format, actually, so to make the data also available for machine access, not only for human access. It needs to be formatted in a standardized way to actually describe in a consistent way the biological context and also what the data is about. You have also terminologies for the semantic descriptions of the data content and of the biological material behind. And finally, you have also minimal information catalogs. I mentioned that before. So guidelines, what kind of metadata is needed for what kind of data. And this forest of standards is really complex. You have hundreds of formats for the data, hundreds of terminologies and hundreds of guidelines, because you have all the different subdomains, all the different technologies that you have to apply in your system. And so it's really a forest that you hardly can look through. So there was one missing, no, okay. To give you a few examples, here I picked one example from the modeling world of such standards, and they are very often driven by the scientific communities as grassroots standards. So they are just developed fit for purpose that is needed to standardize a certain type of data. In this example here, you see the modeling systems biology markup language, SBML. And this can be used to have a standardized description of computational models of biological systems. I don't want to go into detail here, but this is a standard that was actually, as a grassroots standard, developed from the scientific community in order to exchange computational models on biological systems. And this can be used for different kinds of computational models. And another example that I want to show you is an approach to actually standardize the visual display, so the visualization of biological pathways and biological models. It's the systems biology graphical notation, SBGN, and there you can have different levels of detail to describe actually your model, a very detailed process description map, a less detailed, more information flow model for the entity relations, so which entities in the model connect logically, and then a very bird's eye view display in a standardized way of the information flow, the activity flow. And that can be compared to the electrical engineering field, where engineers have developed standardized ways of displaying electric circuit diagrams, and that can be also used quite interestingly in the life science field here to standardize the visualization of biological models. And a third and last example from the community standards is the synthetic biology open language, ESBO, that is used to actually connect lab automation systems, laboratory information management systems, and high throughput systems to repositories and databases, and also to connect the data of these systems and the repositories to the modeling world. You also can exchange their sequence information and corresponding information in a standardized way. And so you have this whole forest of the standards coming from the scientific world, defined as grassroots standards, and how to look through all these standards. You need some kind of guidance here, and that's why we established more than 10 years ago the Combined Network, Computational Modeling in Biology Network, that comprises the most used standards in the field and harmonizes them to become more interoperable, that comprises standards for knowledge representation like SBOL, what I showed you, standards for visual representation like SBGN, but also standards for the models themselves like SBML, and corresponding activities like controlled vocabularies and other infrastructures. Then you find some resources in the web to actually help you looking through the forest of standards, like a fair sharing portal, and there you can find collections of domain-specific standards, like the Combine collection for the modeling standards. You can find also the implementation of the standards in databases and tools and repositories, like here shown in the how this works. Okay, like you find here in the orange dots in this graph, and then you find information on the standards themselves and how they are implemented, and in the tools, and also related standards. Another world actually of the standardization is a more formal world of the standardization. We heard from the Senelec group, from Andres just before. Internationally, we have ISO, the International Standardization Organization, and especially here for the field, the ISO Technical Committee 276 Biotechnology, that defines standardization in the field of biotechnology processes and includes terms and definitions, biobanks and bioresources, analytical methods, bioprocessing, but also data processing, annotation, analysis, validation, comparability, and integration. And that is also reflected by the composition of this committee with 5 working groups that deal with these different aspects, as I mentioned, and I lead the working group 5, data processing and integration of this ISO technical committee. And in this working group, we define standards for traceable, searchable, and interoperable data for biotechnology and the life sciences. And this is quite a parallel effort that fits really well with the FAIR guiding principles that I introduced to you before. And the focus here in the working group of this ISO committee is the definition of data and model formats and their interfaces, but also the definition of metadata and the relations of data and models, and also quality management of processed data and models. And we have about 150 committee members currently in this working group. I mean, the whole ISO committee has much more attendees there. And in my working group, we have 23 countries coming together. So it's really an international effort that tries to standardize these data. One example that is quite relevant, I think, for the MPS world is the ISO 26691 draft, that a standard that is coming out this year as a published standard, ISO standard, and it defines requirements for data formatting and description in the life sciences. It can be considered as a meta standard for data formatting, description, reporting, integration, and sharing. So as a catalog of criteria and requirements for those formats, for data formats, and also the semantic data description. And this as a prerequisite, for a framework of interoperable standards, when you think of the forest of standards that you connect the trees in the forest. And one example why this is really so important is an analog from the world of fire. There was a great fire in the city of Baltimore here in the US in 1904, and the whole city burned down there. The problem was at that time you had 600 different variations of hose couplings for the fire hydrants. And that means that the fire brigades of one district in town could not help to fight the fire in another district, not to speak about fighters that came from far away from other cities. So the whole city could not be saved because the firefighters could not connect their hoses to the fire hydrants. I think that is quite a good analog to our world here, because we have all these different kind of data that have to come together here. And you can see this data connections, the interoperability as the hoses that need to be coupled to the hydrants, to the platforms that you use, to the experimental platforms, the MPS platforms. And also the different readouts that you have, they also have to fit to each other. And this is what we try to structure in this ISO draft that will become a published standard this year. It's a reference framework standard for community data standards, and really defines the requirements and rules for the concerted application of such community standards for the formatting, but also for the description and documentation of data types, of different types of data actually in the life sciences. It also defines a catalog of criteria and requirements for such interoperable life science data formats. And I don't want to go through all, for the sake of the time, through all the specifics of this. I mean, you can read the standard once it's out. And I guess you will find a lot of important information there. The table of content shows you that it's not only defining the requirements for the technical and organizational recommendations for the data formats and the semantic recommendations, but also refers to examples of common formats for life sciences, life science data, sorry, and for the minimum reporting standards for data models and metadata. And as an example, you should not read that now, but just that you get an impression how such a standard looks like. I picked an example of a minimum consensus information for the annotation of biological data. So this chapter describes how minimum consensus information should be used for annotation of biological data. And also it defines, oops, sorry, can I go back? Okay. Yeah. Okay. Anyway, you can read that. And in the annex, as I mentioned, you have already 250 or more standards referenced here, formats like the SBML format that I mentioned in the beginning, or minimum information standards and terminologies. And these are, so the printed standard is actually complemented by a kind of live annex available online in the fair sharing platform that I mentioned before. And there you have the ISO 26691 collection of standards with all the 250 and more standards that are referenced there. And the advantage of such a live annex, an online platform, is that it can be constantly updated and maintained. I mean, when you think of all these community standards, they change over time. And so this cannot be really well reflected in a printed standard. And such a live annex platform can help to actually be updated in the future. I think I skipped the last example that I wanted to show you for the data provenance. It's a quite early stage development in the ISO working group that describes the history of biological samples and the data history. You are welcome to also contribute to that. I think it's quite relevant for the MPS technology too. So what I wanted to show you is actually that formal and community standards both are relevant and important for describing data in the MPS world, but also in the whole world of biological data and biotechnological data. Thank you.
Thanks so much, Martin. If we might have some time at the end, is it a very quick one because we're running? Is it?
Maybe.
Go on, go ahead.
Anyway, Mary Jane Cunningham from LifeNet Health. I was wondering in your standards if you dealt with the issue of numerical data versus non-numerical data and how to bridge those.
Yeah, sure. I mean, there are, I was running through the examples, of course, especially in the ISO 26291. We have also, some rules and requirements for numerical data and for non-numerical data, like metadata on the biological topics of your experimental setup and of your system. We can maybe have a one-to-one discussion on that, but definitely numerical data is an important aspect that is also covered there, but also in other standards that we develop. I mean, these were just a few examples of the standards. That's not really the whole world of standards that is out there already and fit for use in the MPS world.
Okay, thanks, Martin. Okay, we'll move to our last speaker. It's Nicolas Verplanck from Grenoble, from the University of Grenoble and also the CA Leti. Nicolas is going to talk about guidelines for microfluidics, how to simplify our lives. I like the sound of that.
I will try. Okay. Good afternoon, everyone, and thank you for the invitation, and especially for the GRC for this specific invitation. So I would like to talk about guidelines for microfluidics and why guidelines are not standards, because most of the guidelines that we will produce in the coming years won't become standards because this is a long process. This is not mandatory to have standards for every guidelines that we will produce. Who will produce these guidelines? We have created 2 years ago Microfluidics Association. This is a non-profit organization which has been created following a European project, but this is an international association which is currently chaired by the NIST in the US, the National Institute for Standards and Tests, and which regroups the main key stakeholders in microfluidics, such as manufacturers like microfluidic chip shops or Microneat or Frigent, for example. Sorry. So I'm a board member of this Microfluidics Association and also the convener of the ISO working group and CN working group in charge of the standardization of the microfluidic devices. So how do we split the work between the Microfluidics Association and the ISO working group? Microfluidic Association works as a pre-normative group. So the idea is to build a large community of microfluidics I would say partners to develop the guidelines that we need in the coming years for the microfluidics domain. This is not focused on the MPS domain, but this is widely used for all the microfluidics development. Sorry. And once we have these guidelines, we can then transfer it, if needed, as a standard. So the Microfluidics Association is involved National Metrology Institute, and we have also a relationship with the FDA to build the next needs of standards. Okay. It has been identified by Monica that the ISO 10991 is in the scope of the organ-on-chips standard, and this standard is part of our group in ISO. So there is already an existing microfluidics definition, which has been produced in 2009 in the previous version of the standard, and there is currently a new version of this definition, which is under revision. As introduced by Monica, there is the PSIS workshop, which has been held last year about the standardization of organ-on-chip. And during the microfluidic session, we have tried to build a roadmap, which is interconnected between the roadmap from the Microfluidics Association and the roadmap of the organ on chips. And what are the main topics identified during this session? Generic protocols for production and material characterization are essential. Testing and calibrations methods need to be standardized. Interfacing and various flavors of hybrid integration. Modularity is one of the promising aspects of microfluidics, and performance characterization standards for flow parameters and fluid leakage can facilitate interoperability. You can find all these details in the PSIS report that has been published by Monica and its team. So if you look on the chart on the left, you have the main topics placed on the chart depending on the importance and the feasibility. And I will address 3 points today. The first one, the terminology standards. We have to know what we are talking about before working on it. The second part, modularity and hybrid integration. That means interoperability between chips and the system. And the third part regroups a lot of things around the protocols and the catalogs of components. I will skip the materials thing because this is a challenging topic to find a consensus about the material to use in microfluidics, PDMS or not PDMS, etc. And as you can see, this is not the most feasible one, but maybe the most important one. We will see in the coming MPS World Summit if we have more advance in this topic. So first, the terminology standards. As described in the previous, in the first slide, there is a 10991 microfluidic definition. So this is a full standard with regrouping a lot of definitions with all the components in microfluidic, all the functions that you can have. We are currently working on the final revision. So this is a DIS version draft international standard, which is under ballot. That means that the countries are currently voting for this standard or not. And then we will publish the final standard by the end of this year. This will regroup a few categories: general terms relevant to microfluidics, general terms in microfluidics, flow-related terms, interfacing, and modularity. All these definitions could be used in the future work. About modularity and hybrid integration. So with the first standard really dedicated to microfluidics is 22916, which has been published earlier this year in January, which is related to interoperability requirements for dimensions, connections, and initial device classification. So this is a work started a few years ago through a European project, MF Manufacturing, and then published first in an international workshop agreement hosted by the BSI in London in 2016, and then pushed into an official standard published early this year. So the main part of this standard is to define the chip dimensions and the pitch position, and also application classes of the different components. I will just give you a brief overview of this standard. You can, of course, download this one and use it for your specific device. First of all, we have to define. Okay, yes. This is one of the drawing of the standard. You have the X and Y axis. No matter if this is top or side connection, you have to define a reference point, and then with the X and Y axis, you can have different inlet and outlets with a specific distance between the first port and the reference point, and also a pitch which is a multiple of 1.5 millimeter. We prefer 3 millimeter for microfluidic components in polymer to avoid any leakage, but this is on your preference. Why did we choose a 1.5 millimeter pitch? This is because with this multiple, you can have the 3 millimeter pitch, the 4.5 millimeter pitch, which is compatible with the mini lure, and the 9 millimeter pitch, which is compatible with the micro titer plate. So the idea is to have a broader compatibility with all the formats that we can find in microfluidics. It's more or less like the USB connector. You have different versions, but with the same objective at the end. We also have defined, so officially, microfluidics has official standardized format. The first one, of course, this is a microplate format because this is the main use format in microfluidics, especially in MPS, as we can see on the different booths. Also, the microscope slide is one of the format. I have indicated here compatibility with microplate format and with microscope slide because there are already existing standards for this specific format, but we have to adapt it to microfluidics. We have also defined an intermediate format for the credit card format if we want to use a size in between these two specific format, and also the microfluidic building block, which has been introduced by Andres a few minutes ago. The building block is a multiple of 15 millimeter for the sides, and this is a multiple, so you can have different versions. I illustrate here the interoperability that you can have with the thanks to the connectors. Because if you want to have interoperability with the system, you have to find the good connectors for that. So I illustrate with a few catalog components that you can find. For example, the micronit and the dolomite chip connectors, have a 3 millimeter pitch. They have been designed in compatibility with the standards, and especially for the micronit one, because it was not compatible initially with this pitch of 3 millimeter. So you can use the one that you want to connect your chip. And the third one, this is the row of 4 connectors provided by Microfluidic Chip Shop, and this is with a 4.5 millimeter pitch, so compatible with the mini lure chips. Also introduced by Andres, the Fluidic circuit board. So if you want to have a modular and hybrid integration of your system, you can develop a Fluidic circuit board with more or less three dimensions to place your different building blocks on it. We also provide clamping solutions. This one has been designed by Micronit, but as you have seen in the Andres presentation, there is also different alternatives. And I illustrate it with this top platform presented earlier. This top platform has been the first example published after the MF manufacturing project, because Twente was involved in this project. I used this standard to demonstrate the ability of the standards, of the guidelines, because it was not a standard at this period, including the microfluidic building block. Oops, sorry, this one. The microfluidic building blocks, which are clamped to the FCB here with a microplate format. You can see the different clamping solutions, and you can interchange all the building blocks as you wish. Other companies are introducing some modularity and some standards, and I have just an illustration of one of the company which are the booth today. This is a Netri with its Neobento solution. They were not involved in the MF manufacturing project. They just developed their own standard, but which fits with the 22916 standard. So the main advantage is this is a 96 well microplate format. There is a modular approach to experimentation, so you can easily change the different building blocks on the microplate. You can test it on the booth. And this is compatible with high throughput screening and with confocal imaging. So we can see that different companies are today developing the modules compatible with the future standard, with the existing standard, sorry. So you can also publish if you are fully compliant, that you are ISO 22916 compliant. So the next part, which is ongoing work, because to work on this testing protocol, we first had to work on interoperability. Now we are able to connect with a good reliability the chips. We can work on the testing protocols. And the first part, which has been discussed with the FDA, this is the leakage testing. This is maybe the main point, the main concern for them. They want to protect the operator or to protect the patient from leakage. So the successful development and widespread use of microfluidic technologies are greatly dependent on the community's success in establishing widely supported test protocols. And one such area that requires consensus guidelines is leakage testing. This is an extract of a review that has been sent under peer review yesterday, which has been built with the FDA and the Microfluidics Association and should be available quite soon. We have also published today on ResearchGate a white paper about the protocols for leakage testing. So this is more review about the leakage testing, and now we are working on the future protocols to be standardized. And to do that, I introduced in the beginning that we work with National Metrology Institute. This is very important to have them on board because we are able to provide a roadmap to explain what we want in the coming years, but we are not able to develop very precise protocols. That's why we have been funded with Euramet, the European Association of Metrology, and Empire participating project, the project establishing metrology standards in microfluidic devices. So this project aims to contribute to the development of globally accepted standards for microfluidics. So we try to develop measurement protocols and guidelines and the dissemination of metrology standards through the different normative committees, especially the one of microfluidic devices. So you can see that there is interconnection between all the main stakeholders, the FDA, so the regulatory, the Metrology Institute, the Microfluidic Association, and the Normative Association. And another ongoing work, this is AWI, so that means approved work item. So it has been voted last year. This is a work item about microfluidic pumps, the symbols and performance communication. The idea is to, through this first example, so it won't be a standard, it would be a technical specification, but the idea is to do a first tentative to build microfluidic datasheets. Because we, as end user, we want to be able to buy a microfluidic component with the associated datasheet. And for that, we try to do this exercise with the microfluidic pumps. So as a conclusion, microfluidic community is addressing the main standardization topics for OOCs. You can join this microfluidic association if you want to contribute to this roadmap and to help to the white papers and different contributions. And standards are a tool to improve quality, to ease integration, and to accelerate innovation. Sometimes we think that if we have to follow standards in research, it will block the innovation, but it's not the main idea. The main idea is to provide guidelines to save time, to have reliable components and connections. And my last slide, I would like to acknowledge, of course, MFA board members and other members, and also the ISO working group experts, which is an international group. And also to mention that Grenoble is hosting the Eurox yearly conference, the 4th and the 5th of July. So you can join us if you want to discover the French Alps. Thank you very much.
Terrific. Well, I'm afraid we've come to the end of our time, allocated time, so I don't think we'll be able to have a question, but I'm sure you'll be available to meet people outside. Well, that was wonderful. I hope you enjoyed, and we hope we managed to convince you that not only standards will help translation of innovation, but will actually drive innovation. And maybe from this day forth, it will be microphysiological standards. That'll be the new definition of MPS. So thank you all for joining. And the next session actually is dealing with also issues around standards and reliability. So thank you.
Alistair's a first name or a second? First name. Stewart is second name. Okay. Exactly. I respond to either. You just have here the. That's the pointer. Yeah. That's the pointer exactly. And then this is play. This moves forward, backwards. This is forward. The big one. Big one. Yeah. And the small one is backwards. It's off. This is off. Exactly. Okay. And you have your slides there. And you have the timing. Yep. Yeah. Alistair. Hi. What did you hi? I'm trying not to do that again. So you basically have your third. And it's a bit up to you how much of that time. So I think you have that's you have it there.
I was like, dang, you got there early. I was thinking, you're right.
So
Good afternoon, everyone. We will have to start with the next session. So the next session on the program is session S6, reproducibility and robustness of the hardware and cellular models, standardization and harmonization best practices. My name is Olivier Frey. I'm from Insfero, and I have the honor to moderate this session here. The first speaker is Joachim Wiest from Salasis in Germany, and his talk, the title of the talk is Systems Engineering for Microphysiometry. We will have 5 talks during this session. We will have Q&As after each talk, but we will also have a 10 minutes Q&A at the end. If there are more questions, we can use this time as well.
Thanks for the introduction. Welcome everybody to my talk entitled Systems Engineering of Microphysiometry. I like to talk about some engineering aspects of microphysiological systems and give some examples how we approach them. But first, some facts about Salasis. We are a southern Germany-based SME. We were founded already 15 years ago as a spin-off from Technical University of Munich. And we are equipped with laboratories for electronics, electrochemistry, a machine shop, cell culture laboratory. And quite a few years ago, we adapted some ISO standards, 9001 and 13485 for the engineering part, and also GLP for the biological part. We have a long-standing cooperation with the German Animal Welfare Academy, and we are provider of instrumentation and services for microphysiometry. So what is microphysiometry? That goes back to a paper from Science from 1992. So what you can see here, that's a cellular model. And what they did back then, they placed a pH sensor close to that cellular model. And those they were able to measure the vitality of this cellular model in a label-free manner. And that allows toxicodynamic measurements. We extended that a little bit. We are not only looking at cellular we are not only looking at extracellular acidification, but we are also looking at cellular respiration and at cellular morphology and transepithelial impedance measurement. So to do that, for the extracellular acidification, we implemented a pH sensor. For the cellular respiration, we implemented a miniaturized dissolved oxygen sensor. And for the morphology and transepithelial impedance, we implemented impedance sensors. So that's our biochip. It's made on a glass substrate, so it's optically accessible, and it can be sterilized by ethanol. So that is the core of our talk. So systems engineering, that's a systems engineering approach on that systems that we have here. So it's always in the center. So we have a cellular model. We have hundreds of cellular models which are available, and they are always interfaced by a biochip for readout, which is connected to optics or electronics. And then very important, everything is placed in a climate control chamber with a more or less sophisticated fluidic system and with actuators. Usually that is controlled by computer, And what we indicated here, that you want to have not only one channel, but maybe you have more channels for your system. And that is how our approach looks like. So here we have 6 chambers with 6 biochips where we can put the cellular models on. On the back side, we have a pump, and here we have different fluidic modules where we can, before the start of the experiment, place the cell culture media and different flasks, the flask with cell culture media and the drug under investigation, and for example, media for the control. Everything mounted in the incubator, and here we have the control unit right beside it. So to have a look at the historical development of the cellular models, here we have that great comic from Plants et al., how it developed from the end of the 19th century. So first we had cellular monolayers, then they became more complex. We went into 3D models, we went to tissue inserts, then we interconnected them, and on that development way, the fluidic system changed from a static fluidic system to a dynamic fluidic system, and became more and more complex. And I like to show a few examples from us how we develop systems according that value chain. So that's an example where we used cells in suspension. I do not want to go too much into detail here. The main point here is that we can use cells with a cellular model which has cells which are available in suspension. So here we build a connector to our biochip where we just have a cellulose membrane. So say we could fix algae. So in that case, we used algae to make an example for water quality testing. And what you can see here is the photosynthetic activity of those cells, meaning the oxygen production of the algae when we put on a light source. And first we have a nice oxygen production, but as soon as we put metamethron to the cells, which is a herbicide, so photosynthetic activity goes down, and if we remove it again, it goes up again. What is also nice here, that since we are using label-free sensors, we can monitor the dynamic changes. We can add a drug or a compound, and we can remove it, and we directly see the effect of that. So the next step in complexity would be to monitor a 2D monolayer. From an engineering aspect, that's quite simple because those cells just adhere to our biochip. So we just seed it to the biochip and connect it to the fluidic system. So here we used a simple L929 fibroblast mouse fibroblast cell line, and we added 9 concentrations of a drug. And here again, here we see the extracellular acidification rate, meaning the pH change, which is caused by the cells. And when we add the concentrations, we can determine, for example, an MRD50 value. So when the metabolic activity of the cells is reduced by 50%, and that might be an important parameter for that SDS. Next step here is to go 3D. So we grew 3-dimensional spheroids from G2 cells in our lab. We grow those. We had to invest a little bit more engineering capacity to the encapsulation on the chip. So one spheroid would not be enough to make a high enough signal to monitor it with our sensors. So we made a plate here where we could integrate 9 spheroids, and then we have a nice and a high enough signal. And what we can show here, it's just a proof of principle. We see here the acidification rate of those spheroids. So here that's the raw data, and that's the rate. And if we add a toxic compound, the value goes down again in a label-free manner. So we were able to show that we can also use spheroids here. The next step then was to use tissue inserts. So we partnered up with Martech, and we used their epi-intestinal model, and again modified our chip. So now a commercial available three-dimensional tissue insert model can go in here. And what we did here, we changed the fluidic system a little bit. So we have a basolateral fluidic and an apical fluidic, and we also could create an automated air-liquid interface. So we can exchange the media below the tissue insert, then we can add media on the apical side, but we also can remove it and leave it to the air-liquid interface. And only if we add media on top again, we can, for example, make a tear measurement across the barrier of that function. And I have to say, that's all in the incubator and everything is automated. So nobody has to open the incubator to exchange some fluidics or to insert some chopsticks. So the fluidic system is automated and the measurement system is automated. So to sort it in to the development, so as I said, systems engineering, we have the development of the cellular models here, and that's the development, how we went along that to keep up with the developments in the cellular models field. Here are some more details about the fluidic system which we are using. So we are using a commercial available pump, a peristaltic pump, 8-channel pump, which is computer controllable. So very nicely can adjust the flow. So usually we use a periodic flow type, meaning we pump 5 minutes, we stop 5 minutes, we pump 5 minutes, we stop 5 minutes. But of course you can program it as you like. And here we have the fluidic module, and we integrated computer controlled valves here. So you can put 4 flasks in here, pre-fill it with the medium you want to investigate, and then you can transport it to your cellular model with a compound, with a positive control, as ever you like. And I really want to stress that here. So we made a quite simple investigation how the differences between a static fluidic system and a dynamic fluidic system. So most of us will just have their cells in a cell culture incubator and feed them twice a week, which basically is static. But we know that we are human beings, and if we would just eat twice a week, that would be problematic. So we compared it on the system, how the proliferation rate is compared in the static culture and in the dynamic culture, meaning when we exchange the media every 10 minutes. And the proliferation rate is changing drastically. So that's a basic thing which should be considered when we talk about systems engineering. Just the exchange rate of cell culture media will heavily affect the vitality of the cellular model. Coming to a next quite important step when we set up our assays. Here is our so-called Celasys 8 assay. So we set that to 24 hours, so it will nicely fit into a daily work schedule, so to say. But what is really important is that when one sets up an assay, we have to include controls and we have to include replicates. So what we do see here, so as I mentioned before, we have a 6-channel system, so we can make 4 replicates. And here we start with a reference medium for 6 hours, then we add treatment medium for 6 hours, then again reference medium for 4 hours, treatment medium for 4 hours, and then a positive control toxic medium for 4 hours. Everything is automated, and since we are working label-free, we see the effect of the treatment medium, and we see the effect of the recovery. And since I just talked about controls, so here we have a negative control where we do not add the test medium. So we just expect that we have positive signals here all the way until the end. So in the end, when we add our positive control, the signals from the cellular model has to diminish. And if that's not working, something that with our assay is not correct. What we also do, we add a blank. So we do not use cells on that blank just to make sure that the treatment media does not interact with our microsensors. And having that, we have quite a robust assay with all the controls which are needed. And yeah, we can rely on the outcome of those assays. That is an example. So I have two case studies here. A simple example where we just changed from FBS to chemically defined media, so FBS containing media to chemically defined media. The reference medium is standard DIMA medium with 5% FBS. And the test medium is here a mixture of DME with F12, but without FBS. And what we can see here in the metabolism in the acidification rate, if we change from reference medium to treatment medium, there is no effect. So meaning that that medium would really work nice over all that time. But when we have a look at the impedance sensor, we can see that without the FBS, the impedance goes really down, like here in 10 minutes, and stays on a lower level. And when we go back to the FBS, it goes up again. So that's quite aligned with the theory behind it, because all of those proteins in the FBS. But that also shows that it is important to have a look at the metabolism and at the morphology. Which brings me to the topic reproducibility. We are really working with defined media, and we highly recommend to work with chemically defined media, not to use fetal bovine serum, not only because there are ethical issues, but also because there is really a scientific problem in working with fetal bovine serum, because it's not reproducible. There is batch-to-batch variations, and when you really go out of the university lab into the industry and you start to do your assays in different laboratories, and there is different FBS, there will be different results. But there is a community, a growing community, working on alternatives on developing chemically defined media. So here you find a website where you can have a look for chemically defined media. And we also have a poster about that. So you are welcome to visit our poster and to develop strategies how to move away from FBS. That's the second case study, just a quick one. We made our Celasis 8 assay, and we found out that for the CACO2 cell line, DME mixture with a 50 to 50% DME F12 mixture with ITS worked fine in our Celasis 8 assay, so in the 24 hours assay. What we also did, we moved to standard cultivation protocols, which is state of the art, how to move away from FBS to make tests over weeks and months with the newly developed media. That also worked. We saw that the cells need a little bit longer to grow, but they still grew continuously, I think over a year or something like that. And what is also important, at least for the CACO2 cell line, if they stay, or if they still create microvilli, so if they differentiate, and they also did that after 40 days of cultivation in our chemically defined media. So that is just to show, okay, what was predicted by our Celasis 8 assay in 24 hours seems to be valid, and we controlled it in more than a half year of other experiments. So to summarize, the systems engineering approach I recommend. I gave an overview how those microphysiological systems can be categorized, and stated the importance of flow of the cellular model and the temperature, how important it is to have a standardized protocol. And with the automation, we also improve reproducibility because we exclude human errors or errors due to drops in temperature and something like that. I highly recommend to use defined media and to validate the cellular model, meaning to validate cell lines which are used. So that was a problem we had like 20 years ago with the cross-contamination, but we see that especially at universities, the validation of the cell lines is not common practice, so to say. And I want to emphasize that system engineering may contribute to the reproducibility crisis in biomedical sciences. But just wrote that paper, Systems Engineering of Microphysiometry, and I invite you to have a look there because there are some aspects are explained in more detail. And with that, I come to my last slide, and I'm happy to take any questions.
Thank you very much. Are there any questions from the audience? I have maybe more specific questions on the spheroid model that you have shown, that you have the measurement in the raw data. You had these waves of data. Can you maybe explain from the data recording from where they come and how the measurement actually looks like?
Yes, excellent question. Thanks for that. Usually I do not explain that because it's a little bit confusing and complicated and makes my talks too long, but it's a really a central question. So what we do, we run our fluidic system in a stop and go mode, meaning we pump fresh media towards a cellular model for 5 minutes, and then we stop. And then in the next 5 minutes, we can see the acidification rate of set micro volume. We pump there, and then we pump again for 5 minutes. So we have fresh media on the chip. We can use that to recalibrate the sensors. Then we stop again, measure for the next 5 minutes, and so on. And we program it that we have these 10-minute cycles for the first 6 hours, for example. And then in one of the pump cycles, we switch one valve. So we can add then the cell culture media with the drug under investigation. And we call it shark fins. So the highs is the acidification rate or the oxygen consumption rate. Yeah.
Okay, thanks. You mentioned that your system has like 6 replicates in parallel that you are using. Are you also planning to extend this to being able to have more replicates in the same system?
Of course, if you have a look at the graphics, where the graphic is going and what we developed so far. So of course, the next thing is a 24 well plate.
What are the challenges there for the scale up? What are the technical challenges there for the scale up?
I think with our experience, it's just a logical next step. It's not that there are too high hurdles, I would say. Of course, you would use the standard format of a 24 well plate. You would use a pipetting robot. You place everything in a huge incubator. So we have a prototype on that. We are working on that.
And on the fluidic side, I mean, at the moment you use the peristaltic pump. Would it be then a different pumping system if you have much more wells?
Yeah, a pipetting robot.
Okay, okay, yeah. No other questions? Otherwise, I think we can switch speaker. Thanks again for this interesting presentation. So the next presentation will be given by Mizuru Ita from Tsukuba University in Japan. The title of his talk is Identification of Variation Factors for Development of Assays Using MPS.
Yes, thank you so much. A great introduction and invitation. My name is Mizuru Ita. I'm from the University of Tsukuba, Japan. So today I talk about identification of variation factors for the development of assay using MPS. This is today's topic, the 3 topics. And at first, the thought process to establish assay method. And second, the case studies for problem solving. And last, third, utilization of international standards. At first, the thought process to establish assay method. When establishing the cell-based assay, we should consider the several things. This is just one example of about the hepatocyte-based assay. So establishing the cell-based assay toward assay method construction, so that we should consider the several things. So for example, what specification are required for hepatocyte, or what device should be selected for the assay. So we need we should determine if the MPS is appropriate for the our intended purpose or context of use. We reported in the AMED project about this problem. So by the way, the AMED is a Japan agency for medical research and development. So it's a kind of funding agency. And so we aimed at social implementation of MPS as a national project. We create the strategy for establishing assay method using MPS. This is just thought process. So at first, we should consider the intended purpose or context of use. This is the most important point. And the next steps, we should clarify the requirements for MPS. And as we know, the MPS is a complex system. So especially the main two components, it's one needs cells and the other is the devices. So if we want to clarify the requirement of MPS, so we should clarify the requirement of cells and devices. And we do the listing the quality attribute and defining the quality attribute. Finishing these activities, so we can design the assay for optimization and qualification and validation of assay method. Maybe after this process, we can establish the assay method using MPS. This is so in those processes, so we can decompose each phase. So for example, the requirement for cells, so we should consider for example, the specification of organ cells or routine raw materials or availability of standard cells. So in each phase, there are several programs to consider. Okay, the next topic, so the case study for problem solving. In those projects, national projects, we do the multi-center evaluation for assay method optimization. And due to this evaluation, we extracted the variation factors. This is a thought process again. So today I talk about specifically the cell adhesion and the specification of organ cells or protein raw materials. To evaluate the method optimization, with a very simple scheme using a common hepatocyte lot. Drug response studies were conducted at 7 institutes. So for example, the university or research institute participating in the national project. So at first, the cryopreserved hepatocyte was seeded and maturated, and we we treated the drug for 2 days. After treatment, we check, for example, the metabolic enzyme mRNA expression. This is one of the results. So the CYP3 mRNA induction by drug treatment. So this graph is mRNA, normalized mRNA, normalized mRNA. And we are really surprised that this result, because there are many differences, the absolute value of the mRNA expression level. So the low or medium or high. And by the way, this is a gray bar is control, and the red bar is rifampicin treated. So the induction is correctly, but why this trend? So we examined the culture reported from 7 institutes in detail, and we extracted the difference between the induction days and seeding density in process. So where the induction days, 2 days is correct, but 3 institutes is only 1 day, or the seeding density is slightly different. So this difference is correlate to the mRNA expression level. This is a brief summary. So the drug response testing procedure, so that the following factors are seeded cell density or drug treatment time were found to cause variation in the method of measurement. So the problems were found in the creation of procedure manuals. This is just the reflection, and other aspects that we found a point to keep in mind when educating operator. So for example, the seeding time on and so on. After the multi-center evaluation, in order to evaluate these variation factors, we do the evaluation of the impact of seeding density. This is a very similar scheme to the beginning evaluation. So the ability of hepatocyte seeded at 3 different density to induce ship mRNA was analyzed for various hepatocyte cell lots. And seeding density is these 3 concentration. This is one of the results that the CYP3 for mRNA induction. We do the 3 vendors collaborate up to hepatocyte. So these 2 results is slightly similar, and this result is just slightly different. And there is an optimal seeding density. And we know the effect of seeding density on hepatocyte adhesion. This photo is a high concentration seeding, and this is a medium concentration. So cells detach at high seeding density. So this is one of the reasons of variation. There are other point is a difference in culture image from hepatocyte strain to lot. The center of hepatocyte is different pattern of induction. The morphology of this lot differ significantly from other lots. So maybe the conference changes. Next evaluation is evaluation of the impact of induction days. We do the similar theme, and drug treatment days is 1 day, 2 days, and 3 days. This is one of the results that the CYP3 whole mRNA induction to about the 3 different cryopreserved hepatocyte, the same pattern. There is an optimal induction days. In this case, the 3 days is optimal. The brief summary. So the variation factors in the assay process, the number of seeded cells and induction period were found to affect the performance of hepatocytes. It was shown that the consideration of cell adhesion is necessary, and it is necessary to consider the property of frozen hepatocyte, which are the raw material. So we do these considerations toward construction of robust assay system. Okay, so this is the last topic toward utilization of international standards. This is a very interesting review in bioengineering. And in this review, there is the availability of existing standard documents. This is a very, very interesting data. So in this data, there are no standards for assay method using MPS. However, I would like to make the small suggestion. So the example of a standard document that can be utilized. This is one of the ISO standard, the ISO 23033. This document is the general requirements and considerations for the testing and characterization of cellular therapeutic products. This is, yes, this scope is for cellular therapeutic product, but we can use cell for MPS. I explain again about this sort of process, and this standard ISO 23033 can be used in this process, optimize, qualify, and validate assay systems using MPS while accumulating data. Today, I talk about variation factors, extracting variation factors. Example used for variable factor extraction, we can use the ISO 23033. This is a figure in this document, and it's the Ishikawa diagram illustrating common sources of variability. And in this diagram, so for example, the cell source or procedure, this is connected to the variability in test using MPS. So I use this document to extract the variation factors in our hepatocyte-based assay. So last summary, so that even if there is no MPS specific standard, but existing documents such as concept may be utilized. On the other hand, the standards specific to MPS are also necessary, I think. First of all, it is necessary to promote international standardization using, for example, the white paper and so on. This is acknowledgement. Many researchers, student organizations contributed to this project. And this is the last slide, so very important information. So today I'll talk about the variation factors about cell-based assay in MPS. So these post session and roundtable is related to today's my presentation. So please check this presentation. That's all. Thank you very much.
Thank you very much. Are there questions from the audience? I have one question. You tested the different variation factors on the 2D model, or like of a 2D cell hepatocytes. Have you also already investigated more complex systems, like 3D models or co-culture with other cells to understand the variations there?
Yeah, it's a very great question. So now I tried the very simple system, but I understand that we should do the next, as the next step, the more complex system. But I think at first we should identify the variation factor in simple system. And this is a base, this is a base information to create the complex, to extract the variation factor in complex system. So I think it's a step by step.
Okay. Yeah. Where do you expect, if you go to MPS and you have microfluidic setups, etc., where do you expect big challenges or variation factors in the future?
Yes. So today in this summit, every people says about several devices, several materials, several cells. So and the MPS is just a complex system. So most challenging point is a combination. So the very variable, there are very variable combinations. So the exact general variation factor is really challenging, but we should challenge about this.
So I believe there most probably first needs to be kind of a standardization before we can look at the variation. Because as has been discussed during the lunch session, we will have, at the moment, we have a big landscape of a lot of different systems. So it might be difficult to find the common factor to then understand which of these factors is actually creating most of the variability. How do you deal with this large amount of different systems?
Yes, it's a great question. So yeah, I totally agree with your comments. So the one of the most important point is we should same sort of process. And so the dialogue is really important. So we can, if we get the several data to common process. For example, the data management is really important. So data format is really important. So if the data format is a common, so we can discuss, we can do the dialogue. So this is the most important point to achieve the extraction of variation factor in complex system for you, I think.
Any questions? You mentioned white papers. Who would be the ones who write these white papers? So whom do you expect to write white papers on the variation of?
By the way, I talked in this presentation, we prepare the white paper in national project, and I think the reader is, for example, the MPS supplier, because the MPS supplier is slightly confusing how to create the MPS depending on user requirement system. User is, for example, the pharmacopoeia. So the white paper is, I believe that as guideline the white paper is working. So, so at first, we prepare the white paper for MPS developer, but yeah, this white paper is, of course, the other target. So for example, the user or researcher. At first, MPS developer, first target is MPS developer, I think.
Okay, thanks a lot. Not any other questions? Otherwise, let's thank the speaker again.
Thank you so much.
So we are a little bit ahead of time. We have I will keep a 5 minutes break now so that we keep the timing for the online viewers and also that people can switch between presentations from the other track. So we'll continue in 5 minutes. One comment, I'm alone here on the stage. Want to excuse Doreen from Amanda Biosystems, so she couldn't join the meeting and join me as a co-moderator here. break because I wanted to align again with the timing. So the next speaker is going to be Alastair Stewart from the University of Melbourne, Australia. And he will speak about circadian physiology and microphysiological systems.
Thank you, Olivier, and thank you to the organisers for the opportunity to share some data with you. Some of you may be suffering from jet lag, so I think talking about circadian physiology at this particular time in the afternoon is relevant. And so that's what I'll be talking about in microphysiological systems. And forward is that one. Okay, I have a couple of declarations to make in terms of conflicts of interest. So I'm CSO and director of Tianli Biotech, which is developing anti-fibrotic, anti-inflammatory therapeutics, and I'm going to talk about one of those compounds shortly. And I'm co-inventor on the rotary planar multiplex microfluidic pump, more easily stated as the RPM squared. So you have to have motivation to set up circadian rhythms in vitro. It's a rather challenging task. What was our motivation? Well, here's one of them. So aside from intrinsic interest in the impact of circadian rhythm on cells in culture, we identified some time ago that this compound PF670462, which is an inhibitor of casein kinase 1 delta, was able to oppose the TGF-beta-induced EMT in epithelial cells. These are A549 cells, but it does this also in bronchial epithelial cells. It does it in fully differentiated air-liquid interface organotypic cultures of the epithelium. So it's an action which is broadly represented on different phenotypes of epithelium. And this is just the gene expression level being restored by progressive increases in concentration of the agent. It also has a number of other anti-fibrogenic actions, but I don't want to focus too much on those, other than to say that it suppresses the fibrogenesis induced by TGF-beta in fibroblasts from patients with IPF and fibroblasts from healthy non-fibrotic individuals. And we have put the compound into the standard mouse model. As we're here thinking about alternatives to animal models, then there's a extraordinarily important model to develop, and that is a model of lung and airway fibrosis that replaces this Blair-Myson model, because the Blair-Myson model is rather taxing to run emotionally, given the impact of Blair-Myson on the mice. But nevertheless, at the moment, it's necessary to persuade regulators and ethics committees that the compound under development has some prospect of having an anti-fibrotic effect in clinical evaluation. So here it is, compound administered by aerosol to mice, lowering the Bell cell accumulation, reducing the hydroxyproline in the lung, and regulating a couple of the fibrogenic targets, the collagen 1A and alpha smooth muscle actin. And in this respect, it's similar in its extent of effect to that of the existing antifibrotics pirfenidone and nintedanib. So when we're thinking about an inhibitor of casein kinase 1 delta, we have to think about the molecular clock, because this is what this enzyme is best known for. That is the phosphorylation of, beg your pardon, phosphorylation of the period protein, and its phosphorylation eventually causes its degradation. And this is a negative translation transcription feedback loop whereby the period protein will oscillate. And the PF670462 compound is able to inhibit this oscillation by interfering with this negative feedback loop. And so some of the actions of this compound may well be secondary to its ability to change the circadian period and amplitude. However, some time ago it was identified that casein kinase also co-precipitates with the activated TGF-beta receptor. And our contribution in this area has been to identify that it's CK1 delta in particular, which is responsible for the fibrogenic signaling associated with the activated TGF-beta receptor. So this is an action that can be referred to as a non-canonical kinase signaling emerging from the activated ALK5. And you'll probably be familiar with the SMADs as a very important effector of the TGF-beta action, but there are a number of kinases that also add to TGF-beta signaling. It's a very complex signaling molecule with a very broad range of actions as a morphogen and a fibrogen. So it shouldn't surprise us that it has this complexity in its signaling. Well, the quandary that we were posed with by our findings was, were the actions of the compound secondary to clock regulation, or were they related to this TGF-beta action? I might give you the conclusion first. Because we're able to demonstrate anti-fibrogenic effects in cells in culture that had been not recently subject to a medium change and therefore had no intrinsic rhythm, we think that much of the effect of the compound is actually due to modification of the TGF-beta signaling. But it stimulated this effort to try to establish circadian rhythm in culture. And this effort has been driven by Brian Gower, who couldn't be at the meeting, but what he's done essentially is to develop a multiplexed perfusion system or superfusion system to enable us to entrain circadian rhythm in vitro. Here were our first attempts at that, and this was in a microfluidic setting with periodic exposure to hydrocortisone. You can see that there's a break in the trace there. This is the bioluminescence that is produced by the period 2 fusion protein with luciferase. So when the period protein rises in concentration during the circadian period, the output of the bioluminescence rises, and as it's degraded, so too is the luciferase, and the bioluminescence therefore falls. And what you can appreciate here is the resting rhythm decays over about 3 days. And this is typical of a single pulse of entrainment. We see that the rhythm dampens. And so when we're looking in culture, if there's been a stimulus such as exposure to cortisol, then this will have a only short-lived effect. So if we're interested in a medium-term output, like the activation of fibrogenic processes in the cells in culture, then the potential influence of the circadian rhythm on those will be lost by this transience. What you can also appreciate here is that the addition of TGF-beta has increased the amplitude, and as we expected, the compound PF670462 completely ablated the rhythm in this system. So, is rhythm important in terms of the mechanics? And the answer to that question is yes. So here we're looking at aged and young breast tissue slices, and again looking at the output of PER2-LUC. And you can see here that there's some decaying rhythm in the young mice, the 3-month-old mice, but in the aged mice, there's a very flat line. And this, in this study, was correlated with the development of fibrosis in the aged mammary gland, which was shown to dampen the circadian amplitude. Interestingly enough, it shows the opposite trend in lung fibroblasts. So in this study, the seeding of the cells on the, raise this so I don't hit our chair. The seeding of the cells on the stiff plastic gives a good rhythm, albeit one that decays quite rapidly over 3 to 4 days. And the seeding of the cells on a 3D, on a soft matrix, which allows the cells to aggregate with each other and form a small spheroid. In this setting, the circadian amplitude is markedly reduced. And just to draw your attention to these tissues that have been taken from the PER2-LUC mice, showing how rapidly they decay. This is the suprachiasmatic nucleus, liver slices and lung slices, and they're all decaying over a period of about 8 or 9 days to a very low amplitude. So these findings all consistently point to a loss of rhythm in cell culture without some external agency. And of course, I'm not going to linger too long on this because we do appreciate that circadian rhythm is a pervasive influence. So I think that provides us with a really strong impetus to be considering in training the cells that we use in microphysiological systems and in standard cell culture experiments, such as the ones I'm going to show you. So this multiplexing that is enabled by the pump that I'm about to describe is not only used for circadian rhythm entrainment for what we hope will be superior emulation of physiology, but it's also enabling us to change the media in a constant manner and in a dynamic manner to dynamically compose the cell culture media so that the cell culture media can reflect what happens in the interstitial fluid, which is in a disequilibrium, if you like, with the plasma contents, and therefore is a dynamic entity, unlike the usual setting with a static culture system, such as the previous speakers have been referring to. So this pump enables us to change the medium in that manner, and also enables endocrine emulation other than the exposure to cortisol, and of course, very importantly, pharmacokinetic emulation. So here is the pump. It's a stackable device with multiple ports at each stack, and this stacking means that it's scalable, modular. It's intended to be affordable, and it's intended to be user-friendly. It has precision, it's compact, around about 7 by 7 by 7 centimeters in size, durable and reusable. So it enables us to perfuse multi-well cell culture plates for drug testing, and is scalable up to about 100 lines and can operate continuously for weeks at a time. And because we're using open plate cultures, there's no issue about bubble formation, which is an impediment to long-term microfluidic perfusion systems, or at least to many of them. Okay. So here's what the internal aspect looks like. So the motor and the heat sink are on the outside of the incubator. This is the inside of the incubator, where you can see the stackable unit, and you can see the fluidic lines. The media is kept in the refrigerator. This is actually quite important because if we're doing a long-term experiment, there can be significant degradation of some of the components. And so we want the exposure over time to be consistent, so we keep the media at 4 degrees, and it heats as it's traveling through these lines before it reaches the cell culture plate that you can see here. And this diagram has the effluent collection occurring within the incubator, but of course the effluent collection can take place outside of the incubator. And I should point out that we're working with a company in Melbourne, Trajan Scientific and Medical, and with them we're looking at modularizing all the fluidics. One of the elements of this system that poses some challenge is the connection of all these lines, and what we're working towards is a single sterilizable, consumable unit that will then make the ease of operation of this system very much greater and more attractive for novice users or those who are not used to fluidics systems. And so the conclusion that we have at this stage is that rhythm does matter. And here is what the plates look like. And this is the emission of photons from the luciferin. It's been converted by luciferase. And the imaging is made by a high sensitivity camera, which is mounted at the top of the incubator. The incubator is modified to have no extraneous light, so that the sensitivity of the system is optimized. And we're looking at here on the left-hand side, a static system, and on the right-hand side, the flowing system, and the summary images of these cells that have been exposed to the entrainment stimulus under static conditions and flow are shown here. And you can see in the static system that on day one, there's a strong signal, and that this shows oscillation. So here we've got the circadian time running on the x-axis, and we're looking at days on the y-axis. And you can see that by day 3, the rhythm is essentially gone after one pulse of the cortisol. Whereas when we've got the daily pulses of cortisol, and I don't think I can get the pointer to the particular point, but on this graph here, you should be able to see a green spike, which is a 90-minute exposure to 500 nanomolar of cortisol, which is able to stimulate the oscillations in the period protein. And we can see here that the circadian period is very well maintained over the 4 days, and we've gone out to 7 days with these kinds of experiments to show that it's maintained over that longer period. So I want to turn your attention to the medium. We've had some discussion in this session about the quality of the medium, the composition of the medium, and I think it'll be obvious to all of us that there are a number of features of the medium that change over time. But one that I want to draw your attention to is the conventional culture medium is designed to last for a number of days without the need for change. And that design feature builds in a hypernutritional status. And so some of the amino acid concentrations, for example, are 20 times what they are in our plasma. And so this does represent a metabolically aberrant state for the cells to be exposed to. And this is something that I think many of us doing cell culture have ignored for a very long time. And perhaps one of the reasons why it has been ignored is there hasn't been a very practical solution. So what I want to suggest to you is that the multiplexed pump is a practical solution to address this issue of using hypernutritional medium. And so here it is in stylized form. So here's our commercial medium, DMEM, but it doesn't matter what you substitute here. All these commercial media, other than the human plasma-like medium, HPLM, contain a hypernutritional content of not only the amino acids, but a number of other factors. And often glucose is presented at a diabetic level to the cells. And so what we expect to see happening to this medium over time is that it declines in concentrations of the various components, but actually remains hyperphysiological for the whole of the duration of the contact with the cells. Now, if we were to use a plasma-like medium, as we do, and we've devised a formula called the Melbourne medium, but there is a commercially available version called human plasma-like media available from Thermo Fisher. But if we were to use that in a static culture, then the levels would decline to a hypophysiological level within a relatively short period of time. And that's just as big an issue as the hyperphysiological hypernutrition that you can see with all the commercial media. And so what is the solution to this? Well, the solution is to use the fluidic system to pump the media through the cells. Now, you may not be used to looking at this style of diagram, but and you're not intended to be able to read all the components. In fact, it's a secret. But what I want to say to you is that these are the known components of the media, and these other ones on the other side are the metabolites that appear in the media when the media is subjected to metabolomic analysis. And we're comparing here a static system with the Melbourne medium at day 5 to what happens with superfusion. So I'll draw your attention first of all to these, and it's a log scale, by the way. So the number of components of the media, the known components, which are reducing in concentration over the 5-day period, and that reduction in concentration is more or less ablated by, as you would expect, the continuous perfusion of the medium through the system. On the other hand, you can see a number of what we might call waste products or metabolites here that are accumulating during the stasis over the 5-day period, and that accumulation is much reduced when there's a flow system. And this is true not only for the Melbourne medium, but this same pattern is observed for conventional media. And so I think here the RPM squared gives us the capacity to address this issue of unphysiological media. And with that, I can see that my time is almost up. So I'll just summarize that the RPM squared enables a modular, scalable system with variable flow rate, suitable for the open cell culture environment. And I want to thank particularly Brian Gowers, who's done the work on the design of the microfluidic system, and the team who've done the work on the cell culture. And thank you for your attention.
Thank you very much. In this case, we have to have the questions towards the end of the session or just privately. So we directly move to the next presentation, which will be given by Kevina Otar from the University of Central Florida. And her title of the talk is Development of a Functional Human IPS Cortical Neuron MEA Model for Long-Term Potential Analysis and Alzheimer's Drug Testing. Stage is yours.
Okay. Hi, I'm Kavina Autar. I'm a PhD candidate in the Hickman Hybrid Systems Laboratory at the University of Central Florida, and I'll be talking about the development of a functional human iPSC cortical neuron microelectrode array model for long-term potentiation analysis and Alzheimer's drug testing. So I'll just get into a little bit about Alzheimer's disease. It's caused by the accumulation of amyloid beta proteins that form plaques, and that's also theorized to have a downstream effect on the tau protein, causing the disintegration of those microtubules that will form tau neurofibrillary tangles. But there are several other mechanisms downstream because of the formation of those plaques that exacerbate neurodegeneration in Alzheimer's disease, such as cholinergic deficits, oxidative stress, and excitotoxicity. And most of these mechanisms in combination affect the synaptic plasticity of the neuronal culture and dysregulate long-term potentiation, or LTP, which is our in vitro correlate to memory and learning. This graphic just details the mechanism of excitotoxicity and how the amyloid beta plaques inhibit the reuptake of glutamate, causing downstream excitotoxicity and cell death, and decreasing our synaptic integrity. So this topic has kind of been beaten, so I won't go too much into it, but we do know that there are a lack of human-based models at the preclinical stage, and the current use of animal models does create the species gap in translatability, so there's kind of a need for human-based models. We know that because of the lack of translation, a lot of drugs fail at the clinical trial level, and that does waste a lot of time and money. So in order to try to close that species gap, we did employ the use of a human source with using human iPSCs for the tissue engineering and disease modeling. iPSCs are just reprogrammed fibroblasts. Once they are reprogrammed into stem cells, they can be expanded, and because they proliferate indefinitely, and they can be differentiated into several different cell types. So this was a source that we used to differentiate our cortical neurons in this model. Because they're reprogrammed from human fibroblasts, they retain their source DNA, so we can create disease, mutation, and patient-specific in vitro models and look at the phenotype of each of these diseases. Once those are developed, we can then look at preclinical drug testing using a more biologically relevant model. This slide also details the human on a chip, which is the other aspect of this project. So once we differentiated the iPSC cortical neurons, we employed human on a chip technology, which Mike Schuler talked a lot about this at this morning's keynote. So I also won't go too much into this, but we are able to look at multiple cell types. For this study, we really only looked at the brain, but we also used serum-free media because we wanted a defined media system where we could control the differentiation. And there are many other aspects that this system is advantageous for, such as flow and PK/PD modeling. So the objectives of this project were to first establish and validate an in vitro organ-on-a-chip model of iPSC cortical neurons to assess long-term potentiation via synaptic connectivity, and then utilize this model to evaluate the cortical neurons in an Alzheimer's disease model for drug assessment with the use of A-beta-42 oligomers. In order to accomplish that first task, we wanted to first develop and characterize a neuron astrocyte co-culture on paired pattern MEAs. We could then evaluate LTP through synaptic connectivity and confirm that this LTP was indeed synapse driven. So this figure just details the neuron and astrocyte co-culture. We did differentiate the iPSCs using a previously published iPSC cortical neuron differentiation protocol. We patterned the, we patterned the neurons on MEAs where we had 5 pairs of neuronal populations that were connected synaptically, so we could look at each of those 5 pairs individually. This stain of our co-culture was actually done on our MEAs, so we are able to repurpose the MEAs to do ICC on the exact chips that we're testing for neuronal activity. And those last 2 figures, C and D, are just our confirmation of our biological signals on our MEAs through the use of lidocaine. So our culture details are just that we pre-expanded the astrocytes prior to co-culturing the neurons and astrocytes on MEAs, and we waited approximately 28 to 40 days before we started experimentation because we have previously seen that that's when the neurons are mature and are able to be electrically active. Figure B details the protocol that we actually used. So we first were able to measure the baseline activity. Once we noticed that we had activity on an electrode, we then went back and stimulated the paired electrodes. So we didn't want to directly stimulate the neurons that were active. We wanted to stimulate the neurons that were synaptically paired to those neurons to see if we could enhance the neuronal activity of the active electrodes purely through that synaptic connection. After that, we then went back and directly stimulated the neurons that were active to see if we could further enhance the electrical activity or if they were able to fully be enhanced through those synaptic connections. And we did employ the use of lidocaine at the end of our experiments to confirm that we had biological signals in our analysis process. So this figure details what we got just from the induction. So again, we were able to stain the chips to show that we did have synaptic connections on the chips. We ran a non-stimulated control just to see if the effect that we were seeing following the stimulation was just a result of continuous testing for 5 minutes, and we did not see a significant change in our non-stimulated control over time. But we took a baseline reading, and when we stimulated the paired electrodes, we did see a significant increase in activity compared to the baseline. Then when we went back and stimulated directly those electrodes, we did not see any more increase in activity. And when we compared the fold change from baseline or the enhancement of neuronal activity from those synaptic connections versus the populations that were directly stimulated, they were not significantly different from each other. So we were able to fully enhance that neuronal activity just through the synapses. We then wanted to go back and block our postsynaptic glutamate receptors, to make sure that the LTP that we were inducing through our synapses was through the synapses. So if we were blocking the AMPA receptors, and we repeated the protocol, but we actually took it out to 1 hour in this experiment because for long-term potentiation, we wanted to see that we had an enhancement of our neuronal signals for a long-term period. So we did a baseline, and then we dosed with a vehicle control or the NBQX, which blocked our AMPA receptors. And we saw an immediate drop in activity just from adding the NBQX. But interestingly, we didn't get any enhancement of activity repeating our same stimulation protocol in the presence of the NBQX. And we were not able to get a maintenance of LTP at 1 hour. We repeated this protocol, blocking the NMDA receptors, which are voltage dependent. So we actually didn't see that immediate drop like we did when we blocked the AMPA receptors. But again, we were not able to get LTP induction or maintenance in the presence of the NMDA receptor inhibitor. Okay, so in summary, for the first part of this experiment, we were able to develop and characterize a neuron and astrocyte co-culture on paired patterned MEAs. We were able to evaluate LTP induction through synaptic activity and confirm that the LTP that we were seeing was in fact driven by those synapses. For the next study, we wanted to look at the LTP capabilities of our cortical neurons and the ability of our model to predict the effects of drugs in a preclinical setting using A-beta 42 oligomers. First, we had to confirm the presence of the drug targets that we were using. We were then able to assess the neural activity in the presence of the oligomers, but co-administered with the drugs through whole cell patch clamp, and also assess the activity on the MEAs. So kind of just going back to this graphic really quickly, I did mention that the formation of the A-beta 42 oligomers, which we dose the oligomers in our culture media so they would accumulate into plaques and cause several downstream effects in these mechanisms of neurodegeneration. So we tested several drugs that looked at several of these mechanisms, such as cholinergic deficits. So we looked at donepezil, which is a acetylcholinesterase inhibitor. We looked at a couple of secretase inhibitors, and we looked at an NMDA receptor antagonist, memantine, to target that excitotoxicity. So I'll just quickly go through the drug targets because I do have slides for that. So again, donepezil is an acetylcholinesterase inhibitor, which is supposed to inhibit the breakdown of acetylcholine and increase the available acetylcholine in the synaptic cleft. Memantine blocks the NMDA receptor mediated excitotoxicity, which is part of that excitotoxic mechanism. Saracatinib inhibits the FIN kinase, and this one's actually kind of interesting. So the FIN kinase is responsible for the phosphorylation of APP, which then leads to the production of the A-beta plaques. It's also partially involved in the hyperphosphorylation of tau, which then leads to neurofibrillary tangles. But we're dosing A-beta oligomers externally in our culture media. But even so, the presence of the A-beta, the accumulation of plaques, should actually trigger this cascade response internally within the cell. Lastly, we looked at roloprem, which is a phosphodiesterase type 4 inhibitor, which inhibits the breakdown of cyclic AMP, and it should increase the available cyclic AMP within the cell to increase the neural activity. So this graphic just looks at, again, we were able to culture our cortical neurons on our patterned MEAs, where we were able to get distinct neural populations that were connected synaptically, and we were able to do ICC to confirm the presence of all the drug targets. These are just some representative traces. We use A-beta scrambled as our vehicle control in this case for the administration of oligomers. We wanted to make sure that it wasn't just the administration of the oligomers that was causing neurotoxic effects. And we, through these whole cell patch clamp recordings, did not see any effect on the currents, the induced firing, or the spontaneous firing of our neurons in the presence of A-beta scrambled. But in the presence of A-beta 42, we did see a reduction in all three of those parameters. But we have some representative waveforms for how the drugs reacted. So we co-administered all the drugs separately with A-beta 42 for the same time point. And we did see that that co-administration was able to block the neurotoxic effects on the currents and the induced and spontaneous firing. Similarly, when we cultured the neurons on MEAs, we were able to see a very similar trend in the the neuronal populations that we were looking at. So we had our baseline and post-stimulation, and at this point, all of the conditions were exactly the same because we were doing a baseline and inducing LTP, but following LTP, we dosed with and either of our conditions, and we did see that with the A-beta 42, we had a reduction of activity, whereas with the A-beta scrambled and the A-beta 42 co-administered with the drugs, we did not lose our long-term potentiation. So I'll just be going through all the drugs separately. These are just the quantifications of a few of the patch clamp parameters for each of the drugs, as well as the long-term potentiation. So for donepezil, again, this should increase the available acetylcholine. We were able to get a significant rescue of the sodium currents and the AP amplitude, but more importantly, we were able to significantly block the neurotoxic effects of the A-beta 42 oligomers on LTP induction. Similarly, for memantine, we were able to rescue a few of our patch clamp parameters, AP amplitude and spontaneous amplitude, but more importantly, again, we were able to spontaneously block the neurotoxic effects of the A-beta 42 on LTP. The saracatinib actually had the most robust rescue. All of the patch clamp parameters were maintained all significantly, and we again were able to significantly maintain LTP. And lastly, for rolopram, we didn't see that all of the parameters were for patch clamp were maintained significantly, but this does coincide with the drug mechanism. So we don't expect that we would get a rescue of the sodium currents significantly with the co-administration of rolopram, because it doesn't affect the sodium currents. It only affects the spontaneous activity of the neuron. And this just kind of summarizes all of the drugs. So with our system, we were able to show that in all of the conditions, we were not able to get LTP because we did not have a significant change with our 1 hour LTP from baseline in any of the conditions. But with our A-beta scrambled, our positive control, we did have a significant change. And all of our drugs were able to retain our ability to have LTP by blocking the neurotoxic effects of A-beta 42. Okay, so in conclusion, we were able to develop a protocol for evaluating LTP as a function of synaptic connectivity and validate that that protocol was indeed able to stimulate the neurons and that it was synaptic. This model was applied to assessing drug efficacy and inhibiting the neurotoxic effects of A-beta 42 oligomers on LTP deficits. For future applications, because we differentiate our cortical neurons, we're able to now look at patient-specific AD iPSC cortical neuron models and look at the ability to look at LTP in those models, and also look at drug evaluation in neuroenhancing therapeutics. And with that, I'll have my acknowledgements. NIH, my advisor, Dr. Hickman, who spoke yesterday, and all the members of the Hickman Lab at NSTC. Thank you.
Thank you very much. Are there any questions from the audience?
Great work. Thank you. Congratulations. Obviously, you brought a nice group of support. Can you talk a little bit about the reproducibility when you actually differentiate from different batches? And have you tested that? And how much of the variability you get from batch to batch preparation of cells?
Yeah, so we do have to have some quality control. We do separate checkpoints for quality control throughout our differentiation. I do have a paper that details the differentiation protocol, so that's a really good point. So typically for these experiments too, yeah, we try to stick with one batch that has already been validated for QC so that we have that internal control. Any other questions?
Just maybe adding to that, the variability that you saw, do you think the reproducibility is sufficient to see in the next step the differences when you go with different patient cells?
Yeah. Yeah, like you could see even within some of the dosing with the A-beta 42 oligomers, we did have a little bit of variability there. I think usually our answer to that is just to get a lot more Ns. And usually when if we were looking at patient samples, we would try different differentiation batches that met our quality control standards to make sure that those results were consistent if we were looking at a drug effect.
Do you need to change the system or like engineer the system to increase kind of the number of replicates if you go for more patients that you're going to test in parallel? Or can you use it as it is now?
We would probably use it as is now. I feel like we usually try to do a large number of Ns, and then we try to do several experiments that repeat that.
Where's the limiting factor at the moment on scale?
Just time, honestly, to test all the systems, probably. Yeah, and then just cellular resources, manpower, probably. That would be our most limiting factor at the moment.
Any other questions? To clarify, were you asking her whether or not it was applicable to personalized medicine or more towards drugs that are could be tested over applicable to large patient population?
More the personalized one. So, I mean, she mentioned.
Okay. Sorry, could you repeat the question?
Yeah, I was just wondering, I mean, you had the questions about variation, reproducibility, and if you start with different cells from different IPS cells, if the variation that you see now might hinder to see differences between different patients, but apparently this works quite well, right?
Yeah, at the moment, yeah. And typically when we get an iPSC batch, we do several batches of differentiation to make sure that we have a consistent phenotype. Yeah.
Good. Any other questions? So thanks again.
Thank you.
So we are coming to the last talk of the session, which is given by Kimberly Homan from the IQ MPS consortium. Her title is IQ MPS Affiliates Accelerating the Development and Adoption of MPS Models in Industry.
Okay, hi. So this will be an informational talk. I'm a previous developer of these systems, and for about the last year and a half, I'm now the director of a group, Complex in Vitro Systems Group at Genentech. So I'm happy to talk about separately from Genentech. I'm part of this IQ MPS consortium of pharma companies that's helping to push this technology. So I'll start with some acknowledgements. The IQ MPS affiliate has a secretariat that does a really great job of helping all of us with things like these slides. So I'd like to say thank you to them. And they have lots of other divisions that you're probably aware of. But this particular one is an affiliate of groups that are very interested in MPS technology. And the idea is that if we get together as a group between industry, developers, regulators, hopefully we can increase the pace of advancement of these technologies and into the hands of people who really are going to push them forward for testing drugs. A lot of the representation inside of our organization is from drug safety, but there's also 3Rs representation, ADME and PK experts. A former past chair is in the room here, Jason Eckert of this consortium. Current chair is Ree Hardwick. And current membership has about 20 companies representing a lot of the major pharma companies to date. And each member company is provisioned 2 members to be on the steering committee of the IQ MPS affiliate. But any member from any of the companies can participate in the work streams, which I'll go over in a few minutes. So quickly, I just want to mention that the MPS definition as defined by the affiliate in several manuscripts here as an intro is pretty much anything more advanced than a 2D cell culture system, usually incorporating flow. And I'll kind of back up and say that the IQ MPS affiliates has a couple of goals. The first is to help serve as thought leaders, and I'll go over that in depth. to provide a venue for cross-pharma collaboration, cross-pharma data sharing, and to create engagement between industry and regulatory and really try to push this forward. I know an earlier talk in our session talked about that quite a bit. And then to develop, external partnerships to help enhance the inclusion of industrial priorities in a lot of the development. So one thing I find particularly useful is the thought leadership from this organization. So from when they were started in 2017, 2018 until now, they've done quite a bit of outreach of presentations, published manuscripts that really help drive how these systems can be qualified, validated, and what the industrial perspective is on how to qualify the systems and how that would be important for their processes. And there's a couple of work streams that help enable all of the goals that I just talked about earlier. The first one is this organotypic manuscripts work stream. Then we have a regulatory outreach work stream, a pilot team, which consists of people who are interested in actually engaging in scientific pre-competitive projects together. So we as pharma companies will get together and co-fund a study if we feel like the data from that study would be useful for all of us. Strategic partnerships, communication, reaching out to stakeholders across the industry. And then we also perform some landscape surveys to understand how are these MPS systems being utilized across the industry, across developers, understanding where are the needs currently and where are the gaps. So I'll then spend the rest of this presentation going over all of these different work streams in depth, starting with the organotypic manuscripts. So well before I joined the affiliate, there was an achievement of the 1.0 manuscripts that they like to call it, where there's 9 manuscripts, as you can see here, lots of different topics of those first couple of manuscripts. Some of them were specific to organ systems. You have an intro there and then some on ADME applications, et cetera. And Jason did a nice job of kind of outlining where those are available in previous talks and on our website at the IQ-MPS. And then there's a new 2.0 manuscripts now in current progression. There's 2 that have been published so far in this second series of manuscripts, and a lot more that are to come out of the IQ-MPS affiliate. And I would note that all of these manuscripts typically have all authors from across those member pharma companies. So the opinions are expressed as shared across all of those pharma member companies. And this is a lot of the 1.0 manuscript series, and you can see kind of the different topics that they undertook. But I think that I wanted to kind of go back for a second to the intro manuscript, because some of these messages that were written several years ago now still largely hold. Like just defining the definition of what is an MPS has actually across multiple industries been an important facet of this when you talk to FDA or you talk to other regulatory engagements. Understanding that context of use, a lot of times has been engaged in safety and in ADME, but the context of use in general is super important to qualify your system. And that characterization of system is inherent to building confidence in it. Animal chip counterparts, I'll harp on this throughout the presentation. Industry feels very strongly that having animal chip counterparts will really help give credence to the human chip results. Compound lists are available in a lot of these manuscripts, and as has been discussed earlier today, while it's not complete validation to have every compound on that list evaluated, it doesn't represent complete qualification. I mean, it does give you a place to start, and it would give you credence to your system to say, here's my context of use and here's my compound list that I've already tested in it. Gaps can be filled by MPS, and the challenges that were introduced in the beginning, I think, largely remain, which is, money, time, resources are what's hard by pharma to kind of uptake these systems. There has to be increase in throughput, which I've seen a lot in this conference so far. So I think some of these challenges are definitely being tackled. So I'll reach out to the second one here, which is the regulatory outreach. There's been a bunch of achievements in 2020 and 2021, having different workshops and webinars conducted to kind of get people from the industry together, between FDA, between the IQ-MPS affiliates, and others. And then just last month, there was a workshop between health regulators globally and the IQ MPS affiliates. And all of those are going to come with corresponding manuscripts. So if you really want to know what was discussed at all of these workshops, we always make that publicly available. And the regulatory engagement team has several branches and has really reached out in recent years. You've got this webinar series that I was talking about, deeper dives in these workshops and having a lot of FDA and industry attendance, joint publications with FDA, some that have happened and some that are ongoing. The workshop from last month, and generally engaging on what does a roadmap towards regulatory guidance look like for these technologies. This was the 2020 IQ MPS workshop that was in person, and here was the manuscript in case you want to go back and look at it. And we'll be publishing a manuscript on what happened last month as part of the workshop as well. The pilot team projects. So again, these are projects where the companies get together and define a common goal, something that we're very interested in to maybe define across model comparisons in certain organs or particular types. And the way we go about this is the IQ MPS affiliates does not or cannot endorse any one single vendor. So instead, what we do is we define a common goal we'd like to go after, and then we submit a request for information that's open to anyone to submit to, and then we down select from those. I just put it down here. We down select from those from that RFI release process to a request for proposal. And in that request for proposal, we would then consider, all of the financial implications, and then we would initiate a study. And as I kind of said in the beginning, from all of the member companies, any person in the member company can participate in this work stream, not just the steering committee members that are part of the IQ MPS affiliate. There are two ongoing larger projects as part of this pilot work stream right now. One of them is in GI models, and it was identified early that toxicology biomarkers and stem cell toxicology were two really important facets that the committee as a whole would like to look into. And so far, we've narrowed down from 8 to 5 vendors, and the RFPs are getting reviewed at this moment. And on the kidney side, we were interested to kind of join forces with CPATH and look at different biomarkers in renal proximal tubule damage. And so that one is also at RFP stage and is being reviewed to go to proposal stage. And lastly, at the bottom here, there's also a bunch of opportunity within the IQ MPS affiliate for different industrial pre-competitive sharing, that data sharing that all of us that do model development inside industry would like to save some time by helping each other in that regard. Strategic partnerships and communications. So we've got a couple of those, and I won't read all of these outright, but I would say that we are in touch with a lot of different groups, UROCs, NA3CRs. We would like to enhance that and ensure that these connections remain viable and stable. So if you're part of any of these organizations and you would really like to talk to us about having joint seminars or workshops, we'd be very interested in continuing to assess that reach here. And then lastly, I'll talk a little bit about the 2 landscape surveys that we've done. So the goal here was to identify major challenges to realizing the full potential of these technologies inside of industry. And there were 2 definitions put forward in the 2 different surveys, which makes a little bit the results a little bit muddled. It's a little bit harder to understand the difference from year to year. But there was a 2019 survey where we were defining an MPS as with having all of these green check marks here, where you would include pre-patterned and 3D microtissues in the definition. And then later in 2021, we only included printed static flow or microfluidic systems in the definition. I think the idea there was to try to understand where the barrier to adoption of those latter versions really were. And so this team has put together several of these surveys so far, presented the results. And the ongoing activity of this team is to really publish both survey 1 and 2.0 results, which is coming out soon, I hope. And if you look between 2019 and 2021, I'd say large companies formed the majority of the responses in both cases, followed by mid-size and then smaller companies. The thing that I found particularly interesting was that of all the respondents, they said that their top priority organ of interest was liver, followed by gut and kidney. And then after that, there was some divergence of the interests. But that was also, it was interesting because the IQ MPS affiliates had decided to go after as pilot projects, the gut and the kidney first, mostly because the liver was already being undertaken in a lot of those companies heavily and thoroughly internally. Okay, so the narrowed focus of the definition led to the 2021 survey having slightly less respondents, we think. The decrease in number of FTEs associated with working on these particularly challenging systems was decreased slightly from 2019 to 2021, but again, it might be because of the change in definition. I would say that some conclusions, the human MPS models that are used are awesome. And they're used mostly in safety and in mechanistic action, MOA studies. And really, if you look across many of the companies, they're being used largely for small molecules and kind of in that order that I'm showing here. So majority being used for small molecules more than biologics, and you can kind of move down the line here. And I think that there's still somewhat of a resistance to filing them in regulatory filings, which we're really working hard to overcome. And so what we found a lot is that they're being used internally, but for internal decision-making and not always going into the hands of regulators for final pending like IND applications. So 2022 and beyond, I think there's a couple of things that we would really like to focus on as the affiliate. And I think that all of these things have actually been touched largely upon today. So the idea that your input cells are super important no matter what device you're using. So how do we make those a little bit more consistent? And then your context of use is incredibly important for qualifying the system. If you have a system, it's not just going to be qualified for every potential use. You have to actually really narrow that definition. And then the animal to species translation and really pushing the idea that in addition to having your human chips available, we'd really like to have those animal chips on hand so we can do the in vitro to in vivo translation, because pharma companies have a lot of data from their animal use cases and studies. So with that, I'd like to take any questions.
Are there any questions coming up?
I do have a lot of questions, but I also don't like speakers feeling awkward that no one asked the questions. So can you talk a little bit about your work packages and RFPs? So the idea is that the vendors will actually do the work and then you'll review it rather than a third party kind of using chips and emulating what you would do yourself in the lab later if it works.
Yeah, we have quite a bit of flexibility with that. So it is mostly asking vendors to do the work in their own houses, but we've found that if we want to make cross-chip comparisons, that's not always easy to do. So part of it you can do there, but part of it we're imagining chipping in kind. So, you know, if some of that, you know, cytokines can be sent to one lab for analysis, we might do that in some cases. Yeah.
So pretty much everything I saw up there were single organ systems, whereas the future really of MPS is looking at interconnected multi-organ systems. Are you going to consider those in future aspects, or is this not something on your radar right at the moment?
I'd say it's on our radar for sure. I'd say that, you know, we have that publication on ADME that talks about, you know, linking and things like that. So we're very aware. I think that coming from the developer side, the industry tends to be a bit more conservative and really wants to see the general use and qualification of individual systems, in many cases, robustly before we move on to the next thing, right? So I wouldn't say it's not coming and I wouldn't say it's not on our mind. I think we have talked about it, but it's kind of like, let's get to step A, let's do step B. That's kind of how I've I've felt as part of the organization so far.
And are you starting to couple the in silico? Because obviously in silico has been one of the big pushes for big pharma recently with the MPS systems at this point, to be able to create the PK/PD models of the in vitro systems, the MPS, and then couple it with your advanced in silico models?
Good question. I haven't personally been part of discussions of that with the IQ MPS affiliate, not that it's not happening though, and not that we wouldn't consider future projects on that. I think a lot of us though internally are having those discussions and are already trying to do that internally with our systems and our model development. Yeah.
Hi, Kim. Nice
Hi.
talk. I was fascinated by your, especially the comments toward the end about validating these models using the animal models as kind of like integrate. I guess I've never really thought about it, but it does make sense, you know, with in vivo models. Is there a preference like mouse, baboon, gibbon, you know, like it, how would you direct, especially academic researchers to, you know, direct their funds away from a lot of the work that we still have to do within the human models and validation, also doing it simultaneously in a separate like animal model?
Good question. Yeah, it takes a village, right? So I'm hoping that developers that are making systems that pharma's adopting, that are, you know, being used more scalable, more routine, all of that, come up with these animal chips almost, I wouldn't say first, but in a way, for, you know, because they're the ones that we can get this robust, deep data set off of, and then hopefully translate that, because there's scaling factors we want to understand. There's, you know, and towards the idea of which animal, that's really going to depend. So of course, you know, mouse, rat, cyno, you know, those are used routinely, but there's others too. So I would say definitely going to need a non-rodent and a rodent species, but in general, I think it's really important for us to kind of take our robust in vivo from animal data, compare it to the chip, and then be able to do that scaling to what the in vivo human, you know, dosing and all of that should be. Yeah.
Thank you.
Thanks.
Any other questions? Maybe one last from my side. You mentioned these pilot projects.
Yeah.
Defined by the IQ consortium. Are you also open for proposal coming from the outside? I mean, clearly they need to be addressing a general problem, but maybe they can be embraced and by the IQ consortium because it is really kind of something of general interest.
That's a hard one because legally we have a lot of restrictions given that we are affiliate, and we can't then just say I would like this one vendor to do this one thing for me, but at the same time, actually I'm just gonna punt that one. I actually don't know the answer to that question really, because I haven't seen it done, and where I have seen it done, it's always an open call. So even if the idea came from somewhere else, I think it would still have to become an open call.
Okay,
like, you know, for anyone to participate.
It would be basically just the idea. I think a certain vendor would need to step back and say, okay, we are confronted with that question from, let's say, end users. Why do we not do a larger study that is kind of comparing a lot of different systems based on that, what we kind of experience, and then approach the IQ consortium and kind of say, okay, from our perspective, this could be interesting. Also including the vendor itself, but kind of in a general context. And
yeah. And those are the types of things we want to do. We want to do the pre-competitive data gathering, comparing multiple systems and knowing where the pluses and minuses are for each of them. That's what helps us all move forward. So if you have a good question, I'm sure if you brought it up to any of the steering committee members, and then the steering committee members can, you know, propagate. Yeah.
Okay, great. Any other questions? So thanks again. So we have another 10 minutes for Q&As. I mean, there are we were not able to have questions for Alistair Stewart, for example. Are there any questions or discussions to be brought up? Otherwise, I would like to adjourn this session, thanking all speakers again. The next topic or program is the poster session that will take place on the other side, and the macro party this evening. So have a nice rest of the conference and a great evening.